before we get to the talks, I wanted to spend a few moments on talking about the Association for Creatine Deficiencies, our mission, what we've accomplished, and then spend a little time on the talks in the upcoming day. So without further ado, hopefully we will have some more people joining as we go through the talk, but I would hate to take away from our lineup of speakers, so I'm going to go ahead and get started. Okay, so for those of you who are new to this meeting, ACD is a parent-led association that was established in 2012. Our mission is to provide, there we go. Apologies, getting started over here. So our mission is to really provide patients, families, and the public with education about CCDS, to advocate for early intervention through newborn screening efforts, and to promote and fund efforts for treatments and cures for CCDS. So the three pillars that you see here on education, diagnosis, and research are the pillars of our vision. And by moving efforts on each of these axes ahead, we want to achieve our goal of finally diagnosing and treating CCDS at birth. So just a quick recap about the three different disorders that are described collectively as cerebral creatine deficiencies. These are all single gene disorders arising from defects in the genes coding for these critical enzymes in the biosynthetic pathways for creatine. So you have AGAT enzyme, which is the first enzyme in the creatine biosynthesis pathway. The defect associated with mutations in this gene is called AGAT deficiency. It was first reported in 2001, and it's it's the rarest of the three CCDS diseases. Upon diagnosis, it is treatable with creatine supplementation. The second deficiency is GAMT deficiency, which arises due to disruptions in the gene coding for the GAMT enzyme, another enzyme in the creatine biosynthesis pathway. And this is the cases of GAMT are now being more rapidly diagnosed because of advances that the association has made in newborn screening. There's about 100 patients or so with GAMT, and upon diagnosis, this disorder is treatable with creatine supplementation, ornithine, and sodium benzoate. I think the critical piece that I want to say about GAMT is that it is treatable. If it's treatable at, supplementation can start at any time, but prognosis is a lot better if it's caught early. Hence, a lot of efforts are really focused on being able to catch it early through newborn screening in different states. We've made some advancements towards this in this past year, which will be shared briefly by me as well as others through the symposium. And finally, we come to CTD, which is a creatine transporter deficiency arising due to a lack of or defects in the SLC6A8, the transporter coding for creatine transport into cells. This is among the larger indications under the CCDS umbrella. There's over 500 cases that we know about. It was also first reported in 2001. It is an X-linked disorder, and so the chances of seeing it in males is higher. And it's thought to be the second most leading cause of intellectual disability in males after Fragile X. We're in a tough spot with this one because there are no treatments for CTD. Supplementation is recommended with creatine, arginine, and glycine, but it's unclear if that has an impact on the disease prognosis. So our efforts collectively are focused on advancing screening, advancing understanding of the population with these diseases, and advancing research efforts to get us to translational cures for these diseases. As I alluded to in 2 slides ago, education is a big part of what we do, and we do this through various different types of forums. Earlier this year, we had the opportunity to lead an ELPFTD or patient-focused drug development meeting with the FDA. This meeting the goal of this meeting is to inform policymakers as well as industry experts who are looking to get into this particular area, what the prevailing need is, what's the prevailing standard of care, as well as what the need is for therapeutic intervention. So this is a way for us to make our voice heard, voice of the community heard, and that's exactly what we achieved earlier this year. We had another opportunity through the PCORI funded Parents Project, an ongoing project that you'll hear about from Heidi later today, to engage with clinicians as well as caregivers, to provide a platform for caregivers to talk about the problems that are most relevant to them in their day-to-day management of the disease. This was highly educational and informative, not just for the caregivers, but for the clinicians as well, who came away with new appreciation and new perspectives and understanding about the disease itself. Not leaving our family and siblings behind, ACD has partnered with SIB shops as a way to give siblings of children affected with disabilities a platform to talk about their feelings, talk about their challenges as well as their successes. This has been, and we heard from the key coordinator of SIB shops for ACD at the Caregiver Symposium last year about how this effort was hugely significant in improving outcomes for families as a whole. So these are some of the aspects that we are focusing on as far as education and support for the community is. The next aspect that I'll touch on is the second pillar of the ACD vision, which is diagnosis. As I mentioned earlier, a big part of what we do is newborn screening advocacy, and we were lucky to have newborn screening for GAMT adopted in 2 different states earlier in the past 5 years, and the efforts resulting from or the sorry, I should say that differently. The results from those particular efforts enabled us to advocate for GAMT to be added to the RUSP, and this was officially achieved and endorsed earlier this year. The addition to the RUSP means that many of the different states in USA do seek to adopt or align with the RUSP guidelines, and so we are now able to get more states to screen for GAMT, to adopt the screening guidelines for GAMT. And this basically means that we will be able to identify children with these disorders, with GAMT particularly in these states faster, allowing for early intervention and treatment with supplementation. We're not that far for CTD and AGAD. We do not have a newborn screening test for both of those yet. However, efforts are ongoing for us to be able to understand this population better and pursue research efforts as well that will get us to a screenable point for these diseases as well. One thing that I want to mention over here is that the biggest resource that we have is our community. And to understand our community better, we collect data from them through our CreatineInfo registry. So a couple years ago, the ACD had was a pivotal moment for the ACD because we initiated our own registry. This allows us to collect data through different types of surveys from our population directly. We own and manage access to the data to collaborators, to researchers, and as well as industry partners. So owning this registry is a big resource for the ACD and the community. So we thank you for your support in that. And I encourage everyone who has not already done so to please go ahead and provide your information to the registry, as this really helps us understand the number of people affected by these diseases, as well as symptomatic presentations and challenges. There's numerous different types of seizures that are deployed, and Emily, who's our registry coordinator, will talk about this later today. I spoke about understanding, understanding patient mutations and getting to a point of screenability. So what are we doing in order to support this initiative? Just like data is a resource from the community, biosamples are also a resource from this community. So when biosamples from patients are deposited into a repository, it allows researchers to access them and perform studies on them. We from the ACD are trying to encourage researchers to access these patient samples and understand and characterize mutations and the impact of the mutations on the functions of the cells better. So how do skin cells that are basically that are obtained from a biopsy cultured into fibroblasts, how do patient fibroblasts grow? What is their structure? How do they perform when they are treated with certain types of drugs? What's the metabolic and proteomic fingerprint of these cells? Understanding these could get us to a point of being able to look for a particular metabolite or series of metabolites that could be used in a screening assay. These types of efforts will also help us understand when and what type of treatments will be effective for certain categories of mutations. So for all those of you who are on the fence, still considering whether contributing patient samples is a worthwhile effort, and I know that the barrier to doing this can be high, please reach out to us and we'll provide information on how this can be achieved. And hopefully these types of efforts will get us to that newborn screening assay for CTD as well. I'll switch gears now to therapeutic development for CCDS. A large part of what ACD's charter is, and certainly where a lot of fundraising dollars go, is in supporting research for CCDS. And so here's a quick schematic for the enzymes and the organs where creatine biosynthesis is taking place. So you can see here you have AGAT and GAMT enzymes enriched in certain areas, and then they are responsible for synthesizing the creatine, and then the creatine itself is transported through the SLC6A8 transporter. And we also know from previous studies that creatine deficit in the brain is particularly impactful. So the lack of creatine in the brain early on predisposes the brain to several types of developmental challenges that then manifest as behavioral and or capacity issues. And so there are certain types of cells within the brain that are able to make their own creatine. These are ones that have the two enzymes, and there are certain types of cells which only have the transporter and therefore are reliant on other cells to synthesize the creatine and then be able to transport it. And I'm talking about this because some of the therapeutic approaches that we are looking into focus on these core principles or findings associated with creatine availability in the brain. And so mutations that impact the net levels of creatine in an organ system or as well as the brain fall into a couple different buckets. On the one hand, you have mutations in the enzymes itself, AGAT and GAMT, which can lead to a reduction in synthesis or less efficient creation of creatine in the brain. It can also lead to buildup of toxins such as GAA, where in the case of GAMT, where the deficiency of GAMT results in the buildup of GAA, which is a toxic substrate for the brain. Mutations in SLC6A8 can result in their misfolding. So because SLC6A8 is a membrane transporter, its ability to be folded appropriately when there are mutations is challenged. So it gets stuck in other parts of the cell and doesn't make it to the membrane where it is needed to exercise its function in transport. So on the one hand, you have misfolded proteins that are stuck in certain parts of the cell. You can also have a transporter that is that makes it to the plasma membrane, but is less efficient. And because it has these folding or other, it's unstable because of the physical defect conferred by the mutation. Finally, you can have transporter that is just completely non-functional. It's non-existent. It's not misfolded anywhere. It's not making it to the surface. It's just non-existent. And so our therapeutic strategies for CCDS also fall along these couple different buckets, addressing addressing how mutations can result in disease. And we've been fortunate to have really strong partners that are helping us move our research goals ahead. And here I'm highlighting the couple institutes that we do have active collaborations with. Our approaches for treating CCDS for SLC6A fall in the bucket of small molecule, large molecules, as well as delivery. So on the small molecule front, we are looking into both novel as well as repurposable opportunities with small molecules, and with the goal of being able to stabilize a misfolded protein. We've looked at understanding if there are small molecules that can leverage a different type of transporter for increasing creatine activity. We have thus far not found one like this, but efforts are ongoing to think about different ways where we can identify something like this. For GAMT also, we have supported several small molecule efforts, both designed to reduce the amount of toxic substrate by way of AGAT inhibition, as well as trying to understand if there's a way to increase residual GAMT function. And on the large molecule side, you hear from several speakers who are working on both of these types of efforts today as well as tomorrow. On the large molecule side, we have several groups that are looking at SLC6A8 and GAMT gene therapy, and we also have a group that's looking into conferring the ability for certain brain cells to synthesize their own creatine de novo. So by essentially replacing a different gene in the creatine biosynthesis pathway. Finally, because we know that another critical component for successful therapeutic intervention is going to be delivery to the right organ and the right types of cells, we're beginning to look into efforts that help us achieve this type of targeted delivery of the type of payload that we may go with, be it a gene or a small molecule, and the like. So this work is obviously made possible because of our community. Here I'm highlighting members, the ACD board as well as staff who are, who are essentially involved every day in moving these efforts ahead. And we, this is a plug for more members of the community to be involved so that your voice can be heard. We want to hear your contribution and make sure we're addressing the needs of the community. It is indeed, we wouldn't be here today over the next 2 days without you, whether it is our community as well as our academic and industry partners. So I'm hugely grateful to be a part of this community and be a part of this effort for development of CCDS therapeutics. And hopefully together we will get to the next phase of success. We've been supported along the way by several collaborative partners. We fundraise from them, and we actively engage with them on projects that are relevant to our community. So a shout out to all of our partners who made our progress thus far possible and continue to support our progress in the future. And with that, I'll switch gears to our schedule for today and tomorrow. I've told you a little bit over the past 15 odd minutes about what ACD's mission is, and the things that we fund. But really, these next 2 days are about the people that are actively moving this field ahead with their research, with their advancements. And we have about 30 different speakers, a panel tomorrow, and many different sections of talks or categories of talks that we are excited to bring to you. So stay tuned for all of the different sections. I spoke about some of this already. We have therapeutic development with small molecules today. We do have updates from our natural history studies in the first part of of today in session 1. So you'll be hearing a little bit of that today. And then later today, some of those aspects that I touched on, which is what's the impact of mutations? What does it mean for disease? How can we understand these better? How can we identify more patients? We'll touch on that and close out the day on with reports from our registry, as well as from experts who have worked in the field of caregivers and clinicians. And then touching lightly on tomorrow, we have an exciting schedule tomorrow as well. We're going to hear from different types of CCDS models. So the gamut of organoids, mouse models, learnings from mouse models where human disease is replicated, and then gene therapy efforts in the field of CCDS. And finally, to close out, we will have an interactive panel with experts in the field who will talk about their thoughts on the therapeutic development for CCDS. So really exciting schedule, and I'm grateful to everyone who's going to spend the next 2 days with us. A couple more new features for us to be aware of. And I believe Faith is sharing this actively. We do have a translation feature available for the conference, brought to you by Wordly. So if you can click on this QR code, it will take you to a website where if you follow instructions, you will hear this conference in the language that you select. And Faith will be providing instructions for this time to time in the chat window, so people who join late can also take a look at it. Before we get started with the real session, couple house rules. Most of our talks, apart from our opening session today, are going to be 15-minute talks. We will have 3 to 4 talks in each session, followed by a panel style Q&A. Our Q&A will be moderated by a duo, and one would be a caregiver from the community, as well as the second part of the duo would be an academic. So this is a way for us to have you involved in the sessions. For those of you who are attending, please do post questions in the Q&A so that the moderators can refer to this when talking with the panel. And this is just a quick plug to say that we want this to be interactive, even though it is virtual. So please do post questions in the Q&A. Make sure you talk about which speaker the question is addressed to. Say hi and chat in the chat feature, and we will have polls throughout the sessions, which is an opportunity for us to get to know you better and for you to know that you're not alone. So please do take those polls. And with that, I am ready to hand off to Julia. So it's my pleasure to welcome Julia today. Julia is MILA'S mom, but more than that, she is a force of nature, really, for a force of nature for the field of rare diseases. To tell you a little bit about Julia, Julia founded MILA'S Miracle Foundation when she learned that her up to then healthy 6-year-old daughter had Batten's disease, a fatal genetic condition with no cure. And Julia really left no stone unturned in terms of talking to researchers, clinicians, academicians, industry folks, everybody to find a therapy for her daughter. These efforts led to the first ever drug tailored for just one person, named after her daughter, Mila Sen. And Julia is here with us today to share some details about her story, her inspiration, her raison d'etre, and her learnings and passion to move this ahead. With that, I'll hand it over to you, Julia. Thank you so much for joining us today.
Thank you, Sangeetha. Thank you so much. And thank you to ACD for inviting me to speak. I apologize in advance that I'm getting over being sick, so I'm going to be coughing. You know, I've learned a lot about your community in the last few weeks speaking with Sangeetha, and there are some similarities to what I've gone through with my daughter, and there's some differences. My hope is that just by telling you my daughter's story and our journey, that there will be some elements of that that will be really helpful for your community as you move towards you know, a potential pipeline of therapeutics. So I will start by showing you a video, a very short video of Mila. I think it's important just to, I'm sure all of you parents out there can understand, it's important to know that this is not just a name, you know, Mila, but this is actually my daughter, and give you a chance to kind of get to know her very briefly before I tell you more. And I will speak for a short time and then really hope to open up the second half to all of you to ask any questions where you feel like I could be helpful in any way. So let's just start with this very short video. Thanks, Sangeetha.
Sangeetha, we do not have audio.
Yeah, there's no audio on this, so you might want to.
We may need to change you to be the highlighted speaker for the audio to come through. If Faith could do that. Got it.
Sunshine, you make me happy when skies are gray.
So our daughter's name is Mila Virginia Makovitz. She was born on November 5th, 2010. When I think about Mila as an infant, I think about smiles and laughter and health. And
half an hour after she was born, she was strong enough to hold on to my finger, and I could almost pick her up off the bed.
when Mila was a toddler, she was a climber, she was a hiker, she was a runner. How old are you, Mila? Here's our sugar high nutter. Slide.
Very physical, always loved to ride a strider and play in the snow or sledding.
We noticed that Mila was getting stuck on her words. She would say, "Mommy, mommy, I have to, mommy, I have to," and not be able to continue. And then suddenly around 3 years old, we started noticing that her feet were slightly inturned and she was walking strangely. And then at 4 years old, we noticed that she pulled books in close. And then around 5 years old, Mila's feet started moving really quickly in this strange pit-pattering way. And then she would fall back on her butt. And every time she stood up, she would fall back and stand up and fall back. You know, we took her around to different doctors who never could really kind of figure out what was going on with her. And then time passes, and then in just 1 month, everything can change. She can no longer see anything. What's brought her so much happiness is playing with her toys and looking at her books and watching TV and playing with her brother. And suddenly it looked like she could no longer play with anything. It's so hard to see such a outgoing little girl not be able to play with other kids and not be able to play with toys and not be able to watch Elmo anymore.
Thank you, Sangeetha. It's very hard for me to see that video, even though I've seen it like hundreds of times. Feels like it's someone else's life. I don't need to tell all of you though, just how hard it is to go through figuring out what's wrong with your child. And then once you figure out what's wrong with them, what to do next. You know, in my case with Mila, as you just saw in that video, she was very, very typical. And so this was a very long process for me, being told by 100 doctors and therapists that Mila would grow out of these various symptoms that she had, even though she had been completely typical and was suddenly tripping and falling and pulling in books close to her face, stepping on her toys, and breaking them because I didn't realize at the time she couldn't really see them. She was stuttering in a way that no one had heard before. She had been hiking for, hours on end and rock climbing and skiing when she was young and swimming. And suddenly she couldn't do those things anymore. I was told that I was a crazy mom by most doctors and therapists over the course of 2.5 years, between when she was 3.5 years old and 6. I brought her around the country. I lugged my little newborn son with me by myself. I look back and I'm not really sure how I did all that. At some point when Mila was 5 years old, everything got significantly worse. And my little list that I'd been carrying around with me that said neurological symptoms, got longer and longer. And each one of these problems she was having became more complicated. And at 6 years old, I gave up. I couldn't take another neurologist telling me that it was going to be, 4-month wait to see that neurologist. And they were going to have her stack up some blocks and run down the hall to tell me that she was, delayed, but she had never been delayed before, and that it was going to be okay. She'd grow out of these symptoms. But there was a really, really rare chance that she had something which they weren't going to mention because they didn't want to scare me. And I was told that so many times that at 6 years old, I had found myself crying so much that I gave up. And I brought her into the emergency room at Children's Hospital, Colorado, which was the hospital near us. And I brought a duffel bag, and I just thought there's no way they're going to, release her. And if they see all these symptoms, and I was right, unfortunately. And they kept her. This was at the time, about 6 or 7 years ago. And at the time they kept her for an entire week, a battery of tests followed by genetic sequencing, which came back with a result of Batten CLN7, which was a rare type of an already rare, very rare disease. And I will be honest that at first I was incredibly relieved because I found that there was an actual real problem and that I wasn't making this up and I wasn't crazy. Obviously it took me minutes, probably Googling Batten CLN7 to realize that it was 100% fatal, that no child had ever lived. There were maybe 30 children known in the world at the time that had it. There was maybe 2 researchers working on an animal model, barely. And there was little to nothing known besides kind of that was the gene. And there was one problem along the way, which was that this is autosomal recessive. And so there was 2 mutations required, one for myself and one for Mila's dad, and they could only find one. And so on one side, I was starting a foundation, talking to researchers and scientists, like many of you I'm sure have done, and trying to kind of work towards, is there any hope at all? And finding that there was basically no hope, except for this little glimmer that said, maybe we're at the place where we could stop genetic disease, and that gene replacement therapy was maybe an option, but that it would be $4 million, this was at the time, and take many, many years, of which I knew I didn't have with Mila. But I said, okay, if there's a glimmer of hope, like I'm going to do everything I can to make it possible. So I started Mila's Miracle Foundation. I started putting the scientists together, reading white papers, of which I didn't understand 90% of in the beginning. And then in parallel to that, I was facing this issue that Mila was missing a mutation, and that, really honestly, I couldn't test my son, and my son was 2 years old, and I looked at him and he was the same as Mila had been at 2. And every day I just thought I was going to lose both of my children, and I could barely make it through every day. I'd cry on the floor of my closet when my kids weren't looking. And I needed to be able to test him and test both of those mutations. So that was my really strong driver to figure out what was this missing mutation, and no lab could find it. The other was that if I was going to be replacing the gene and trying to spend, raise millions of dollars and try to save my daughter's life, I wanted to be sure that I was replacing the correct gene, and having one mutation wasn't enough. So while I pushed my foundation forward, I also tried to figure out how I could find this missing mutation. And I learned about whole genome sequencing, which is more common now, although it's still not very common, and was very difficult to find any lab to do that at the time. So I reached out on Facebook and asked if anyone could help me speed up the process with a lab in Harvard that did it. And it was $25,000 and was going to take 5 months. And so I was not able to connect to that lab, but I did, as a result, hear back from another Harvard MD, PhD, who was studying actually nothing related to Batten disease. Dr. Timothy Yu was studying underlying genetic causes of autism, but he had a particular interest in difficult to find mutations. And so he very kindly reached out, he and his team reached out within 24 hours of my Facebook message, of all things, passing through the hands of a number of different people and landing on his desk. That turned into what I don't have a time to tell you all, but it turned into a very different turn in events that I had not expected. So while I was pushing forward a gene therapy, and I did actually spend almost $1 million and raise that money and put that towards a gene therapy for all children with Batten CLN7, which many years later did turn into an actual gene therapy. It was not in time for Mila. But what happened was that this unusual missing mutation, which Dr. Yu and his team ended up finding after staring at a screen for, honestly, 1 month, day and night, weekends, they couldn't find it because it was not obvious. It was not in the exons. And they ended up finding this very deep intronic mutation, which was very unusual, very hard to find, and also very difficult to show that it was the disease-causing mutation that we were looking for. Their team did an incredible job at doing that and finding that and confirming that that was the missing mutation. And at that time, Spinraza had just been approved just weeks before for spinal muscular atrophy, which is another terrible, fatal genetic disease in children, where children, are kind of born relatively normal, and then they kind of lose all their abilities, and they're on respirators and in wheelchairs when they're, 1.5, and they die by the time they're 2. And Spinraza had been a game changer and had just been approved, and it was antisense oligonucleotide, or ASO. And at the time, Dr. Yu and his team thought, well, what if we, MILA'S mutation looks like it's amenable to this approach, but we can only find one child that has this mutation, Mila. What if we made a Spinraza-like drug for her? And it wasn't easy to find mutations that actually were amenable to this. It happened to be that MILA'S was. And so their team came up with this idea. And of course, many people said, that's crazy. What do you mean, a Spinraza drug for just one person? But luckily, Dr. Yu and his team were really, really willing to say, why not? She's dying rapidly. She's losing all of her abilities. And let's see what this can do in the lab. And over the course of the year, from Mila being diagnosed and meeting Dr. Yu soon after that, over the course of the next year, an ASO that targeted Mila's very unique mutation was designed, tested in numerous independent labs across the country, showing that it was restoring health to her cells. And at that time, Mila had lost her last words. She had finally lost her vision. She had lost her ability to swallow, and so she had a G-tube. She was tripping and falling, and I was having to hold her from behind all over the course between, 6 and 7 years old. And by the time we got towards kind of the end of that first year, the drug had been, I can't even express to you how many hundreds of people from companies and academics and all over the world really rallied to make this happen, working on weekends, evenings, holidays, because they saw the hope for Mila. And Mila really had just an incredibly bright light and really drew people in. I think many people felt that it could have been their daughter or their granddaughter or their niece, but also because they saw the potential of this for medicine. And they saw that this was kind of the ultimate precision medicine that people had been thinking about and talking about for decades, and that maybe this was possible. And so people, just an enormous amount of people rallied. And within 1 year of meeting Dr. Yu, 1 year of Mila being diagnosed, we moved to Boston, and Mila began receiving this drug, which I didn't even know until I think maybe days before that they had named it MILA'S after Mila. And I think only when Mila started receiving that drug did I realize it was the first moment I had to breathe and to stop. And I think only then did I realize, I was told that we were 10 years away from stopping Batten disease. And it was incredibly complicated and devastating, and that they weren't even close to that. So for me to be walking through the hallways of Boston Children's Hospital while Mila was receiving this genetic treatment, I don't think I really realized what a big deal that was till then. And then in those coming kind of weeks and months, I think I realized what a big deal it was for science and medicine as well, that, I didn't know, and that we weren't trying to be the first in the world to, receive an individualized medicine, but to learn what a big deal this was, and to learn what the potential was across, all rare diseases was, really powerful. And I felt a really strong obligation to do not only everything for my daughter, but to kind of explore what this meant for rare disease as a whole. And Mila did incredibly well in her first year, despite the fact that she was spiraling downward. Just 2 months before she received Milasen, she, started having seizures. She started, really just declining and not being able to hold her body up, not being able to swallow anymore. She wasn't able to push her little stroller that I would walk behind and hold her. And she was her legs were crumbling, and she wasn't laughing and smiling as much as she had been before at the same kind of places in the songs and books that I always read her and sang to her. And that was by far the most devastating thing. Really, just looking back, it's hard for me, because I think, what if she had received Milasen 2 months before she kind of had gone off this cliff typical of Batten disease? But there's not a lot of use in looking back at that. What there is use in looking at is that despite that rapid decline in a neurodegenerative disease, what I saw in the first year was incredible. And that MILA'S, she stabilized, which for a neurodegenerative disease is improvement. And to see her seizures that were 30 a day, lasting 2 minutes, go down to nothing, pretty much. There were nothing many days, and other days, a few little myoclonic jerks. That was obviously extremely helpful in our quality of life, but other little, that was what was ended up being kind of the outcome measure that was eventually published on by Dr. Yu and his team in the New England Journal of Medicine. But there were other things that were extremely important that may have been subtle to others, but those parents listening now can appreciate that sometimes the littlest things are the ones that matter. And for me, her holding her body up straight a little bit more meant that I could hold her from behind, and she could, her legs were stronger, and she could walk up the stairs with with both feet, and with me holding her from behind. It meant that she could swallow better. And when she swallowed better, it meant that she could eat by mouth again, just pureed foods, but she could taste food, instead of it being pumped into her stomach. She was smiling and laughing more than she had been, before, at the same spots she thought were hilarious, like when people sneezed or babies cried, in books or in stories. And so, the first year was shocking. I didn't know what to expect at all going into it. And I had a pretty good set of expectations of not having too high expectations, but that we had to give it a shot because it looked so promising in the lab. And in the second year, Mila was not, it was unclear how stable Mila was. She was having some days that were really not as good as others. And by the third year, Mila started having problems relative to her disease that I didn't even think about. Her hip started coming out of the socket. And I was given options of morphine for the rest of her life, or breaking her bones and putting them back together, neither of which were things that I would ever put my daughter through. And so I was just faced with just the worst decisions a mother should never, ever have to face. And 2 years ago, after 3 years on Milasen, Mila died. And I work here from her bedroom because it's the place where I can be closest to her. And I don't know how it's possible that so many other parents go through this. And in the meantime, I've also learned from many other families who've reached out to me the pain of continuing this with lifelong debilitating diseases. So even though I've lost my daughter, I've really started to appreciate from other parents what it's like to lose your child or feel like you can't connect to your child for their entire life, even if they don't die when they're 10 years old. And, You know, the reason that I am here now, the reason I have dedicated my life to this, and that I spend every waking moment trying to push forward and create a better path to treatments, is because I saw with my daughter the potential. If Mila had received Milacin 2 months before, 2 years before at birth, I feel very convinced that her life would be significantly better and she'd be alive now. It's hard to know, and we'll never know, but I do know that she showed what is possible. She showed that, you know, we can now find the underlying genetic cause, not just the gene, but you can find the mutation. So you can find exactly and pinpoint what's wrong, and you can target a medicine to it independent of the number of people, even if it's unique just to one person. I think her story points to a future that is very different than what we face today in drug development. So instead of one drug for tens or hundreds of thousands of people, Mila's story kind of points to a future of maybe tens or hundreds of thousands of drugs, each for, you know, one or very few, 10, 50, 100 people. I think the number is less important. It's more the concept, you know. And what really keeps me going is that we don't have great statistics, but, you know, if you look at global gene statistics, it points to 60 million children who will die before the age of 5 from a rare disease. And that doesn't include the many, many communities of children who have lifelong, very debilitating diseases that are so difficult for them, and their quality of life is extremely reduced, and their siblings and their parents and their grandparents and their community. And this to me is like a global health crisis. You know, it's a silent pandemic, and often literally silent, with children like my daughter who couldn't speak at some point. And, you know, I think about the fact that we used to not have the technology or the science, but we do today. So yes, we have to perfect it, and we have to learn a lot more about it. But you know, we need to learn about route of administration and dose and frequency in the case of ASOs. But we have, in my mind, a list of tens of millions of children on the left, and we have a growing list and kind of maturing modalities on the right. And there is no connection between those unless you happen to be part of a disease that's, you know, 2000 and above and is commercially viable. And I feel like the only way to connect children with like technology that exists today is either a proper clinical trial or like compassionate use. And neither of those are fit for purpose. They are not helping us connect children to technologies that actually exist. And to me, this is a kind of like a moral imperative. You know, it's not whether we can do this anymore. It's kind of whether we will. And what's missing in the middle here is like this infrastructure. It's this very rapid, expedited kind of clinical path from early diagnosis to rapid treatment. There's what's missing is the regulatory path, which is very central to this, somewhere between a middle path between proper clinical trials, you know, traditional clinical trials and kind of compassionate use, pulling elements from both, but kind of discarding other ones that are not necessary for this approach, and reimbursement. You know, right now, this is a purely heroic academic effort between academics and families, you know, raising, you know, selling lemonade to raise millions of dollars, working with academics who are trying to get grants and to supplement, you know, what the families can't pay for. And that is not sustainable. It's not scalable. It's not right to be asking parents and academics to also become, on top of their many other positions and responsibilities, drug developers as well. And so I think there needs to be a shift towards kind of an academic, commercial, you know, collaborative model. And it's really not, you know, Mila gets written up about and talked about a lot as an N of one. And I try to kind of stay away from that term because it's not, in her case, we may never find another child that has her same mutation. So she really may truly be a one. But that isn't true in most cases. Like, we are going to be able to better diagnose, and we will likely, what starts as a one could end up being many more. And I think that, you know, really we need to build an infrastructure that allows for a connection between these children, our children, and the technologies that exist today, of which more and more are filling up that toolbox. And there's a lot of programmable modalities in there. And why that's important, when I say programmable, what I mean is you can kind of swap out the ASO. So the whole process stays the same for Mila or any other child, but you swap out an ASO with a new one that targets the next child's, very specific kind of, mutation. It's not, I'm oversimplifying that. There's a lot that needs to go into getting to that point, but the idea of a platform or a process approval, as opposed to a product approval, is the way that we're going. And that's a lot of what I spend my time on, is trying to shift to a model that allows for programmable medicine. So we don't have to go gene by gene, which I very much appreciate the gene by gene approach. Gene therapy is amazing, and thank God we have it. But eventually, to kind of conquer these 7,000 to 10,000 rare diseases, we do need kind of platform approaches or process approaches where the whole process stays the same and is approved, and the regulator is not looking at every single molecule. Otherwise, we'll never get there, and we'll never help. It'll be thousands of years before we help our children. So some of this, pertains to your community, and some of it doesn't. I think mostly I tell this because I want all of you to kind of, I hope we can all start kind of thinking in the same way. And I think that the on-ramp, I talk about getting from Mila to millions, and the on-ramp of getting from Mila to millions is going to be the hard part. And we're on that now. We've been on it for 5 years since Mila was treated. And there's been a lot of progress. And there's also been, I wish we could have done, 10 million times more than we could have. And I think we can. And so I think that for the parents, pushing the urgency and really believing that you have the ability to play a really important role. And I know many parents out there probably can relate to this, but, there's a lot of parents out there. There's parents that are also academics and physicians, and people in biotech, and regulators are also parents. We're not just parents. We just have extremely strong motive to learn quickly. We cannot become overnight MDs and PhDs, but we can certainly find, the places where we need to do things better, and can hopefully learn and contribute to making this process expedited. So, I think that the risk that we are willing to take on in these early years, it's harder when the modalities are maturing, but they are maturing, and we are able to learn more about how to, I'm losing the word here, is be able to monitor. So monitoring children like Mila after they are treated, knowing what sort of trends we're seeing in adverse effects and dose escalation. There's many things that we can do. It's not black or white, don't have an access to a treatment or have access and something terrible could happen. We're learning a lot about this, and there are safer ways to get involved. And so I think that the risk that a parent is willing to take on in these early years really depends on that family and that parent. For me, as you saw with Mila's story, it's black and white. She was going to lose everything. So the risk of not treating Mila was losing all her abilities and death. The risk of treating her was 30 years of work going into ASOs in animals, and then more recently in a lot of humans. And to me, that seemed like very worth it. And it's really up to parents to speak up here, because physicians, academics, regulators, for good reasons, are very concerned about doing more harm. I think that the parents need to really step up and really express what amount of risk you're willing to take, because it is your child and it's you. And together as a family, you need to decide what you're willing to try, because otherwise the other option is no treatment. And we're getting better at these, and we're learning a lot. So I think that that's another hot topic to really talk about, because without that, we may not be able to get up this on-ramp. I would love to stop there, and I'd love to take any questions that anyone has. I know there's a lot coming in, but I will let the moderator or Sangeetha feel free to help out with any questions if anyone has any.
Julia, thank you so much for sharing your story. That was moving beyond words. I know I've read the written story, and certainly read articles on this, but it was very different to hear it from you. Very different to see the video and see what Mila had to go through. So thank you for sharing that. It is so powerful to be able to, no matter what you read about a disease, in in literature, it's just so much more powerful to see what actually happens in kids, what happens to kids, and to share the perspective of what it is to watch those abilities that she once had go away day by day. Thank you so much for sharing it. I really appreciate and grateful for the fact that you're here today sharing the story and still continuing to work in this field and moving things ahead. It means a lot. It really does. I want to invite Dan Collar and Randy Allen. Both are parents of children with CCDS. I'll give them a minute to introduce themselves, and they will be moderating and funneling questions your way for this session.
Great. Thank you, Sangeeta.
Good morning. I'm Dan Collar. I'm the board chair for ACD, and I have a son with CTD. And thank you so much. I'll be honest, I'm trying to hold it together a little bit. Lots of questions, and really appreciate your presentation.
And Julie, I'm Randy Allen. I have a son with CTD, Creatine Transporter Deficiency, and yeah, thank you for sharing your story. It's pretty, when you're able to kind of zoom out, and the fact that you and Mila can really transform healthcare for folks like us is pretty special. So thanks for blazing the trail, and we'd love to get to some questions for you. We got a lot of them. So one of the first ones that kind of came up from the group was like, do you have a background in science? I think people know that you were kind of a business owner, but did you kind of learn on your own, or did you have a foundation?
I did not have anything. I don't even remember taking science practically, besides 7th, 8th, 9th, 10th grade or something. No, I think like many of us, and I'm sure both of you can relate to this, is you do your best to kind of like start with a white paper and be like, how in the world do academics read these all the time? Because I can't even get through one. And then eventually you start catching on, So I think watching my daughter lose her abilities so rapidly was a very, very strong motivator to figure out how to read white papers and how to figure out what lysosomal storage diseases were and what was going on in my daughter's cells as much as I could, And so I've been very, very fortunate through this process to be surrounded by many amazing academics, physicians, researchers, and I just try to be a sponge, like I'm sure many of you guys have learning.
Okay. And, obviously looking at your journey, you've raised a lot of money to push us forward with kind of, a arm in arm support from academia. Walk us through that. I mean, is that something that was a new daunting task for you as much as white papers? Is how do I raise money to push this ahead?
Yes, I feel like overnight I had to become like a scientist, pseudoscientist, and pseudo fundraiser, of which I knew nothing about either of them. And I'm sure many of you can relate to this too. I mean, yeah, it's extremely daunting. I remember one day when my daughter was declining so rapidly that I knew that I felt like I needed to do this all in a year. And when I did the math, thinking it was going to be gene therapy, like I didn't have a number. I just was being told kind of $4 million. And at the time, MILA'S was not on the radar yet. And I remember doing the math one day, which was the worst idea ever, to take $4 million and divide it by like 365 days. And I'm not even going to tell you what the number is, because I was like, I remember falling on my floor just in desperation in my living room and being like, I think it was like $10,000 or whatever it was. I was like, there's no way I can do this. And so I think for me, what I learned was I didn't personally have the money to be putting into it. What I had was Mila's story. And for some reason, people were being very moved by Mila's story, and they were very moved by Mila. Mila really was a very radiant child from like the day she was born. And I say this not just as her mother, but many, 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 many people around us. It's just, she just radiated something that drew everyone in. And so I had Mila, and I had a story of extreme desperation of a completely typical child suddenly like spiraling downhill towards death. And I also had hope, and I really pulled on that hope. And this is what I really encourage everyone to do, is that it's easy to listen to people that are really deep in the weeds in this and the scientists and researchers, and this is not meant at all as a negative thing, is that understandably they know a lot more than we do. And they know that there's a lot of barriers still to get through with these technologies. But as a parent, I saw this as hope, which it is that there used to be no modalities. There was nothing you could do. We couldn't even find the gene, but now there are modalities which are maturing, and every day that passes through animals and through humans, and we learn. And so to me, that was hope. So I kind of really, really tried to express both the desperation and the hope. And I tried to tell it to, I didn't want to talk to anyone. I didn't want to go out to dinner. I didn't want to talk to anyone, let alone let news media into my house, who are airbrushing my face and asking me to repeat the same thing 50 times. Like it was miserable doing that. But I knew that if I didn't tell MILA'S story, the desperation and the hope, that I would never get there. And that's really kind of what I did, just as much as I possibly could.
Got it. Sorry, Dan, I'm going to hijack this. You can hop in next. Kind of mentioned you became a PhD overnight, became a fundraiser overnight. So now you kind of go, okay, I understand it. I'm going to raise the money. How difficult was it for you to convince researchers to actually work on MILA'S mutation?
Yeah, it's a really good question. I would say, I first started with gene therapy. And when I started with gene therapy, it was hard to try to get who I wanted to be the best kind of gene therapist at the time, Stephen Gray, to take on my case compared to the many others that were being pitched to him. That was really hard. And I decided to use the little bit of money that I had raised, the tiny little bit, to put towards a scientist, and have her help me kind of work with a bunch of different companies that needed to be involved for gene therapy and for the gene therapist, for the scientist as well. And eventually got him on board to work with it. In terms of Tim Yu and then the MILA-SEN approach, that one was, it feels like kind of like luck, in the sense that Tim Yu so amazingly stood up to help me, and he and his team, I think it was a combination of genuinely just wanted to help me find a mutation. They weren't even thinking about treatments. They just were like, this poor family is missing a mutation. Their daughter's dying rapidly, and they wanted to help. And that was part of what their lab did, was look for difficult to find mutations. But I think that it happened to be that the stars in many ways aligned, which is very weird to say when your child's dying, but in the sense that her mutation was very perfectly amenable and her disease was very perfectly amenable. And where she was in the disease was actually a good fit, in the sense that she still had very much of herself, and she was laughing and smiling, but was going downhill quickly. So she checked the boxes that were needed at that time, and Spinraza happened to just be approved. If it had not been approved, we wouldn't even be having this discussion. So a lot of boxes were checked, and I was very lucky that MILA'S case was a very good case. And I was told from the beginning, expectation wise, the doors may not keep opening. And if they shut, we need to stop, but they kept opening. So I felt lucky there. So I think I was very lucky to work with a really amazing group of people that were very dedicated to this. And it's not easy to find that. And it's one of the struggles I have with other families that ask me advice.
Thank you. I think there's a few more, let's say, scientific questions pending, but one that's come up before then is, there weren't many, but did you connect with any other Batten disease families when you're on this journey? And it sounds like you continued the path solo. If you can talk about maybe the whys and hows of that, would be appreciated.
Yeah, sure. Yeah. So Batten is a funny disease where it's actually not one disease. There's 14 different genes that all are kind of under the umbrella of Batten disease, which is a little controversial. The symptoms are very, very similar. So the onset is different. And the order of symptoms is different. MILA'S Batten CLN7 was very tiny, and there was 30 people in the world, like I mentioned, that had it. There was no other foundation around. And so when it came to Batten CLN7, which is what, many of us start with our specific gene, what I started on in the gene therapy, there was no one else out there doing anything. So I felt alone. I did connect with as many of those parents as I could, and I still know them. And many of them have lost their children since then. And, I reached out within my community. I then reached out to the other Batten disease. I think maybe there was 5 or 6 other foundations of the kind of more prevalent it's a big word, Batten genes. And I know all them well, they're my good friends, you know, and I connected with them, and we tried to find ways to overlap. I will tell you, this is actually a very critical part of the story, is that we are all doing this by ourselves in like silos. And we've heard this word silos a million times, and it's true, right? So like everyone, the next person starts from scratch and does it all on their own again. And so we all were running kind of parallel efforts, very specific to our genes. When MILA'S story kind of took this turn and it kind of became this individualized medicine approach, what I pushed for now, my foundation is not, of course it focuses on Batten as well, but it is really shifted now to be promoting individualized medicines across diseases. It's not about Batten, it's not about ASOs, there'll be other modalities coming down the pipeline. So I'm trying to shift away, because I think that a cross kind of platform approach is the only way we're going to kind of get anywhere. So I still stay very much in touch with them. And my hope is that soon our individual efforts will be, you know, pushed down kind of a funnel, which is appropriate for all of us, and not just start from scratch again, which is what was happening within the Batten community.
I think members of our community can relate to a lot of the things that you just said. And when I think we feel a little small and alone in the world, hearing a story where it's even smaller is a little mind-opening. There's two questions I'm going to kind of combine here, because I think the answer might be the same. So I'll read them both. The first one is, when the FDA approved Millicent for trial on your daughter, what data did the FDA want to see to allow you to continue treating her? And the second one, first of all, says, thank you for sharing this powerful story. Can you tell us a little more about moving towards the platform process approval versus a drug by drug approval?
Sure. The data that, you know, so this had never been done before. I mean, it makes me kind of laugh, because Tim Yu called the 1-800 number of like the hotline of the FDA. He didn't have any special connections or anything. And when he ended up speaking with them, MILA'S case happened to fall in CEDAR under this group, this inborn errors group, which I don't know, you know, which group you guys tend to fall in terms of your therapeutics, but this was not neurology, which tends to be significantly more conservative. So we lucked out in the sense we had an amazing group in the FDA that was understood this had never been done before. And, you know, there was a government shutdown the day that they gave the letter of approval, and they did it like minutes before the government shutdown. So they were really, truly not the barrier. They were absolutely incredibly helpful with us. And they wanted to see this happen. They came up with an agreement of dose escalation in the animals simultaneously in order to get going. So Mila received kind of fortunately and unfortunately a very low dose and escalated over a course of the first 3 months. That could have been a big difference for Mila, of course, if they hadn't done that, but that was the only way to get in, where they were dose escalating the animals just in front of Mila. So that was one of the agreements that they came up with. Mila's seizures started significantly decreasing pretty quickly. And that was in itself enough evidence to show that something was happening and it was good. There were other things that I was seeing and that other clinicians were seeing as well. But that one in particular was, I think, strong enough because it was measurable and meaningful, kind of, you know, the intersection of these two, that I think that was enough for them. And Mila continued to do, you know, very, very well. She didn't have any adverse effects at all throughout the whole 3 years. And I think I also played a role in that, in the sense that I felt that even towards the end, that in the worst case scenario, Millicent, you know, we tried to stop Mila's disease, and it did for a while. At some point, we realized that too many kind of genetic dominoes had already fallen before she began, so that we were kind of in a limbo. That limbo was an expectation, which I think Tim, you did a very good job of explaining expectation-wise, where he said, you know, from the beginning, and this is important for all the parents listening too, is that there's a range. It's not like it cures and it's amazing or it doesn't work. You know, there's quite a range of what can happen. And I feel like the word limbo is the word that describes a lot of treatments right now that are happening, because we're learning still. one day we may learn that Mila received 100 times too little of the drug. Very, in my mind, I always feel like that's kind of what I felt like. We had no reference of what the dose was. Maybe she should have not received it intrathecally. Maybe she should have received it in, ICV. I don't know. We won't know that. But the point is that I knew that we weren't there with knowing about route administration, dose, frequency. We played with what we could, frequency being one of them. So instead of being 4 months, we went to 3 months and then to 2 months, and we dosed more frequently in the beginning. We did what we could, but I felt like I still knew that it was early years, and we are still in the early years where some symptoms could be better, which they were, and some symptoms were not better. And so we ended up in the middle. And I think that expectation of limbo was really helpful. And the FDA knew that. And I was able to continue pushing and saying, in the minimal, it's keeping Mila's life, like her seizures, under control. And her quality of life is relatively stable compared to being on 9 seizure medicines, which she was on 0.5 ml of 1 seizure medicine instead. So my voice was important here of keeping her on Milasen, even at the point where they were like, well, I don't see any adverse effects, but I wonder if this is working kind of at the end. And I said, if you take her off of this, it could be that it greatly decreases her quality of life. For the second question, you ask about moving from, just let me make sure I remember it well, as moving, the comment I made about moving from kind of the product to the process approval. Is that right, Dan? Okay.
Sorry, yes.
Okay. So and what was the exact question on that? Was what my thoughts are on just
Can you tell us a little more about moving towards platform process approval versus drug by drug approval?
Yeah. So, I think the premise is, as a parent, is there are children that are dying and sick and going to have lifelong debilitating diseases or die early. There are technologies that could help them. We need to allow access. And right now, the current product by product, one medicine at a time for rare disease is not going to cut it. It's not ethically okay. It's going to take us, according to NCATS, who's done the math, 2,000 years to do that. So knowing that that's not possible, and knowing that the FDA doesn't have the resources to take on hundreds or thousands or tens of thousands of individualized drugs that are, maybe for 1 or 10 or 50 or 100 patients, we need to move towards a process approval where, an example I'll give you is if we put up kind of guardrails and say, starting with ASOs, if you use one of these, 3 chemistries that have been used clinically and been used in patients, and if you, use accredited partners, whether they're academic institutions or individuals or companies, if you stay, patients have this certain type of mutation or certain type of disease, if you stay within these guardrails here and everything kind of stays more or less kind of in the approved process, then you should be able to go from diagnosis all the way to a patient-physician consent rapidly if everything stays the same. And that it's kind of a process approval or a platform approval, so that in the case of ASOs, if you're swapping out an ASO, as long as you're staying within those guardrails, you should not have to stop at any point of the way. You can certainly submit data. And there are other countries like the UK where they have like a do and tell policy, which I think is great, where you can submit data, but you don't have to stop and wait for approval to keep going. You just keep moving. And the only reason you would stop was if something, was brought to the regulator's attention that was so disturbing that they had to stop that. If we don't move from the product to a process approval, we will not ethically be able to say that we're giving, allowing children to access technologies that look like they could help them. There's just no way. We don't have the resources, and we don't have, the system never developed. The regulatory system never developed for this paradigm. And so one analogy that I give is like, if the drug development system is a house, like one could say, I'm not going to make any comments on this, but one could say that the drug development house, let's say, works for large indications. It does not work for small indications, whether it be 1 or 100 or 1,000 or whatever. It doesn't work. It was not designed for that. And so we can burn the house down, which sometimes I'd like to do, but no one likes to burn a house down, so we won't do that. We can also renovate the house, which is what's been trying to be done for the last 5.5 years since Mila was treated. Renovating isn't working. It's just not equipped for it. In the last 5.5 years, 10 to 15 children have received individualized ASOs. That's it. There's people, popping champagne bottles, but that's ridiculous. And we had 4 FDA guidances come out in 2021. Since then, there's been 5 children treated. I started the N of 1 collaborative with Tim Yu and many others, and there's hundreds of academics and companies around the world raising their hands to do this in 2021. I started a company to create a viable business model around this in 2021, and yet almost nothing is changing. So the only way to do this is you build a house next door, and the house next door is in response to new technology of how to find patients and how to treat them. And it is very different than how we've done in the past, and it requires a different way of doing things. So that's, I could go into more depth of this, but I see this path as a new regulatory path in the middle of traditional clinical trials and kind of one-off compassionate uses. This could be a mainstream way, not just a compassionate use one-off, one at a time kind of approach. So I don't know if that helps, but.
That's great. Okay. Kind of shifting back to like the family side of it. Obviously, when you have a child with a rare disease that's fatal, having you and your husband on the same page is critically important. Can you talk a little bit about kind of the process?
I can start by saying I'm divorced. Sorry, I have to laugh just because, a lot of parents out there, I shouldn't be laughing, but like it is extremely difficult. And I would say I was not on the same page with my ex-husband. And I think that's very common. I think that in my case, just the stress of MILA'S situation, and it's true for many parents who've reached out to me, I think it just forces to the surface the difference in how people face things in life. And in our case, I wanted to fight. And in the case of my ex-husband, was not thinking that was worth it for Mila. So it was very, very difficult. He was on board for doing Milacin, and he understood the concept of it, and he, consented to it and did all the things that you have to do in a treatment. It wasn't that he didn't agree to the treatment, but it was that we faced this devastation, our life falling apart very, very differently. And I think there are many parents reach out to me actually to ask about this. And I think in some cases, it's amazing to see 2 parents that are on the same page. And I just admire that because it is so hard with the stresses of what you're faced with. In other cases, in my case, I felt like I had to put my daughter first, and I had to put blinders on for everyone. My community, there's parts of my family that were just like, give up, and in my case, I saw hope. So I, went for it. And I don't want that to come across in any incorrect, cold way, but I think just to say that it is very, very hard to align. And if you can, absolutely do that, because those families that are doing that are doing amazing things with both parents on board.
No, thanks for sharing. It's not cold or hard, it's just real, and I think you just make a great point that it is difficult, but this is our time to really stand for what we believe in and what we want to fight for. And then, on a lighter note, Mila's brother, how's he doing?
Thank you for asking about him. Aslan is 9 now. He lost his sister when he was 6. And he was actually 5, just about to turn 6. And I thought his life was going to fall apart because I had no time for him. And I know many of you can relate to this. He didn't learn to ride a bike. He didn't learn to swim. I couldn't read books to him at night. I was crying, trying to get Mila to go to the bathroom with an enema every single night. And I would be on the floor for 45 minutes while she was in discomfort and pain. And Aslan was asking me to tell, he made a Lego he wanted to show me or read a book. And I just had nothing left in me, and I just was irritable and I felt terrible all the time. And I just wanted to spend time with him, but I couldn't. And when Mila died, right before Mila died, I spoke with a mom. I spoke with a number of parents who had lost their children, and I asked them how they did it, how they survived, because I didn't know how I was going to survive. And I knew that her time was kind of coming. And I remember one mother telling me that she'd lost her daughter around 6 or 7 years old to a rare disease. And she told me that her son thrived from the day that her daughter died. And I remember being like really shocked by that. And shocked that she was even telling me that. And then, but I digested it and I was like, and she said, look, he finally could live. And I thank God she told me that, because that is exactly what's happened with Aslan, in the sense that he's gone through waves of being like really into rare disease and telling anyone at the coffee shop who asks him like all about Batten disease and rare disease to like, you know, and that his sister died and these poor people that met him like 30 seconds ago with their family, like don't know how to respond. And then, you know, other times he doesn't want to tell anyone and he says he's an only child and, you know, he doesn't know how to respond to that. But I would say that, that I was really honest with him. I didn't know how to act with a little child like that. And I had some, lots of different advice. And I went with the advice to be completely transparent with him and ask him what he wanted to know, about the disease and about the end of her life and what was happening. And I told him things that I cannot believe like a 5-year-old would have been able to hear. I can't wrap my head around. I was playing in the dirt, you know, with animals when I was that age. I can't imagine hearing what I had to tell him. And he wanted to know a lot in spurts. And I just said, you know, I will tell you everything and you can say whatever you want to anyone. Know that other people are going to probably judge you for what we're going through with Mila and the decisions we're making. And that's up to you, but just know they haven't lived what we've lived. And many people in this community know this, is that until you're in this situation, you don't really know what it's like. And so as a result of being transparent, it was the best thing I could have ever done, because there is nothing that I hide about anything in my life, with the sadness that I feel, the happiness that I feel, the decisions I had to make, our divorce. I mean, all these things, I just really honest with him, and he is thriving. And finally now, after going through his parents getting divorced, you know, 1 year of school when he first started, then COVID, then his sister dying, 9 months later, his grandmother, my mother, dying, you know, he finally now is like, you know, he's in 4th grade and he's like got a whole school year in where he could like decide whether or not he talks about rare disease, but he doesn't have to. And I see he's just becoming this amazing person for what he's gone through. So I thought siblings of children with rare disease like had the most horrible life. And now I see like, no, it can be that he will grow up to be an amazing person because of what he's seen and lived with Mila, with me, with how our community has rallied. Like he's taken it all in, and I think it's going to make him an amazing person, you know, which I'm very grateful for.
Beautiful share.
Thank you. I know that myself and many other parents who have children with siblings, it's one of the major stressors in how we raise them properly. And I'll just do a quick plug for SIP shops for everyone listening. One of the services we offer, it's a really good tool. I know we're coming up on time here, so if you could maybe quickly, I don't know if you can quickly answer this question, but the last one, did you talk to any pharma companies about making drugs for Batten? And if so, what was that like?
I didn't have to really go down that route too far. I did have a gene therapy, like I mentioned earlier, which ended up going to an academic institution, which spun off company Taysha, which was not a pleasant experience. They didn't end up taking our gene therapy fully, but they did somehow seem to be using it a lot in press releases and all sorts of other things. I think that there's a common trend right now, which is very, very disturbing, about you know, all the companies out there that, to give them credit, are trying to come up with some sort of viable business model around rare diseases, which we know is very difficult. And they're coming up with these portfolio approaches. And I see what's happening, as many of you have probably seen this, is that they end up getting shelved because another disease pops up that looks slightly more like it might be more of a blockbuster, or, you know, more children, or what, you know, whatever. And their investors, you know, promote that, and then they, you know, they decide to shelve one of the other programs. This is happening left and right. I barely know anyone. I know many, many families who've started treatments and worked with biotechs across other, well beyond Batten disease. And I would say almost all of them have been shelved. And to me, that is extremely disturbing. Now, I think that throwing the companies under the bus and throwing mud at them and destroying them is not going to help anyone, because I firmly believe that if we do not have companies, hence why I started one of my own around individualized medicines, is if we don't have them, this will never scale. Like, academics are amazing. Thank God they are doing this. Thank God. Without them, I don't know where we'd be, but we cannot scale this. There's not enough capacity. There's not enough money to do this at scale. So we need companies involved. And My hope is that we switch to a new model where it's not just, you know, a portfolio model. My hope is that, like the company I helped start, is that there's, and this is not a plug for them, this is what I hope sets the stage, like Novartis did for CAR T, is that there's someone that kind of like sees the future and kind of is willing to forego financial gain for many years to try to create a model where there's many, many, many different drugs and potentially a much smaller margin on each one, covered by, you know, reimbursed by payers. But there's many more, and you can push all of them through. You don't have to deprioritize and prioritize just the top 2 and then get rid of the other 8, which is kind of what's happening right now. So not a great model right now, but there is hope, I hope, you know, for the future.
Thank you. Thank you so much, Julia. I'm gonna thank our moderators, Dan and Randy, for going through that session. Thanks for making time for us today. I know we had a lot more questions that we weren't able to get to, but I'd love to be able to send them your way and share an update with the community. Thanks very much for sharing your time with us today.
Yeah,
thank you. Great kickoff to our session.
Yeah, thank you for letting me tell Mila's story, and I'm happy to answer any questions offline later. Thank you.
Great. At this time, I'd like to introduce our next speaker. So our next speaker is actually Dr. JUDI Miller. I think you have heard from her before. But before JUDI gets started with her talk, I believe Melanie Brandenburg will be joining us briefly for a forward. And I want to take the opportunity to introduce both of them just really quickly. So for context, Dr. Brandenburg is a senior medical director in the global clinical development team at Ultragenyx, where she works on the development of therapeutic agents for neurodevelopmental rare diseases. And Dr. Brandenburg will be sharing a few, few words on the update from Ultragenyx before we get to Dr. Miller's talk. Dr. Miller is a psychologist at the Department of Child and Adolescent Psychiatry and Behavioral Sciences at the Children's Hospital of Philadelphia. Her areas of expertise include autism spectrum, neurodevelopmental conditions, and quality improvement. She has been involved in the Vigilan natural history study that was taken over by Ultragenyx. And so together, we'll hear a little bit about where things are for CTD. Thank you both for joining us today. Melanie, all yours.
Thank you, Sangeetha. Can you hear me?
Yeah,
okay, great.
I'm gonna I'll get the slides up.
Okay, thank you. Before I start sharing my slides, I wanted to say thank you so much to Julia for sharing her story. I remember I got chills up and down my spine when I heard Timothy Yu give a presentation on Mila and her treatment at the American Academy of Neurology meeting a few years ago. And I was sitting next to another neurologist friend of mine who's also gone into the pharmaceutical industry, because we go into the pharmaceutical industry partly because we think we can make a difference for more people that way. And we both looked at each other when he said he was going from kind of conception to drug administration in less than a year. We literally dropped our jaws. We could not believe it. And it was that talk has really stayed with me. And so I'm saying that because in the world of small molecule development, which is what we are doing with our attempts to treat creatine transporter deficiency, sadly, that does not happen, and it cannot happen because, as Julia mentioned, with the ASOs, that there's a template laid down by Spinraza and other ASO development programs that can be extrapolated to get more rapid approval into the clinic, or at least ability to start a clinical research program with FDA approval for another ASO. In fact, our company is doing the same thing with a drug for Angelman syndrome, an ASO for Angelman syndrome. But unfortunately, small molecules don't work that way. So you have to do this very well delineated, and I'm so sorry, but painfully slow series of steps in order to prove that the drug is safe and likely to be effective in patients. So what I'm going to present are just a couple of slides that we have presented before at the town hall we had last year. Just to kind of give you an idea what that process is like, and to assure you that we are still very much committed to this condition and going through these slow but necessary steps in our drug development process. So could I see the next slide, Judy, please?
Sure. And it looks correct, right? Is it? Let me.
It does, yes.
Okay.
Yes.
Well, let me. Does this also look correct, or is this wrong now?
I'm still seeing the very first one. I saw the other one for a second and now it's gone.
Okay. Is this right?
I'm not seeing.
We're not seeing your display
Next one.
changing. So if you have dual screens or the like, maybe
I will, well,
you
Judy-
might just need to reshare.
Oh, okay, now I'm seeing it. I just wanted to say
Yeah.
that when Ultragenyx took over the Vigilan study a few years ago, we were so delighted to meet and to work with Dr. Miller. She's been a terrific partner on this. Our collaboration has been very fruitful and often very fun because she's so delightful to work with. So even though she may not be the best at tech support, she's a fantastic researcher in this condition, and we were so lucky to get her to work with. So I will just, this is sort of a general, as most of you know, the company Ultragenyx was founded by Dr. Emil Kakis, who has dedicated his entire life to bringing treatments to families and children with rare diseases, often rare genetic diseases. So this is kind of what we do, and this is why we're still in here doing the hard work that we need to do to hopefully get this drug to the clinic someday. But as Julia mentioned, it's not always guaranteed that you get to the clinic. First, you have to do this work. So we are conducting preclinical work. Our program is called UX068, with the key focus on conducting the appropriate studies that will hopefully enable an IND submission at some point. We don't have a date for that. I know that's everybody's going to ask. The chat is going to start lighting up with questions about that, but we do not at this time have an IND submission date. But we are doing the necessary work to hopefully make that happen in the future. We always, as always in our work, and now is becoming much more prevalent in this drug development process in general, is engaging with families that are actually experiencing these disorders to understand their lived experiences, collect insights in partnership with ACD, and to generate the kind of data we need to inform our decision-making and plans. And a good example of this is a survey that ACD did for us on dietary and oral medication taking habits and issues among patients that we are still digesting and learning from. So this has been a great collaboration with ACD, and we hope to continue that kind of collaboration as we go forward. So we have completed the Vigilan study. We took it over from Lumos several years ago. It's now completed. It's the first longitudinal natural history study in the CTD population. We studied 50 patients in the United States and Canada, and this is what Judy's going to be talking about today. Next slide, please. So this just kind of shows you what the preclinical process looks like. You've got to do the basic science research. You've got to do animal studies. You have to have some kind of preclinical proof of concept. Then there's the whole chemistry, manufacturing, controls process, preclinical IND enabling studies, which is where we are now. And then finally, if you're lucky and everything pans out the way it's supposed to, then you get to the clinical human studies. So all of this work that's happening now is ultimately intended to support the clinical trial design, and the Vigilan data will definitely feed into that as well, because now we know a lot more about the population and about the rate at which patients with CTD develop skills and language and so forth. So the status is that we've identified a lead candidate after screening over 250 prodrug candidates, and prodrug means the drug that will carry creatine into the brain ultimately and then release it so the brain can use it. We call it a prodrug. We've confirmed the lead candidate. We've confirmed that it can deliver creatine to the neurons in preclinical models. And now we are, in addition to exploring opportunities for optimal drug development methods, which, as many of you know, we were initially thought we'd have to go into the clinic with an IV, which would have had a lot of, wouldn't have been the end of the world, but would have had a lot of disadvantages, mostly in terms of patient comfort and so forth. And then selecting the optimal formulation based on the patient population and disease characteristics, developing the best procedure for optimizing manufacturing, and determining the route, frequency, and duration of exposure. So the last step is kind of really the focus right now, in addition to assuring safety of this molecule. Next. So the key elements of our preclinical development are answering this question: what's the best way to deliver enough pre-drug to achieve optimal levels of creatine in the brain? How does the drug get absorbed, distributed, metabolized, and/or excreted? Because obviously, after you put a drug into the brain, you want to make sure that nothing's going to build up or create additional problems. What oral formulation is the safest for patients and the best tolerated? Which is one of the reasons we wanted to get the patient medication survey. Would be very helpful for this sort of thing. And then what are the potential dosing ranges? So we're doing research to address these questions, including studying multiple doses versus a single dose, fasted and fed animals, accumulation in the plasma, creatine in the brain, et cetera. So all of those things are happening right now. And then the drugs, ADME properties are being examined in the animal studies, and mass balance studies, which allow us to measure the prodrug and any metabolites in the blood, because again, we want to make sure that anything that's created from this drug can be processed and eliminated by the body. And then the safety and tolerability of our formulation is the part that's happening right now that's so critical. So this is just kind of a brief update, not really an update because we've presented this information before, but just to reiterate, yes, we're doing it. We're doing the hard work. We know it's slow. It's slow for us too. We would love to be in the clinic right now, and hopefully will be someday, and appreciate your patience and the support of ACD and the community in helping us get there someday. So thank you so much.
Right, thank you for that. And I echo everything nice you've ever said about me. It's been really, really lovely to work with Ultragenyx. They've been really nice colleagues, great partners, and we're trying to do something that's tricky, and it's been really nice to be able to work together on that. As with all the sites that we've had, so we have 11 sites. I have a fly in my office who's really active, so if I'm waving, that's me and my fly. We have a lot of really talented people, and here is a short list of some of them and the sites that they've been in. So I'm going to give you a brief update, and I'm going to try to, I know we're going to be short on time, so I'm going to try to go quickly. This will probably be, I think this will be the last update on the Vigilan study, because we have now finished it and I can present the final analyses, and we're writing up the manuscript. So this has been a big effort for the past. Vigilan started in 2016, so we're in year 7. We've finished data collection and we're analyzing it, and we have a lot of really interesting data. So I showed you the collaborators. Here's potential conflicts of interest. And just to give a little bit of background, most people in the audience probably know CTD is X-linked, caused by SLC6A8, and the typical symptoms are intellectual disability. Many people have autistic features, not all, but many have autistic features. Most people have delays in speech and language development, and many people have problems with behavior, and we'll go through some of the other characteristics that we've seen. Vigilan, the goal for the study was originally a 2-year study to see if we could identify what changes or doesn't change over time without intervention in children with CTD, males with CTD. It turned into a 4-year study, and we finish data collection in the fall. We stopped data collection a little bit early because we had so much data, and it was time to just analyze everything. So the model was caregivers completed surveys about every 2 months, and about every 6 months, people came in for an in-person assessment. That changed a little bit with COVID, but then we resumed. The purpose was really to understand the symptoms, the developmental course, and gather information that could be helpful in a therapeutic program, and really think about what measures could be helpful. So trying to identify study endpoints. So most of the kids in our study, most of the participants, we did have some adults. Most participants were 12 or younger. There's our age range, 1.5 to 24, almost 24 and a half. And you can see the pie chart in the left. Most of the kids are, there's about an equal 38 to 40% in the 0 to 6 age range and the 6 to 12 age range. The figure on the right is the time to diagnosis. The average across everyone is about 3.2 years, but that really has a wide range. There are some kids who are identified almost immediately after birth because there's a family history, and there are some who are older, and when CTD was, when they were little, CTD was much less known about than it is now. So their diagnosis really took a long time. So on average, we have this 3.2, but there is a wide range. Neurological symptoms in our sample are listed here. It's really important to note that uncontrolled seizures was an exclusion criteria for the natural history study. So when we talk about seizures as not being, seizures were prevalent in our children, in our patients, but we have to be careful what kind of information we generalize from that, because if you had seizures that were uncontrolled, you couldn't participate in this study. But you can see the list of neurologic symptoms and behaviors listed here. Secondarily, GI symptoms are pretty prominent in this group. Constipation is a big one, and incontinence, kind of two sides of the same coin. And you can see how things progress from there. But so many of our participants have either current or a history of difficulty with GI issues. Our baseline level of language is displayed here. So we've broken this down by age range. And so you can see that as kids get older, there are more people talking. So language is generally diminished in everyone with CTD, but it does develop. So the older you get, the more likely you are to have increased your language development. And we do have some participants who really had a language burst in early adulthood. So after puberty, they really blossomed and started speaking in much longer phrases and sentences than they had been their whole life. So there's something really important and interesting happening, and it's helpful to have some evidence that there are still a lot of developmental things that are happening in this population. Some developmental windows close, we know, in human development, but when you have a specific disease, we don't necessarily always know what that is doing to the developmental windows. And so it may be that there's more potential for language development in CTD. We looked at behavioral assessment or behavior over time. This is a graph from the aberrant behavior checklist. This is really focused on these 5 domains down below. So irritability, hyperactivity, lethargy, speech, and stereotypic behavior. The lethargy, speech, and stereotypic behavior are most often associated with autism spectrum disorder, especially the speech and stereotypic behavior. Irritability and hyperactivity are more general, and you can see in our participants, we have higher rates of irritability and hyperactivity, but it's still not in everyone. So one of the reasons we really wanted to look at this is that this measure, the ABC, has been approved by the FDA for other medications in other populations, specifically in autism. So it was important to know if this could be a measure in our CTD population. And I think we've got pretty clear evidence that it can be helpful, but it's not going to be a good primary outcome measure because there are plenty of kids who really don't struggle with hyperactivity or irritability. So we wouldn't want to base the outcome of a treatment study on something that doesn't affect everyone. All right, other things, one of the most important things we looked at is independent skills. And the reason we looked at this is because it's obviously important, but we also surveyed parents before Vigilan started. And the most important areas that parents wanted to see meaningful change in had to do with independence and skill development. So the Vineland is the best measure of that, the best standardized measure of that in that's around right now. So we have been giving the Vineland every 6 months, and this is kind of the bulk of what I wanted to talk about today. So, and some of you may have heard us describe this phenomenon in earlier talks. And if not, this may resonate with what you've seen from your IEPs and when you had your child tested for part of their school or psychoeducational evaluations. Standardized tests are designed from a statistical perspective to create a bell curve and to create a normal distribution. And because of that, they are often really difficult to include as a measure, because in order for your child who is already below their peers in some skill, for them to be able to show change, they have to not only change relative to themselves, but they have to change more than their non-disabled peers do so that they can be on a new spot of that normal curve. That is a very high bar that is just not really a viable statistical approach to finding change. So one of the important features of a test like the Vineland is that they also have another score called a V-scale score, sometimes called a growth scale value, and most generically called a person ability score or an ability score. And that ability score is not on this normal curve. That ability score is calculated for a specific set of subdomain skills. And when you give it over time, you can identify how much that child has changed relative to themselves. So instead of that child having to or having to surpass the development of all other children the same age, they only have to change relative to themselves, more than you would have expected without treatment. So ability scores are used in educational interventions. They're in a lot of tests that you might see given in IEPs. And it's just a really helpful way to see if the child is making more progress than you would have expected just from time passing. And that's what we think is going to be a good outcome measure in CTD and in intellectual disability research in general and intervention in general. So this figure is the 9 domains from the Vineland. These are the different areas. So receptive language, expressive language, written communication, personal skills, domestic skills. So personal skills is bathing, toileting, getting dressed. Domestic skills are helping out around the house. Community skills are being safe when you're crossing the street, being able to talk to strangers, find the bathroom, that kind of thing. And then the third row has interpersonal playtime and coping skills. So this figure shows us that when we look at the standardized score, which is the V-scale score, you see all these kids on the floor. So you see how there's these deep rows of kids who are at the 1 mark. That's because that standardized score is so pushed into this normal curve that our kids are just always at the low end, and they can't really, you can't even really see their range. So this is why standardized scores are going to be a problem in this population. But since this test also has growth scale values, we can use that to really understand this population more and potentially use that as an outcome measure. So these are the same 9 domains, and there's 2 things to kind of point out. The red line is what you kind of trajectory you would expect just with time. And so so you can tell each one has its own curve. These are not a standardized set of numbers. So every single domain is going to have its own specific curve for this specific test. And that's important to keep in mind. And then you'll see the circles. Those are all the individual participants and their various data points across their study participation. And you can see there are still a few domains where a lot of kids are clumping at the floor, but not in most domains, they are not. They are off the floor, and you can really appreciate their ability at the beginning and their ability at the end of their study participation. So this is going to be much more helpful in terms of being able to identify change over time. And I think what we will do is pick the domains that really seem to be the most important and the most promising. And so far that seems to be receptive and maybe expressive communication, personal care, and coping. I think that would hit a nice range of skills. Okay, so in conclusion, we learned a lot from Vigilan. We didn't present all the results. I have presented other results at earlier conferences. We are writing up the manuscript, and it will hopefully be submitted very soon. Our colleagues at the NIH have been incredibly helpful with data analysis. And I think the things that we are learning from Vigilan will be applicable to a lot of other rare diseases. And our colleagues at the NIH often talk about how they, in particular, have been partnering with researchers on multiple rare diseases. And CTD is really kind of the furthest out in front, where we have more longitudinal data than a lot of other diseases. So I think what we've learned from Vigilan will be really informative for other populations. So we know that in CTD, the symptoms occur early. It can take a long time to get the right diagnosis. We've got this nice evidence and data on the neurological and GI symptoms and the kind of persistent cognitive and functional impairments over time. But we do see improvement. You saw those kids with those growth scale values scores. They are making growth and developing skills over time. And so that's what we want to capitalize on, and what we want to measure, and what we want to see change even faster with good intervention. And that is the end of the slideshow. And I will stop sharing. Happy to take questions when we get to that point.
Thank you, Dr. Miller and Dr. Brandenburg. I'll request both of you to please stay for the Q&A session that will happen at the end of the panel. At this time, I'd like to invite our next speaker, Dr. Seth BERGER. Dr. BERGER is a physician scientist, board certified in pediatric medical genetics and biochemical genetics at the Children's National Hospital and Research Institute in Washington, D.C. His research focuses on bioinformatic approaches to improve molecular diagnostic testing for rare diseases. His talk is titled Evidence for Cardiac Arrhythmia Risk in Creatine Transporter Disorder. Thank you, Dr. BERGER. Over to you.
Thank you very much for that introduction. And I just wanted to say that I feel like all the presentations thus far have been spectacular, and that really kind of leads into certain key points I want to make here. So let me just start by saying that I have no disclosures. I am a physician scientist in DC, and I actually have 2 patients who I follow with a creatine transport disorder. And I kind of want to share some of their stories because I think it highlights a aspect of the disorder that's not that we don't speak about quite as much. So this is basically the agenda of what I'm going to talk about. I'll briefly start about the common features of creatine transport disorder. talk a little bit about cardiac arrhythmias, present 2 cases, and then kind of give my perspective of how I feel that there's implications for both clinical management, as well as some of the drug discovery that we have been discussing in some of the previous talks. So really, when we talk about creatine disorder, and I just pulled these features from Gene Reviews, and they're the same things that were mentioned in the previous talk, it's really a lot of discussion about the neurologic features. We hear about the developmental delays, we hear about the people who have seizures, we hear about the behavioral differences, we hear about the hypotonia, sometimes people with movement disorders. We hear about some of the GI features, which may or may not be secondary to the neurologic features, including things like poor weight gain, constipation, and all the other things that were mentioned in the previous study. But I mean, I have not heard anyone mention in the talk thus far about that the emerging evidence that were published in 2021, namely that prolonged QTC on EKGs have been reported. So what does that actually mean? And that is basically so basically when we talk about EKGs, this is looking at the electrical activity of the heart. A normal EKG, you know, you have what's called a P wave here, a QRS complex, and a T wave. This is basically the top of the heart, the atria depolarizing, the QRS complex is the ventricles depolarizing, and the T wave is the repolarization of the top of the heart, again, getting ready for the next beat. Oops, sorry. So essentially, there are some people where this interval from the QRS complex to the end of the T wave is kind of stretched out, and that predisposes to a very dangerous situation known as torsades, which is a polymorphic ventricular tachycardia, where the heart is beating so quickly and the electrical activity is not really consistent with basically a functional squeezing heart, that you're not getting perfusion. And this is a very dangerous situation, and people die from this. Now, at a single cell level, when you look at a cardiac myocyte, each of the cells have what's known as an action potential, where you look at the cell depolarizing, the cell repolarizing. And this is basically what you see here in the QT interval, that you have a stretching of the whole action potential of the myocytes. And these are various different currents that play a role in here, and there's energetics of transport of ions across the membrane. But, you know, this is dangerous because, again, fainting, sudden death, ventricular arrhythmias, and torsades. And there are a lot of single gene disorders that have been associated with this feature. But what I kind of want to bring up here is that many medications are known to lead to this as a side effect. And this is actually, since there's a lot of discussion about drug development here, having a drug that prolongs the QT interval and leading to potentially arrhythmia risk is a reason that a lot of candidate drugs do not make it to market, because they're doing a clinical trial, somebody develops torsades, and then suddenly the drug company says this is too dangerous to bring to market, and they don't, or they have to have some sort of warnings on it about this as a potential side effect. So that's why I think it's important to know if this is a part of the risks of creatine transport disorder, especially as you're thinking about future clinical trials, because you need to know, are you seeing this as a side effect of the clinical trial, or are you seeing this as an underlying feature of the disorder? So, I mean, this was a paper that Mark Levin, who I know has spoken to your group several times, has presented, where he was looking at the NIH cohort and found that, you know, there were a significant number of people who had this EKG abnormality on their testing. This is, again, important because, you know, this is a previously not well-defined feature of creatine transport disorder, so we don't always look for it. You know, when people know about seizures, we send people to get EEGs. When people know about developmental issues, they get all the developmental evaluations. But having a cardiac rhythm abnormality is not something you typically know to look for unless somebody's having symptoms such as fainting, such as exercise intolerance, such as you know, having a near fatal arrhythmia when given the wrong medication. So that's again why I really feel that this is something that as a group needs to be discussed here. So let me tell you about 2 patients that I've followed. I know that they're both members of the community and may be listening in now. So if you are, hi, hope everything's well. But they've given me permission to share their stories. So the first one is a 10-year-old boy who had a history of autism, who basically, he got sick. He got some sort of viral illness. We're still not exactly sure what happened, but in the setting of this severe illness, his liver started looking really, really unhappy. He was sent to a regional, like one of the top hospitals in the region for dealing with liver dysfunction. And, they're working him up. And one of the things that we do as part of our workup for children with liver dysfunction is see if there's a genetic reason. And so the way genetic testing is typically done these days is often we can't get coverage for inpatients to get, the right genetic test, which would be doing like maybe an exome or something in this case. So they sent a cholestasis panel, and they were mainly concerned about one gene called POLG, which can present with like autistic features, liver dysfunction. And what came back on that report was nothing that they thought was contributory to this presentation. Basically no variants in POLG and a few variants in genes that were not thought to be related to his presentation. So now we have a child who has a history of, some behavioral and developmental differences, and he's in PICU recovering, and notice his liver started doing better spontaneously. Probably whatever viral process was going on managed to work its way through his system. But you have this, child who's in this really a PICU setting with, lines and tubes and everything. He starts getting agitated. And one of the treatments that they do for agitation in the hospital, sometimes they give you medicine to address that agitation. In this case, they gave him a medicine called Haldol. And one of the Haldol can have many side effects, but one of them is in someone who's susceptible, it can induce cardiac arrhythmias. And that's what happened in this child. He developed torsades. And, I've spoken to his mother and she described how scary the situation was for the entire family. And it was scary. So he ended up being transferred to our hospital at that time because we have a cardiac intensive care unit, and he continued his convalescence with us. Now, again, because of the complexities about what testing we're able to order on inpatients, and we know that a certain percentage of cardiac arrhythmias can be due to an underlying genetic etiology of a cardiac arrhythmia, we sent a cardiac arrhythmia panel, and again, did not give us an answer. At this point, he did actually end up getting an ICD placed because, when you have a risk of a cardiac arrhythmia, you always have to make sure that if it happens again, you can it doesn't turn into a fatal event. If it happens in a hospital, we're good at keeping people alive. But if it happens when you're walking down the street, you need a way to keep you alive until you can get to a hospital. So essentially, at this point, we had no genetic answer, and he was enrolled in a research study we had. And I was really interested in Julia's story before because, she describes a classic situation a lot of people end up in where they have genetic testing, we don't find a variant. And in this case, this is what happened. He enrolled in our research genetic study where we were able to take the clinical genetic testing he had done previously, which was a panel, and open it up and look at the entire whole genome that was performed. So the variant we ended up finding was actually an interesting variant. You've heard us throw around terms. I know Julia mentioned during her talk about MILA'S variants was that, this whole idea of intronic variants versus exonic variants. And this is kind of what we found in this child. We found a near exonic, but an intronic variant. It was a 2-base pair deletion of 2 bases in right near this intron exon junction. Depending on how you align, it's either at this -2, -3 position or -4, -5 position. But to the nerds in the room, it leaves this A intact, and because this A is intact, what is called the canonical splice site is intact. Interestingly, the only reason this variant was ranked highly in our machine learning approaches for variant assessment was because of the fact that we had included his feature of autism. According to the ranking at the time, it wasn't the arrhythmia that triggered this variant to be high. It wasn't the liver issues. It was just the autism. So, when we find variants like this, we try to classify them. And based on our classification, this variant was considered what we call a variant of uncertain significance, because when we try to predict the effect of this variant, there's a tool called Splice AI that says, is this going to break a splice site? And what we actually see here is that it gives a somewhat inconclusive score. It gives a score of 0.21. 0.2 is kind of a wishy-washy Splice AI score. If you get a score of like 0.9 or above, it breaks splicing, but this one, it's not clear. So, one of the benefits of creatine transport disorder and from a diagnostic point of view is you don't have to rely on molecular diagnostics. There are actually clear biochemical diagnostics. So this is the magnetic resonance spectroscopy of his brain. And you can see over his basal ganglia, most people have a lactate peak over here and an N-acetylspartic acid peak here, but most people also have the creatine peak here. And this is the pathognomonic sign of a cerebral creatine deficiency, is that on the MRS of the brain, you have a decreased creatine peak. So we knew this was his diagnosis, but we wanted to explore the variant a little bit further. And we also did the urine biochemistry and looked at, guanosine acetate and urine creatine excretion. And that was all consistent with the diagnosis. So biochemically, we knew this was the diagnosis. But we went ahead and we did RNA sequencing. So RNA is, of course, the product of, when you transcribe DNA, you make RNA, and RNA is what is the instructions for making protein. And what we see in him is that in a panel of typical people, they have around, 1,000 or 10,000 reads of over this particular region, where for him, he is markedly reduced, so below 2,000. So again, looking more closely at RNA reads in a slightly different way, this is a panel of 11 typical controls, and we see that this is what the normal splicing pattern of the RNA looks like. And with a variant right in this intron over here, for our patient who I'm talking about, we see that you have some what we call exon skipping. So this variant over here leads to the skipping of this exon. You have some normal splicing and some mildly aberrant splicing. And what this shows us is that, and again, look at the level here. Most people have around 4,000 transcripts, whereas on him, you have only about 200 transcripts that are visible. And out of those 200 transcripts, maybe only about 87 of them are actually typical splicing at this intron, at this exon junction here. So what we do see from this is that this is actually a light leaky splice site variant. So while he does have markedly decreased creatine transporter expression levels, enough that he's not getting enough creatine into his brain under typical situations, we do see that he also has some normal expression, which confirms that, which basically explains one difference that this child had in his presentation. Namely, while we all know that creatine transport disorder traditionally does not respond to treatment, because if you give someone as much creatine, if they cannot transport it into their brain, it doesn't really have its functional effect. Since he has some leaky typical transporter expression, we suspect that this is why when we did start him on creatine supplementation, we saw a marked improvement in his baseline aggressive behaviors. And I mean, his mom, who I think is listening here, has told me on several occasions how he's kind of like a different kid since starting this supplementation. He's able to kind of concentrate in the classroom instead of being in timeout the whole time he's in his classes. So, I mean, she tells it better than I can, but I mean, essentially those are, I think, a main point here. The other main point I make here is that the reason that we found this was because of the autism as a feature. However, as Dr. Levin's paper showed, QT prolongation, which is a known risk factor for developing torsades, is a feature of creatine transport disorder. That wasn't making it into the databases, so that if someone with just an arrhythmia had a genomic test, they would not have picked up this variant. So therefore, we did get the human phenotype ontology to add in arrhythmia as a phenotypic term for creatine transport disorder, because again, it's important that we are able to catch these kids, whether they present with autistic features or present with an arrhythmia. So really, the take-home point from this is that this is the first reported case of a drug-induced torsades in someone with creatine transport disorder. There were definitely treatment implications. Specifically, he required an ICD placement due to his arrhythmia risk. I thought it was an interesting example of being able to do variant of uncertain significant disambiguation using RNA sequencing, and that we have a hypothesis about why his response to creatine might be a little bit atypical due to the type of variant. The other thing I would note about this variant in the context of some of the previous stories we heard is that this is actually the type of variant that is amenable to ASO therapies. I wouldn't recommend it in this case because there are, again, small molecule oral treatment that seems to be helping, and ASOs would require, drilling holes into heads and infusing intrathecally. But just saying that this type of variant does exist for creatinopathies as well. And the other thing to keep in fact is it's unclear if the creatine supplement has any effect on the cardiac features either. So case 2 is, a few months after I had met this family, another family was referred to me who had a previous diagnosis of creatine transport disorder. This child had what I would call a more typical presentation of today, where you have a child who has developmental delays, motor delays, language delays, maybe a couple seizures, and then they get an exome. So this exome reported out a variant of uncertain significance in SLC6A8. And again, because of the benefits of creatine transport disorder having biochemical markers, they were able to confirm the diagnosis using, urine creatine and magnetic and MRS testing. Again, this child had no cardiac features, no history of anything that would have raised a red flag. But given this evidence in the literature, I did send him for a baseline cardiology evaluation. Now, the cardiologist, again, did an echo, structurally normal heart, did an EKG, and this EKG did show some of the things that Dr. Levin had seen, specifically a mildly prolonged QT interval with some abnormal T-wave morphology. So the cardiologist, saw this, felt this child was at risk for arrhythmias, and started him on oral medications, namely a beta blocker, Nadolol, because of that risk and to minimize that risk. And knock on wood, this child has never had any cardiac features, never had any arrhythmias, but we're being protective to prevent a potentially bad outcome, knowing that there is this underlying risk. So really the take-home point is, I think SLC6A8, should be on cardiac arrhythmia panels. That anybody who's being evaluated for long QT syndromes, they should also look for a creatine transport disorder. Takes a long time for clinically available panels to add genes on, so it's not clear how long that will take. But also, testing patterns have shifted over the last few years. There's more exomes and genomes being done now. So ideally, with the update of HPO, now this will be picked up as a potential feature. I also think knowing these risks are important for the future drug trials. As I mentioned, torsades can be an adverse effect of many medications and can prevent medication progression to market. Unless you know that this is an underlying feature of this disorder beforehand, people might blame new drugs as they're being developed and avoid them making it to market. This is also a classic thing that we see in a lot of rare diseases, that as you focus on one set of features, other features may actually become apparent over time. The classic example was like PKU, where before we knew how to treat PKU, everybody thought PKU's only effect was on neurodevelopment. And then once we have people treated for PKU throughout their lifetime, we learned that there was a maternal PKU syndrome, and it can cause teratogens in their children. Again, the other thing to keep in mind is as drugs are being developed to target CNS creatine deficiency, it would also be important to consider whether or not these medications would affect the cardiac creatine deficiency as well, and whether that is something that, there are other treatments for cardiac arrhythmia risks. So whether one would have to do a dual medication or come up with a treatment plan to address both of these. So, based on this and, the fact that, the second patient had a finding without, I think that it's probably important for anybody with creatine transport disorder to get a baseline EKG and a cardiology evaluation just to be safe. And avoiding QT prolonging medications is something that you can discuss with your doctors. There is a list of medications that are known to cause a side effect. Again, for other creatine deficiencies, I don't know if this evidence is out there, so I'm not sure whether or not. It's unclear. Dr. Levin wasn't sure either when I was speaking with him. So, you could consider baseline EKGs, but again, this might just be a transport issue, and it's possible that people who have enough circulating creatine from their diet might be enough for the heart, just not the brain.
Dr. Berger, just a quick note that we are out of time.
So I'm just ending now, just saying that if anybody's interested in research opportunities, I do have a study for people who are missing variants. So if you have either GAMT, a single variant, or AGAT with a single variant, we do have a research study that you could participate in if you are interested. The URL is up there. I'll drop into the chat as well. And that's all I have.
Thank you so much, Dr. Berger. Please stick around for the Q&A that will follow. We have one more talk in this session. It's my pleasure to invite Dr. SAMAR RAHHAL. Dr. RAHHAL is a pediatric endocrinologist. Her medical training spans several medical institutions, including the Cleveland Clinic for residency and a pediatric endocrinology fellowship at the Riley's Children's Hospital at Indiana University. She is currently a staff clinician at the Eunice Kennedy Shriver National Institute of Child Health and Human Development at the NIH, where she was a principal investigator on the CTD natural history study. Her talk is titled Elevated Amyloid Beta Peptides and Tau in CSF of Individuals with CTD. Dr. RAHHAL, please take it away.
Thank you, Dr. Iyer, for the introduction, and let me share my screen. Here we are. All right. So good morning or good afternoon, everyone. And at least here on the East Coast. Thank you for inviting me to present our work today. My talk is entitled Elevated Amyloid Beta Peptides and Total Tau in Cerebrospinal Fluid in Individuals with Creatine Transporter Deficiency. I want to thank all the co-authors for their invaluable contribution to the research. To start with a little bit of background, I will start with a quick review of the biomarkers that I mentioned earlier. So A-beta 40 and A-beta 42 are produced by the cleavage of the amyloid precursor protein, which is a transmembrane glycoprotein. It is widely expressed in the brain and other tissues of the body. It's cleaved by, like I said, beta and gamma secretase to release amyloid beta peptides A-beta 40 and A-beta 42, among others. Whether because of decreased clearance or increased production, the increased production of A-beta 42 occurs and aggregates very easily and incorporates into amyloid plaques. These plaques are thought to be mediators of neurotoxicity. There are many hypotheses that underlie Alzheimer's disease pathophysiology, but one of them is called the cascade hypothesis, and it states that these amyloid plaques are actually some of the first manifestations of Alzheimer's disease, and that they contribute to the pathology seen, specifically dementia. What is also known is that in Alzheimer's disease, levels of A-beta 42 are actually decreased in the cerebrospinal fluid, and these can actually be used for diagnostic purposes. And the reason for this decrease is thought that because of the increased aggregation in the brain into these plaques, less A-beta 42 is being cleared and found in the cerebrospinal fluid. On the other hand, another biomarker is tau, and tau is a protein which in the physiologic state has an important role in microtubule assembly and stabilization. Tauopathies are actually a group of neurodegenerative diseases that have in common neurofibrillary tangles. These neurofibrillary tangles form when tau is produced in great amounts or in abnormal ways, and it also aggregates into these tangles. They are also thought to be neurotoxic, and like I said, they are thought to be part of the pathophysiology of many neurodegenerative diseases, specifically progressive supranuclear palsy, Pick's disease, and secondarily probably also Alzheimer's disease. What happens is it's thought that tau loses its ability to bind to the microtubules, affecting the integrity of the cell. In Alzheimer's disease specifically, CSF levels of tau are found to be increased. So back to our findings. So what we did was, as Dr. Miller and Dr. Brandenburg mentioned earlier, these findings are from the Vigilan observation study of CTD. And throughout this 4-year study, participants who opted to were able to get lumbar punctures done once over the 4 years. And the cerebrospinal fluid or CSF that resulted from these lumbar punctures was analyzed for this study that I'm showing you right now. Ideally, we would like a control sample of healthy individuals, but as you can imagine, it would be very difficult to obtain that CSF from healthy individuals. So for our comparison sample, we use CSF that is obtained at the NIH as well from individuals with Smith-Lemli-Opitz syndrome, or SLOS, and this CSF is obtained in the exact same manner. We also, for our comparison sample, included CSF obtained from pediatric patients who presented to outside ERs for various reasons, and these served as what we call our comparison sample, or our pediatric controls. And we have their ages. For some, we do have sex as well, but we don't really have a lot of information on their clinical presentation. And also all the participants from the Vigilan observational study, as Dr. Miller outlined and showed us the beautiful data earlier, had neurodevelopmental assessments, and these, they had neurodevelopmental assessments, and these included the Vineland Adaptive Behavior Scale, 3rd edition, which produces a score called the ABC score, which evaluates the skills needed to meet the demands of one's environment. The population mean for the ABC score is 100. So these are our results. As you can see, for the CTD, we had 12 participants. For our comparison sample, we had altogether 23 participants. Their mean age was comparable. The comparison sample had a slightly higher mean age. The ABC score for the CTD population was, as expected, lower than the population mean, and about 58% of our participants had seizures, which also probably is in line with what we know about CTD. We know that with our comparison sample, none of the SLOS participants had seizures, because we do have clinical data on them, but we don't, as I said, have data on the pediatric controls. And so these are our findings. As you can see here, there are these are the levels of the biomarkers. So we have the total tau, the A-beta 40, and the A-beta 42. In red are the CTD participants, in blue are the comparison sample lumped together. And as you can see, for each of the 3 biomarkers, the geometric mean of the biomarkers in CTD is elevated or significantly elevated compared to the comparison sample, and this was significant in all 3. We also looked at the ratio, which is also looked at in Alzheimer's disease, and in Alzheimer's disease specifically, it is decreased because A-beta 42 is decreased, whereas A-beta 40 is not. But in our population, we found that the ratio was equivalent, which seems to suggest that both A-beta 40 and A-beta 42 increase at the same rate.
Dr. Rahhal. Dr.
Rahhal.
Yes.
I believe the slides are not advancing in sync. We are actually seeing the slide that says results.
Oh, is that the one you're seeing? Oh, okay.
Yeah.
Oh, so this wasn't showing?
No, we were one slide behind for whatever reason. Sorry about.
My presenter view is a little, I apologize. So let me go over the results again, and thank you for pointing that out. So, so again, I'll repeat what I just said. These are our 4 graphs which show the biomarker levels in comparison in CTD compared to the comparison sample. So here we have the red dots, which are the CTD population, compared to the comparison sample, which are the blue dots on the right of each chart. And as you can see, again, the biomarkers are elevated in all 3, significantly so. And we did not find a significant difference when we looked at the actual ratio, as I mentioned. So then we took it a step further, and we tried to see if individually in our CTD population, the biomarkers had any correlation with the Vineland ABC scores. And we found that for each of the biomarkers, we did have an inverse correlation, where the higher the biomarker levels, the Vineland score tended to be lower. As you can see, there is one of our participants here is the outlier in the case. We considered this individual an outlier because he did not fit our correlation, in the sense that his biomarker scores and his Vineland ABC were both decreased. But otherwise, we did the analysis with the other participants and we found that there is an inverse correlation. We did try to explain why the outliers data did not match, but we couldn't find a reason. He has genetically confirmed CTD and clinically as well. And also the CSF, we went back to make sure the integrity of the CSF was valid by looking at glucose and protein levels, and it was valid, at least at the point of collection. Since we only had one CSF sample over the course of the 4 years, we weren't able to go back and see if our results held over time. So in conclusion, we found elevated levels of A-beta 40, A-beta 42, and tau in CSF of individuals with CTD when compared to the comparison sample that we had. Elevated biomarker levels inversely correlated with neurodevelopmental measures. So in order to try to explain our findings, we hypothesized that the creatine deficiency that occurs in the brain and the neurons of the brain results in energy deficiency in the brain. This makes sense because creatine is involved in energy utilization and storage as phosphocreatine in the brain, and alterations in the system as occurs in CTD or likely occurs in CTD may therefore result in cerebral energy depletion. There are studies that show that altered bioenergetics have shown to modulate APP processing or cause abnormal APP processing. And even though we did not look at phosphorylated tau, there are also studies that show that cerebral hypoperfusion, which leads to impaired energy production and utilization, can lead to an increase in abnormal tau phosphorylation. Also to conclude, obviously our study is small and it would be ideal if we can replicate and confirm our findings in larger studies and or other models of disease such as the animal model. Our biomarkers may serve as therapeutic targets. Here I bring in AGAT and GAMT as it would be interesting to study our biomarkers if our theory holds and the creatine deficiency is what causes the elevation in the biomarkers by affecting energy utilization, then we would expect the same findings in AGAT and GAMT. And what would be interesting would be to see if the creatine supplementation would decrease and affect the biomarker levels. It's important to note, though, that we found elevated biomarkers in our CSF, but we do not have any evidence of plaques or neurofibrillary tangles in brains of CTD individuals. I would like to thank the individuals and their families for participating in the study, and I would also like to thank the Association for Creatine Deficiencies for holding this symposium today and for help with promoting this study. Thank you.
Thank you so much, Dr. Rahhal. I'd like to invite all of the speakers from the previous session to please turn on your cameras. And for as moderators, I would like to invite Dr. Feigenbaum and as well as Sung to please moderate this session. As a quick note to all of our audience, we will be skipping the break, or if time permits, we'll be doing like a 2-minute break. So you can grab your coffees, but I'm sure everybody wants to hear from the speakers and the discussion. So I hope everyone's okay with that. Over to you, Sung and Dr. Feigenbaum.
Dr. Feigenbaum, I think you're muted.
10 cents in the penny jar. Yes. Good morning. So we had some questions that seem to have disappeared from the Q&A, but maybe we can start. So Sung, if you can tell me that indeed they have been deleted, but maybe we can start with a question for our Ultragenics representative, Dr. Brandeboer. When I check the website for Ultragenics, it doesn't show anything about UX068 in the pipeline. Is that just because it's preclinical, or usually preclinical is shown on the website?
Yes, we don't generally put our preclinical programs on the website. We have a lot of projects going on, and I think it would be confusing if we listed all of them, so we don't generally do that. I think a few years ago, we released some information about this program, which was kind of an unusual thing to do, but we're sort of back to being more quiet about things that are still in the preclinical stage now.
Okay.
We don't want to create expectations and then disappointment.
Absolutely, absolutely. And then I wondered whether, I know you commented on the reasons for targeting an oral therapy. But if IV or intrathecal may be more efficacious, has the family group parent population been surveyed to see if they would go for that, if that turned out to be the better way?
Well, I think at this point, we don't think that is the better way. I mean, obviously this could change, but at this point, we think an oral formulation will kind of give us more flexibility with possibly less toxicity. Again, too soon to tell about that, but we think that an oral formulation would, for example, if necessary, allow us to dose multiple times during the day without getting the levels high enough to cause maybe a side effect. Again, I'm sort of giving you a blanket answer, but at this point, we think the oral formulation not only will be more convenient for families, but will enable the drug do what we expect us to do. So until we sort of have worked that out internally, I don't think it's appropriate to ask families their preference, because their preference may not be possible, or it may not be the best way to deliver the drug. If we were to get to the point where we had several options that were all equally effective and equally good in terms of side effect profile, then at that point, we would certainly come back to the families and say, which do you prefer? But we're just not at that point yet. And I understand that the families are frustrated because they're saying, we'll take it anyway, we can get it. But we have to be safe, and we have to work with what's the best option to have a therapeutic product that's safe. So.
Great. Melanie, there were a couple more questions that I gather you were planning to answer. Were there any types of correlations between the clustered mutations and the Vineland measures? I don't know if that's for Melanie or Judy, but both.
I don't think this has been looked at yet. See, this is the wonderful thing about the study, is that we got so much information, and we are writing up kind of a basic manuscript about this, but this data will be there to mine additional information from for years to come, and not just for Ultragenyx, but for anybody working in this field. So at this point, we haven't done it. It's a great idea to do that, but it hasn't been done yet. I don't know if you want to add anything to that, Judy.
No, I would echo that. I think there could be an opportunity that probably anything that we found would be subtle. So just a matter of figuring out like what are the different opportunities that we can do. And about the questions that may have disappeared, I tried, in the interest of time, I tried to answer some questions, so they may be in the answered thing. That may be the ones that look like they disappeared.
Yeah, I answered one that way as well. I didn't know it disappeared.
Yes, and the other question that has been posed is whether there is a correlation between the creatine peak on the MRS and the Vineland score. Obviously, I do not know if you have answered that one, but that obviously could be correlated both with the mutation and with the Vineland score. Have you attempted any correlation just between the peak on the MRS?
Not yet.
Thank you.
Okay. So the next question is for Dr. Berger. Is RNA sequencing available for anybody to access? And I think you partially answered that with your research study. But as a clinician, I can say, yes, it is available if necessary, if there's a VUS or
right. I mean, I think the point there is that for that patient in particular, it was an intronic variant. So there was a question to ask using RNA-seq. We actually did it by research. We also had it done clinically. So MNG Labs is which is part of LabCorp, is the only lab that I'm aware of right now where you can clinically order it. There are also limitations, but it's good to know that they can do SLC6A8 at MNG, and they do good interpretations there.
Yeah.
Yeah, it is done at Perkin-Elmer as well, Perkin-Elmer Genetics, but you have to arrange that with the medical director.
Okay. The next question for Dr. RAHHAL. was correlating your findings with Alzheimer's results of the tau and the biomarkers. I see in the literature for Alzheimer's that there seems to be some discussion about which one is the best biomarker. But the question is, are there any approaches to reducing these plaques that have been successful so far? And of course, we know about, well, maybe you can answer.
Yeah, no, that's a great question. So in the Alzheimer's disease literature, obviously there was a lot of interest and a lot of work on therapeutic targets that would involve decreasing the plaque formation. And it seems that in recent years, this has been disappointing. There have not been a great response to agents that have been used to try to decrease the levels of the plaques or of A beta 42 aggregation. So now tau is gaining a little bit more interest in that sense, and I don't think that we have conclusive results on that one. Like I said, the A beta 42, the decrease in the CSF for Alzheimer's disease is used it is widely used diagnostically if needed. So if there's dementia and you're trying to see if it's actually Alzheimer's disease, but as far as therapeutically, it has been on the more disappointing side, unfortunately.
And you only did the CSF once, right? So it's not an indicator of progression of disease or anything like that.
That's the thing. So the study included one lumbar puncture across the 4 years. And that's the one with the ones that we had available, we did analyze. Yeah. It would be nice to look at it over time. I agree 100%.
Yeah, always a challenge.
Yeah.
Dr. Stockler. Any association with the type of mutation? I don't know who that's for, but
for Dr.
RAHHal.
So it is two questions. One, is there any association with age? And the second thing, with the type of mutation.
Yeah, like I said, we do have a small sample, but we did try to look at both of those things and we did not find an association. But again, the caveat is that it's really hard to tease out when we just had the 12 participants.
Okay. And the question that there is also for both Dr. RAHHal and Dr. BERGER. So basically, is there any possibility that any type of successful intervention can affect the level of your biomarker in the CSF or the QT interval in the heart? Dr. RAHHal, you go first.
Okay. Again, I think that it would be ideal to use them as therapeutic targets, not knowing exactly if they are actually causing any part of the disease, basically. But they, when we're looking for objective targets, these biomarkers could potentially serve as that.
One of the challenges with using the QT interval is it does vary over time and based on like hydration status, electrolytes, a lot of things. So if you have someone who has a normal QT interval, one visit and the next visit's prolonged, would be hard to use that as a direct therapeutic endpoint, I think. But I mean, as a secondary endpoint, sure, why not? I don't know.
I just want to interject that we saw the same thing in the Vigilan study, where sometimes they had a prolonged QT interval, and then maybe at the next assessment they didn't, and vice versa. So I would second that.
But my question is, if you do a repeated measurement, is the average QT characteristic, can you differentiate among different patients on the average QT or not? What do you think? But making, obviously, many times we do studies where we request a repeated EKG, and this was the case also for this one. But, and that is my question. I think that we are getting out of time. Go faster.
Okay, I don't know who that was for. You're on mute, Dr. Longa.
Dr. BERGER, do you think that an average QT may be an adequate thing or not?
I mean, I think the problem is that we don't care about what the average QT for this patient is. We care about, are they having excursions that are high enough that it's causing a arrhythmia risk? So, I mean, I would worry about looking at just an average, but I'm not a cardiologist, so I would actually probably defer to Dr. Levin or somebody who has more deeper cardiology knowledge than I do.
So Dr. Levin's article suggested sequential assessments for cardiac, which implies to me that this is a progressive condition, but we have no other real evidence that CTD is a progressive problem. So would one and done be fine? And in that case, we could do a halter?
So, I mean, on at least my patients, they've both have had halters. None of them have had any episodes caught on their halter that were particularly concerning, like no runs of PVCs or anything. Again, N of 2 here, so I'm not making any judgment on that. I mean, I can tell you that at least one of them have had normalization of their QT interval on subsequent checks, but also that's the one who's on medication for rate control, so it's hard to say. So, I mean, we need you guys to generate more evidence. Let me throw it back at you.
Yeah, it's a difficult one. I mean, one doesn't want to subject our families to all these extra interventions. They've got enough on their plates. On the other hand, you want to be safe. And so hopefully a further study and more analysis of the Vigilan study will answer that. I think coming back to also the delay in diagnosis, nobody's mentioned, and hopefully I'll just put a plug in for whole genome sequencing as newborn screening. May answer some of that question. There are those of us who are for and against, but it's coming, I think.
But quick devil's advocate to what you just said, though. I mean, I'm a big supporter of early newborn screening with genomes. However, at least many cases have variants of uncertain significance, especially cases due to missense variants. Newborn screening is not going to be reporting through variants of uncertain significance unless there's a symptom. There is another study that's actually coming out called Project Find Out, which is basically the idea of doing genome at the first sign of neurodevelopmental delay. And I mean, I think that might be an opportunity to capture these children's quicker and earlier.
Agreed.
Yes, I think that we are running out of time. I would pass the microphone back to Sanjita and to proceed with the program. I want to thank all of the presenters for the beautiful and outstanding presentation. They continue to add some important pieces to what we know about brain creatine deficiency syndrome. Thank you to all of you.
Thank you.
Thanks, Dr. Longo, and thank you to all the speakers. There's a lot of questions in the Q&A box, so if you could continue to answer those, that would be great. And for everyone in the attendance, please look at the Q&A box and the answered sections for some of that. We're gonna not go to break, and in the interest of time, just get started with the next session.
Our next speaker is Jackie Gallagher. Thank you. A rising third-year PhD student conducting groundbreaking research in Schlabach Lab at Purdue University. Her research focuses on uncovering structural insights into the native and non-native conformations of SL6A8, the creatine transporter. More specifically, her lab is applying hybrid techniques to identify small molecule therapeutic leads that are suitable for optimization as pharmacochaperone targets for misfolded conformation of SLC6A8. Because we don't have much time, I'll just introduce the title of the talk, towards the discovery of small molecules that restore the expression of function of CTD variants. Jackie, please take it over.
All right, thank you. All right, and let's see. Can everybody see my screen?
What do they see presenter?
Yes.
We do see presenter mode.
Oh, you need to flip to the other side.
Better?
Yep.
All right, perfect. Thank you so much. And thank you for the kind introduction, and also to ACD for inviting me to speak a little bit about my work today. So yes, the title is already given to you, so we'll kind of jump right into this. So in terms of creatine biosynthesis, there are a couple different routes that genes can become mutated and can lead down the road to disease. So seen here is AGAT and GAMT. These first two, creatine supplementation can generally be effective for them. However, for the third disease that can arise, for CTD, it's the Creatine Transporter Deficiency, and there's currently not any treatments for this. And creatine supplementation generally is not very effective for this one. So this is where the majority of my work is focused, and it is on the transporter and mechanisms within it and small molecules. So for CTD, there are about 270, or this is what's currently listed, public variants, and there are a lot of different mutations in the gene that can lead down the road to protein misfolding. So there's in-frame deletions and missense changes, and as you see, the two of those together account for about 63% of pathogenic variants. And these are ones that we think may be able to be rescued by a small molecule. So on the right over here, along this bottom axis is the amount of the protein that is actually expressed and like trafficked to the plasma membrane. And while we see some variants do still retain their expression, some don't. However, on this y-axis right here is their function of the actual creatine uptake. And through the few variants that we previously tested, this is not for all of them, but we previously tested a few, that along this y-axis right here, most of them are losing their function entirely. They're just not uptaking creatine. So when membrane proteins misfold, the problem is that when they're produced and they fold, this kind of dark blue part here is the endoplasmic reticulum. They get stuck in this area and degraded. So they usually have to make their way all the way from once they're synthesized and then folded, all the way through this, through the Golgi, and then trafficked here to the outside of the cell, so the plasma membrane. This is where it's going to function for creatine uptake. However, when it is misfolded, it gets stuck in this area here and degraded. So it never successfully makes it to the plasma membrane. And previously, a former lab mate of mine had demonstrated this, where WT is just going to be wild type creatine. And this is an example of one variant where he showed this. And so we have a stable cell line that is creatine, and then it has a fluorescent reporter that is immediately tagged to it. So this is the amount of total creatine, but then this ER is able to track if it is getting stuck in the ER or not. And so for the wild type, we can see that it's brighter around the outsides. However, for the variant, it is much, much brighter in this central part. So it's getting stuck and degraded in the ER. It is never actually trafficking to the plasma membrane. So there are a bunch of different ways to try to correct for this. And you'll hear about some other ways later, but like my title said, our focus is small molecules. So there are a lot of different ways that a protein can misfold. And there are a few actual drugs currently in the clinic for some other diseases. On this table on the right, it lists them. But what they do is they're considered pharmacochaperone. So they come in, and as you can see, this is the ER, as I said previously, and this is our misfolded protein. The drug should come in to help stabilize this, or help maybe correct the structure, or correct it to where it can at least pass, and then traffic its way through the Golgi and actually make its way and get integrated into the plasma membrane. So there are some currently marketed drugs for CFTR that have successfully done this. So small molecules, we know, can help essentially rescue misfolded proteins, and we want to try to apply that here. So in regards to SLC6 transporters, with this, There's 19 of them in the SLC family. So for CT1, we are looking up here. However, also shared in the SLC family is norepinephrine, dopamine, and serotonin down here, which these have been very, very well studied, and that there are currently a lot of drugs on the market that also target the same method that I'm going to show you about here in just a minute. But as previously kind of introduced by Evander, thank you very much, that CT1, the creatine transporter, has this rocking kind of mechanism. So this is going to be that outward facing, and I'll show a little bit more clear model of this in just a minute. But this is going to be the outward facing model, and then kind of an outward facing occluded. We're going to go to an inward facing, occluded, and inward facing. And this is the sodium that is coming in, and the substrate in this case is creatine. So we want to try to apply kind of the same things that have been done for other SLC families. This is also a demonstration that was done with dopamine previously. So these left two panels, these are controls. On the right panel, this is bupropion and ibogaine, a psychedelic drug. Now this was applied to the dopamine transporter, but what you can see on the left is, in this case, we're looking at green. Green is going to be the amount of the protein that actually is trafficked to the cell surface. So there's very little in both the PBS and DMSO, again, in the controls. And however, in the bupropion, there is quite a bit more green you can see on these exteriors, but on the ibogaine, there is even more. Now, the reason why both of them work but differently is because they work at different points of the actual working mechanism for dopamine. So you have this, as said before, this outward facing model, and this is where it starts, where we've got creatine and sodium. And then it goes to this outward facing occluded, excuse me. And this one aligns with the bupropion, right? And so targeting this one, it does increase the expression. However, when you look at the ibogaine actually targets the inward facing occluded. And so this gives you a higher amount of trafficking of the protein to the plasma membrane. And so this is this inward facing occluded, and then it's released with just the inward facing model. So since this was previously demonstrated with other SLC family members, we wanted to apply this. And Chuck Kuntz, who has previously presented with ACD some of his work, he's a computational chemist in our lab who actually modeled these. And because we don't have a current crystal structure, we modeled these off of ones that are extremely similar, including dopamine, serotonin. And he did this for both an outward facing model seen on the left and that inward facing, the inward facing occluded models. And then from there, we screened a bunch, like a lot, thousands of compounds to see whether they would preferentially bind to the outward facing or the inward facing model of CT1, of the creatine transporter. So when we're looking at, on the left here is going to be the outward facing, and on the right is going to be the inward facing. So along my x-axis here is going to be if it actually prefers, for whether it prefers to bind like the outward facing or whether it's going to prefer the inward facing. So above this diagonal axis right here, all of these are going to have a higher preference to want to bind the inward facing model. And then we kind of have this one outlier that we can use as a control. So our overall general, my overall general experimental procedure with this is, for those that are unfamiliar, a depiction of kind of a cell assay plate. So in each of those wells, like A1, A2, A3, I will plate CT1 cells that have a green fluorescent protein immediately tagged to it. So it's going to show up as green. And then in each one of those, so in A1, I can put one drug, in A2, I can put another drug, in A3, etc. And then from there, we run it through an instrument called flow cytometer. So with flow cytometry, this gives us a lot of very, very useful information. In this case, I'm able to look at every individual cell, but I'm able to look at the health of the cells, as we can kind of see here with like this side scatter and this forward scatter. So it's going to tell me if they're healthy looking, but then we have different lasers that are attached to it. And so as it passes through, it will be able to determine the intensity of how much of each protein, of how much of fluorescent reporter for the protein there may be. And in this case, I'm using green. So we see channel 2 for this green. We're going to look at increases of green. And we did this in a stable mammalian cell line. So in this entire graph, every single one of these squares denotes essentially a small molecule compound that we tested. From Chuck's screening, we had about 53 compounds that were able to be commercially purchased. And so we went ahead and I tested all 53 to see. Now, white is going to be what my control is. And my control in this case was just no drug of DMSO. And so anything that is kind of a red tint, or the darker red it is, the better, it is actually going to increase from just general what it is. And now all of this was just done on wild type creatine to start with. So as we can see, there is 2 that fell out of this screen that we were kind of really interested in to work with a little bit further. And these are right now some of our top priorities. So B2 and C1, and they're just arbitrary names. These were the ones that we kind of looked at a little bit further. Now, B2 on this one, in the graph on the left, this is going to tell you how much of that green fluorescent protein, how much of that GFP there is. So an increase in GFP, because it's directly tagged to CT1, indicates that there is more CT1 being made. So on the left, this peak, this is just no drug. This was my control, just normal cells as to how much there's already being made, how much protein is already being made. And we see a 3 to 5-fold increase when this drug is added. And this was, just to verify, this was actually repeated a second time, and we did see the same results. Now, on the y-axis here is the actual cell count. So this is how many cells that we're seeing this. And so we do see, very nice peaks that indicate there is an increase. Now, this is for BO2. The other interesting thing about this is that BO2, its scatter plot, so these are called scatter plots, it resembled the one on the left just a little bit more. So on the left, this is going to be what a healthy kind of graph looks like, and every single one of these dots represents an individual cell that we tested. And so as you can see on the left with no drug, that there's this big intensity. And so this is kind of where we normally see healthy cells. And this tells us that since they're about at even equivalencies, that these are very healthy cells. Now down here in this bottom left-hand corner are going to be where our unhealthy cells are. They're abnormally shaped or they are dying. CO1, when we tested that and then we looked into it to each cell, you can see that it does not keep that very localized center, very healthy cell type, and that there is some cell death that is occurring. However, even with the cells that are either unhappy or abnormally shaped or dying, there is still just a little under 50% of an increase of actual creatine that is being detected. Now, what we don't know is if the drug itself is actually toxic to the cells, or if the drug itself is increasing the CT1 production enough where an overproduction of CT1 is what is accounting for the toxicity. So for our future directions for this, we have a couple things to do. Now, with the pharmacochaperone, we can correct it and get it to the plasma membrane, as I expressed earlier. But when we looked at this for the first part, this is just telling me total protein. So what I know is that there is more protein, but I don't know if that protein is actually making it to the plasma membrane. So first, we're going to look at if it is actually making it to the plasma membrane. So that's the localization. And then we need to look at the effects on the activity. So is it still functional? And are we recovering some of that creatine uptake? Because that is key. It's kind of a two-part sword, that we have to get it to the plasma membrane and make sure it can still uptake creatine. And then we're going to apply this to, we already have 10 variants, like stable cell line variants that are made. So then we're going to apply this to some of the variants that were previously made in our lab and see how it impacts all of them. And additionally, one of the speakers who is going to speak a little bit later today, Peter Axiro-Chilis, we have sent him some of these compounds to also help look at their function and try to help untease that for us. But I know he hasn't quite gotten to it yet. So that's also for a future direction for us. So we're working on it, but
I'm gonna do a time check here.
I'm good. That's it. Yep. And then just I'd like to thank my lab, and this is my lab at Purdue and ACD, and I use bridge programs. So thank you so much.
Thank you, Jackie. Thank you for the great talk, offering a ray of hope for finding a treatment there.
Trying.
And our next speaker is Dr. Longo. Dr. Longo is a distinguished figure in the field. He certainly was one of the first doctors I talked to after my son's diagnosis last year at the in-person conference. With MD, PhD in molecular biology and pathology from the University of Parma, Italy, Dr. Longo's journey led him to Emory University in Atlanta, where he received comprehensive training in pediatrics, medical, and biochemical genetics. Currently, he holds esteemed position as a professor of pediatric and adjunct professor of pathology, nutrition, and integrative physiology at the University of Utah in Salt Lake City. Dr. Longo's impact goes beyond academia. He also serves as the chief of the Division of Medical Genetics, director of the Metabolic Service, director of the training program in medical biochemical genetics, and medical director of the biochemical genetics lab at ARUP laboratories in Salt Lake City. His extensive clinical research revolves around the molecular basis of metabolic disorders, their identification through newborn screening, their natural history, and the development of novel therapies. Notably, Dr. Longo has a long-standing interest in membrane transporters, for which he works on amino acid, glucose, carnitine, and creatine transporters. His dedication extends to following several patients in brain creatine deficiency, developing new methods to facilitate their detection by newborn screening, improving existing therapies, and developing new ones for these conditions. He also serves as the head of ACD's Scientific Medical Advisory Board. Dr. Longo's talk inhibitors of arginine, glycine, aminidinotransferase, as a therapy for GAMT deficiency is a testament to his commitment to pushing the boundaries of medical science to bring meaningful advancements to those in need. Dr. Longo, please take it from here.
Thank you very much. So I'm going to try to share my screen. 1 second that I hope to get the right one, and I'm going to go. So what I'm going to talk today, I will not talk about a creatine transport, but that has been done very well by the people who spoke about me. I'm going to speak about inhibitor of glycine aminidinotransferase, because what we want to do is really to determine how guanidino acetate is synthesized and what strategy we can use to decrease the formation of guanidino acetate. Why do we want to reduce the formation of guanidino acetate? As you all know, in all disorder of creatine, in the brain creatine deficiency disorder, there is the lack of the creatine peak in the brain. And you can see here that the peak is totally missing, and this is a normal control. And that obviously by giving back creatine, we resolve several of the problems that is in this patient. However, when we look at patients who have guanidino acetate methyltransferase deficiency, even if we give back creatine, that by itself is not sufficient to prevent the development of seizure especially, and other problems in the brain. What does make the brain different from other tissues. What makes the brain different, and this picture is taken from Dr. Olivier Brassens, who will be at this symposium, is that unlike the muscle, the brain requires both creatine transport and the endogenous synthesis of creatine to function perfectly. So the muscle doesn't do that, or it does that with much less. So how do we replenish everything that we can in the brain? Obviously, we give creatine, but also in the case of guanidinoacetate methyltransferase deficiency, which is, is shown. Here, in guanidinoacetate methyltransferase deficiency, you block this enzyme, and again, you have the deficiency of creatine and the accumulation of guanidinoacetate. This makes this condition a little bit different from the other one, because again, we need to relieve the toxicity of guanidinoacetate. And in this disorder, in fact, in addition to creatine, we are giving chemicals that try to block the synthesis of guanidinoacetate by reversing the activity of the enzyme that synthesizes guanidinoacetate. Again, which one are these ones? So what we are doing, we are limiting the amount of arginine by restricting protein in the diet. We can give chemical that bind glycine to decrease its concentration. And also we provide extra ornithine to make this reaction going backwards if we can. Now, what would be the ideal situation? The ideal situation would be to block completely this enzyme, which is called AGAT, to see if we can reduce the formation of guanidinoacetate. So how do we do it? So the way that we do it is that we try to block the activity of this enzyme. This enzyme is expressed in multiple organs, the AGAT enzyme. In the peripheral circulation, it is expressed mostly in the kidney. And what the kidney do, they generate guanidinoacetate that will travel to the liver, and then it will be further converted into creatine, and creatine will be able to reach the brain. So keep in mind, again, that the brain does express, at least in some cells, the enzyme AGAT that will be able to, for the local synthesis of guanidinoacetate and subsequently of creatine within the brain. This is an enzyme which is expressed, it is localized inside the mitochondria. As a result, this enzyme has what we call a mitochondrial targeting sequence, which is a series of positively charged amino acids that will direct the enzyme inside the mitochondria. Now, if you isolate that enzyme in the native state, the enzyme will be very poorly soluble. So what we did at the beginning, we replaced the beginning of the enzyme with the targeting sequence with a sequence of amino acids that can be captured by specific column and then eluted from the column so we can analyze this enzyme. And with different steps that we have developed, we were able to obtain a pretty purified enzyme that was attached on the bottom of 96-well plate. And this enzyme was studied for the production of the two compounds that derive from arginine-glycine, the arginine-glycine aminidinotransferase. So you have the production of equal amount of ornithine and guanidinoacetate when you expose them to glycine and arginine that are the substrate of this enzyme. So the enzyme functions very well. The enzyme is now soluble, and we use the enzyme to determine how it could be inhibited. But before testing inhibitor, we did a virtual screen that was done by a company named Eaton-Wise, in which the 100 more positive putative inhibitor were identified to be tested in vitro to inhibit the reaction. How do we do it? We place the enzyme inside each one of these 96-well plates, and then we incubate the well with arginine and glycine. If the enzyme is effective, it will produce guanidinoacetate and ornithine. Obviously, we use positive control, control in which there is no formation of guanidinoacetate or ornithine, control in which we don't put any inhibitor, and a control in which we had an excess of ornithine. Ornithine is an inhibitor of this enzyme that would tell us that we are basically capable of inhibiting the reaction. We tested a large number of them, and we consider a positive hit the one where we reduce the formation of guanidinoacetate to less than 25% of normal. We confirmed some of the hit, and what we found that all of the inhibitors who were successful shared an arginine backbone. And this is obviously not surprising, since the physiological substrate of the enzyme is arginine. What we have completed since the last time, we were able to obtain a quantitative studies with some of these compounds, and we found that, for example, with compound D3, we were able to inhibit almost all of the reaction, but the half maximal inhibition was observed with about 70 micromolar. This is a little bit too high. We started studying derivative of the D3 compound, and we were able to decrease the ED50, but at the same time, we did not obtain complete inhibition. We had some intermediate compound that caused a better inhibition with an intermediate level of ED50. The lower the ED50, the more effective can be the compound. Now, to go to clinical trials to test in an animal model, we would need an ED50 which is in the high nanomolar range. So we still have to go down about not 1,000-fold, but almost 1,000-fold. So what are we doing now? So at this point, we have a good idea of the compound that can be used to inhibit our reaction. What we need, we are developing a higher throughput screening method to measure guanidinoacetate or ornithine that will enable to study many, many more compounds. We are also in contact with a company that can actually do the study for us, so the product can be further developed. So in summary, we have seen that GAMT deficiency impairs creatine synthesis and results in guanidinoacetate accumulation. Therapy with creatine administration is very effective in AGAT deficiency, while suppression of GAA synthesis is also necessary in GAMT deficiency. The inhibition of AGAT could decrease synthesis of guanidinoacetate, restoring the normal level of this compound in the brain and diminishing its toxicity. An inhibitor of AGAT together with creatine supplement could greatly improve the outcome of patients with GAMT deficiency, both diagnosed by newborn screening or identified later in life. And initial hit need to be optimized further to obtain more powerful inhibitor. I want to thank all of the people in my laboratory who have worked with me on this project, in addition to the people at ARUP who were able to measure all of this compound, and Dr. Neil Erickson from Atomwise who did the initial screening. But more importantly, I want to thank all of patients and their family and the Association for Creatine Deficiency for in part supporting this research, and especially to provide the impetus for further research in the area. I'm going to pass the microphone back to the moderator, and thank you very much.
Thank you, Dr. Longo. Hoping to see AGAT inhibitors develop to improve the outcome of patients with GAMT deficiency soon. Our next speaker is Dr. Peter Axarios-Elias. He is a postdoctoral fellow at the Department of Medicine and Psychiatry at University of British Columbia. His research in the last 12 years has been focused on various brain transporters and how they can be modulated by drugs and consequently used as treatments for epilepsy, schizophrenia, Alzheimer's disease, and other rare genetic diseases. He has recently engaged in the development of new treatments for creatine transporter deficiency. He has 16 years experience in generating lead drug prototypes for various protein targets associated with neurological disorders and rare genetic disorders. The title of his work is Restoring Function to Defective SLC6A8 Creatine Transporter Variants Using Small Molecules and Drug Repurposing. I'll hand it over to you, Dr. Exarios.
It's a hard name to get right. Thank you for the introduction. So I'm working on drug screening against CTD. So our long-term goal has been to identify an FDA-approved drug or natural product that could help this disease, mainly because you can repurpose drugs for use, and it's quicker. Obviously, you don't have to go through clinical trials. And obviously, we're trying to increase the function of the transporter or to increase its ability to go to the surface. The drug has to be safe. It has to be taken orally. It has to have a good safety profile, and it has to have no reported human toxicity. And ideally, the therapeutic dose that we're testing in vitro has to be achieved in the brain. And so the key here is to get a drug that can penetrate the blood-brain barrier. So there are several issues that has been discussed in previous talks. I'll briefly go over. So the transporter can get stuck in the ER. It's inside the cell, and so it's unable to function because it has to live at the surface of a cell to get the creatine inside. The second issue is the variant or the mutation caused it to misfunction. So if it's at the surface, it may not work at the surface. And so these are two issues that you can get a drug to rescue either the amount that goes to the surface or get it to function back to normal. So if we do get a drug that goes to the surface, it may not work still because it may have both issues. So dual therapy is also an option of finding an expression corrector and a functional corrector. So our stages has been to first for the diagnosis stage, stage 1, which is the make the DNA, to make the variants in the lab, and then you can express it in a cell. We usually use human kidney cells, and then you can test it. We use a method which I'll discuss later is electrophysiology, which measures the current when creatine gets into cells. And we can classify all the variants by partially dysfunctional, completely dysfunctional, completely functional, or hyperfunctional. In stage 2, we use a 3D structure of SLC6A8. We use it to help us to understand the landscape of the protein, and then we can design drugs or find drugs that will bind to the protein with high affinity, and hopefully either get it to the surface or get it to function. We can screen a lot of drugs. We can use supercomputers. We can screen 20 million drugs at a time, and then we can select the drugs that look like they're going to bind, and then buy them and test them. Stage 3 is the main thing, is to find drugs that do something. It could get like 20 to 30% back restoration. And then you do multiple rounds of screening based on those hits. You can learn a lot from those drugs to see what the drug should look like and what to modify. And therefore, that stage 3 is just screening, multiple rounds, and for each round, understanding what is needed and what's not needed. And then stage 4 is once you have a compound that you can get to the brain at good affinity, then you can test it in a more representative to a human being, is to test fibroblasts. And defibrillation or skin fibroblasts from patients who have the CTD or who have been diagnosed with CTD. So the screen, obviously, I don't see this. I don't watch this all day long. You do this docking. What other people have talked about is we take all the drugs and then we screen them virtually. They all get scored based on how well it fits into the kind of pocket. It's sort of like a key in a lock. Lock here is the protein. The key is the drug. And then you rank them, and then you rank them, let's say 100, and then you buy 100, and then you test them. So yeah, so you have a model. You do the virtual screen. You do a manual inspection, and you pick out the top 100 drugs, and then you test them in your assay with hopefully getting it. So here we're measuring activity or the ability for creatine to get inside the cells. So electrophysiology is a medium throughput method of, so you're able to, once creatine gets into a cell, it creates an electrical current around the membrane. You can put a electrode or a needle at the membrane of the cell, and it measures the current being applied. And that allows you to measure how much creatine is actually getting through. And here on the bottom left shows the red line is showing no creatine, no application of creatine. And then when you add creatine, you have a dip in the current, which then can be measured, and you can screen drugs in that manner. And then the creatine uptake assay, which has been discussed before, we use mass spectrometry to measure the absolute or the relative amount of creatine inside the cell. So this is the how much creatine is actually in there. And it can also measure metabolites of creatine, which is, I think, important as well. So we initially screened 30 variants with adding creatine, monohydrate. And you can see most of them are dysfunctional or non-functional, and a few are partially functional, and 2 were functional. So it really shows the importance of actually doing these experiments to know how the variant has affected the protein. So the first screen, we screened out with wild type. The initial stage, we found 6 compounds that had a little bit of activity beyond the normal wild type. And then we took those drugs and tested in the variants that we're going to do initially. And the first variant was the 408 del and deletion, and we've shown a little bit of a response with these drugs, a little bit of an improvement, about 20%. And then for the other deletion, ASN 3036, which is arguably the most frequent, and it showed a similar effect, about 20% restoration of transporter function. And it really shows that every variant will be different based on the drug. So it's really important to test. It's really a personalized medicine approach method. So all the drugs, once you have enough drugs that are active, at least a little bit, you can find what's similar about them, what's so common. So you can find a backbone, what we call sort of the portion of the drug that is similar to each other, and then we can rescreen, called an enrichment, which basically means you know something about the drug and you rescreen it with that information. And we got a little bit of an improvement, so about a 40% restoration of those 2 variants, and that was a good sign. And same sort of thing kept on going. So obviously drugs need to be purchased. Sometimes they're slow to arrive. So more and more can come. So we did the enrichment again screen, and we got improvements about final about 50%, which was good as well. And then the expression corrector, this is one, and obviously this is a drug that can push the transporters to the surface. So we can also use this as a method to get the, if the variant has affected greatly the expression at the surface, we can use this drug to help it get to the surface for some of the variants, not all. So right now we're focusing on these patient fibroblasts that are representative to CTD patients, and we're going to measure not only creatine, how much creatine is actually inside the cell, but also its metabolites. And arguably the most important metabolite here is phosphocreatine, because it is the molecule that gives the energy to the cell. If you don't have energy to the cell is unhealthy, may die. So the phospho part of the phosphocreatine is the group that actually makes the ATP, which ATP is the energy of a cell. And we're going to measure creatinine and guanidinial acetic acid as well. So the fibroblasts that we have in-house now that we've obtained from the Coral Institute, we have 2 wild-type cell lines at different ages. So the patients, one was young and the other one was older, 10-year, I think it's 4 and 10. And then all the variants for CTD at the bottom there. And we're going to measure it with mass spec. Mass spec is mass spectrometry, can measure how much something, quantify it. So we're going to measure, as I said, phosphocreatine, creatine, GAA, and creatinine. And we were able to measure this in fibroblasts first of a wild type. And we started with 4 years of age because it fits well with the other variants that are also 4 years of age. And we measured, so we used a labeled version of creatine, which here is called a D3. And it's basically creatine with just a label so that we can distinguish it from the creatine that's already in cells. Because that's the creatine that's already there, we're not interested. What we're interested is the ability for a cell to absorb the labeled version. And we measured at 2 different concentrations initially, the 10 micromolar of D3 creatine and the 100. And we show that there's more in the 100 than the 10, which is good. And then we use GPA, which is guanidinial propionate acid. It is an inhibitor for the SLC6A transporter. So we used it as a way to prove or to confirm that the uptake of creatine is actually through the transporter and not through other transporters. And that was incubated for 1 hour. And the other one was on the right there is 3 hours. At the 3 hour mark, similar pattern, 10 is lower than 100, and GPA was able to block it greatly. And then we measured phosphocreatine. This phosphocreatine is, like I said, the metabolite actually gives the energy of the cell. And that's important when it comes down to understanding how the homeostasis or the profile of the cell, is it healthy like wild type? Because if you don't have phosphocreatine forming for some reason, then it's no use. So here we added the original D3 creatine, and we quantified phosphocreatine at the 1 hour mark, and a similar pattern emerged. And then, of course, GPA blocked it. And then at the 3 hour mark, the phosphocreatine has increased relative to the 1 hour. And here we think that obviously that the creatine, if you leave it inside the cell for an extended period of time, the conversion from creatine to phosphocreatine is increased. And that's what we see here. And then GPA blocked it. So the summary is that we improved potency by using the enrichment method, which increased to 50%. Then we made this pipeline using mass spec to diagnose human fibroblasts. We've tested the baseline levels of all the metabolites in wild type fibroblasts, and we've confirmed that the uptake of creatine is mediated through the SLC6A channel. And our current aims is to test the fibroblast. The next step is to take that 408 del or the variant tested in fibroblasts. And we may also come down to combination therapy if needed, where we add the expression and function together. Also, we are going to diagnose specifically, which we haven't done yet, is the those fibroblast ones with electrophysiology. And it's important to have this cell line because, like I said, it's more representative to a CTD patient. And I'd like to acknowledge the ACD for all their support and their help. And obviously, Dr. Sylvia Stockler, Amina, who's been doing the cell culture of the fibroblasts, and Roger, who's been doing the mass spec, and like a Schellenbach group who gave us the compounds of their research, and we're going to test that using the electrophysiology method. So thank you very much, and to everyone for listening, and thank you.
Thank you. Thank you for the great talk. It reflected your dedication to the restoration of crucial biological functions through innovative approaches. And the last slide, I want to highlight the importance of the donation of fibroblasts from the community. Also, I think it's now time for some Q&As. So I'll hand it over to Laura Baroncelli and Sarah Brownstein.
Can you hear me? Yes. Okay, perfect. So I guess we can start with a question for Dr. Ferrada by Steve Baker. Stephen is asking whether in your docking study of creatine transit throughout SLC6A8, did you model creatine as a zwitterion? And if you have any insight into whether its ionization state can be maintained during the transit through the pore.
I have to think about that. So yes, it is ionized, I believe. We did several tests of this, but I think we do not have the resolution in the analysis, in the computational analysis, to answer that question. So the question is rather the ionization state promotes the transport in any other direction. Is that right?
Yeah.
Yeah, no, I don't think we do have the computational ability to tease that apart, because what we do is to create the molecule computationally, and that there is ionic states, different ones, right? The charges are there, but the docking is done locally, right? So we're not testing both conformers. We're not, to answer the question, maybe some molecular dynamics will be needed.
Okay, thanks a lot.
I think there was another question for you about whether your work would inform which mutations might be rescuable by pharmacochaperones, or maybe even responsiveness to creatine supplementation.
Yeah, so we believe so. I mean, there are many of the variants that we found that we see the pattern, right? The pattern of accumulation in the endoplasmic reticulum on the one side. And then we also do not see several of the other properties, right? Like the expression at the plasma membrane correctly, or we also see a decrease in function. Sometimes we do see this accumulation in the ER, and we also see effects on the stability, and sometimes we do not. So that's, I think, also an interesting way to tease apart this type of variants, as also it is the data on interaction with other proteins, right? We believe that some of this retention in the ER might be because of mutations that affect the interaction with these proteins, some others might not.
Okay, I'm seeing that Dr. Longo is already answering to many questions in the QA chat, but I think that some of them are for more speakers, so maybe we can go live. For example, one question that I am seeing to be repeated sometimes is, well, for all of them that are studying some therapeutic strategies, so Dr. Longo, Dr. Gallagher, and Dr. Axelios, how long do you expect that, let's say, the therapeutic development will take? So if you can predict, I understand that this is a very difficult question, but if you can predict somehow the timeline of your experiments would be great.
I think that for all of us, I think I can answer for everybody, it's very hard because the first thing that you have to do, you have to identify a drug. The second thing, you have to make sure that it is not toxic. The third thing, you have to make sure that it gets inside the brain. And those are all things that require some time. Then once you identify the drug, you move to the animal model to produce preclinical data to determine whether they're effective or not. And finally, once you have this data, you can move to the clinical arena. In the best case scenario, I mean, this would be at least 5 years. Some of the questions, the other questions that I've received is, for example, in the case of inhibition of AGAT, if you don't make homoarginine, can you worsen the cardiovascular state? Well, the problem is we have patients with complete AGAT deficiency and they seem to be doing just fine. Remember, homoarginine is kind of an association, but we do not know if it is a cause and an effect. Based on what we see in patients with AGAT deficiency, probably it is not a cause and an effect. The second thing, there were people, one question was, can you use a chaperone to stimulate the activity of GAMT? Yes, you can. We have not been pursuing that, but and the advantage of GAMT deficiency that many of the mutations that we have there are actually missense variant. Many of the patients that I have with creatine transporter deficiency have nonsense variant that they will not be amenable to a chaperone therapy. So, but that is a very good suggestion. I think that we might need to look into that as well. Thank you.
Thank you, Dr. Longo, for answering all these questions. There is one more question in the other chat for Dr. Axelios from Jerry Lipschutz. He asked whether does GPA get transported like creatine through SLC6A8, or does it block SLC6A8? Or is it not known?
So I think, and people correct me, I think it blocks the, it goes to the same place as creatine. So it competes for creatine. So when we use it, we use a very high concentration of GPA so that it completely blocks it and doesn't allow creatine to get through. So it should share the same, it goes to the same area basically.
Okay, thank you.
So I think that one of the questions is how much creatine transporter activity do you need to put back?
Yeah, I was just asking that.
To correct the phenotype. So in other words, how much do you want to put back?
What is the dose needed?
The dose needed to add for the GPA, you mean? Or for the creatine?
No, for creatine.
Oh, it depends on it would depend on the dose. It would depend on the dose that you add versus the dose that you use for the GPA. Is that the question? Maybe I misunderstood.
Question is just what's the target activity level to confer efficacy in terms of transport?
Of creatine?
10% of normal creatine.
Oh,
transport,
50% normal.
50%. Yeah.
50% normal.
And is that just because female carriers? Are you basing that on some degree? Then
no, no. It's just that's probably we're not getting the full. Maybe there's a limit, or maybe it's not potent enough, not strong enough.
So I do have a patient whose cell retain about 20 to 30% creatine transport activity, and he is symptomatic. As the patient reported previously, he responded to high-dose creatine supplement with an improvement in behavior. So in other words, if you have probably some residual transport activity, you do better. But 20 to 30% is not enough to prevent clinical manifestation. I'm sorry to say this, but that's how it is.
Now I have some questions for Dr. Gallagher from the audience. They are asking what is the size of the compound library used for the virtual screening? And you mentioned that there is a compound, that the compound showed toxicity from CT1. What is the mechanism are you considering?
Okay, so for the first question, I believe our library size was around 3,000 molecules or so that we screened. And the basis of it was we tried to identify compounds that had like a similar kind of core structure as creatine. So we started with those to be able to look for as these structures and stuff. So it was about 3,000 to target. And then the second question was for the toxicity?
Yeah.
Okay. So I should clarify that for the toxicity, this was in cell models. And it's previously kind of been demonstrated in the literature. There's some examples, and I've seen it just in general across the board that an overexpression of creatine in cell models, at least, can lead to toxicity or cell death. And so previously, some studies have used like a creatine-free media and different types of media or supplemented it to make sure that it wasn't present for overexpression. And so again, I don't know what the cutoff is for what is actually deemed like toxic, that it's just too much. And again, I don't know that this wasn't just a toxicity of that individual compound on the cells in general, not necessarily creatine. It could just be an uptake in that and just toxic to the cell as itself.
Okay, thank you. Do you think that the osmolytic action of a creatine can be one of these factors?
Can you repeat that one more time? I'm sorry. Do you think that
the osmolytic action of creatine can be one of the factors leading to toxicity of the overconcentration of creatine inside the cells?
I mean, we'd have to take into consideration the sodium gradient too.
Mm-hmm.
So I just don't have the available data right now to be able to answer that. I don't think that would just take some investigation to figure that out.
Mm-hmm. Yeah, for sure. Thank you. Okay, so one more question for Dr. Ferrada. Does your model can be used also to predict the impact of single point deletions on the stability of SLC6A8 protein in addition to variants?
You said single point deletions.
Yes, this is the question from the audience. Yes.
Yeah, I believe so. Then the model should be redone for that particular version of the transporter. And if it has a quality, if the quality is good enough, I think we can use the same pipeline to make such a prediction. Mm-hmm. But it will depend on where the deletion is. It might really be disruptive, but if it's a variant that exists in human population, it's likely that it's not. And yeah, the same pipeline could be applied.
Thank you. We'll do a quick time check. There's, I think, one more time for one more question. Sarah, is there a question that you would like to ask from the chat?
I think this one from Dr. Gallagher was interesting, whether there are any similarities in structure in terms of the hits that you'd identified so far.
As for the actual molecules, I mean, a lot of them, we kind of started with like a creatine base, so there is some similarity. Similarity is in way of like how the structure is interacting with like the protein or variants. We're not aware of that. We haven't investigated that yet. So that's going to be part of kind of the future work for the functional studies that will be able to put everything together.
Great, thank you.
Any other questions? I think we have about a minute and a half. for the Q&A session.
I think there was a question around how long the fibroblast testing stage took. Dr. Sirio.
How long, so the wild type, we've got used to handling the fibroblasts. They grow a little bit slower than other cell lines that we used. So the setup took a while in terms of getting the mass spec right and getting the cells, the treatments, getting everything, timing everything right. So it was slower than expected for the wild type, but I think we're ready to go to test the variants that are available. So we're ready to go. So it took, months to getting everything right and the data to be kind of stable.
One very last question for Jackie, very quick. Can you differentiate between the GFP tag induced toxicity from, well, CT1? I mean, do you think that the tagging with GFP somehow can change the conformation of CT1? This was a question from the audience, but I rephrased it a little bit.
Sure. Yeah, that's a great question. So it's known to be functional with it. And, we've previously seen from it that it still traffics, the wild type, even this way, still traffics to the plasma membrane. So that's specifically kind of why we use this fluorophore to detect it, because we know that it shouldn't structurally impact it because it still traffics and still functions. So yes, at least for a wild type, it is still functional. It shouldn't be impacting it from anything that we've seen in our previous literature studies.
Thank you all. There are still some questions in the Q&A box, and some questions were answered in the chat. So please check your Q&A section if you asked a question. It may be answered via chat. And I'll hand it back to Sangeeta. Thank you, everybody.
Thanks, Sang, and thank you, everyone. I know we've been going strong without a break, but bear with us for one more session, and then we will break. So at this time, I'd like to invite the speaker of our next session, Dr. Jenny Goldstein. Hi,
yes.
Jenny.
Hi, Sangeeta. Hi.
I'll just give a brief introduction and then hand it back to you. So Dr. Goldstein is an assistant professor of genetics at the University of North Carolina at Chapel Hill. She's worked as a pediatric genetic counselor and clinical research coordinator at Duke University Medical Center for 15 years. While at Duke, Jenny also worked as a genetic counselor with families with GAMT deficiency and coordinated research on newborn screening studies for GAMT deficiency. Now at UNC, Jenny works with a clinical genome resource, an NIH-funded effort to create a publicly available resource on gene disease clinical validity and the pathogenicity of genetic variants. Jenny's no stranger to our community. She's been a part of it for a very long time. She was at the caregiver session last weekend. It's my pleasure to welcome you back here for this session. Jenny's talk is titled The ClinGen Variant Curation Expert Panel for Cerebral Creatine Deficiency Disorders, Classifying the Clinical Significance of Variants in GAMT, GATM, GAMT, and SLC6A8.
Great. Thank you so much for that introduction, Sangeeta, and hopefully everyone can see my slides. So just really appreciate the opportunity to talk to everyone today about the work of our ClinGen CCDS Variant Curation Expert Panel. So I'm going to start just by describing a challenge in clinical genetics. So with new sequencing technologies, we now have the ability to sequence the entire genome of a person in less than a day. However, given the genome of one person will have literally millions of differences compared to the reference genome, how do we know which, if any, of those variants identified in a person's DNA are actually causing developmental or health problems? So this question was really the motivation for starting the NIH-funded Clinical Genome Resource, or ClinGen for short. So the mission of ClinGen is to create a publicly available resource that defines the clinical relevance of genes and variants for use in precision medicine and research. And since it was initiated in 2013, there are now over 2,200 ClinGen investigators across 56 different countries. So one of ClinGen's core curation activities, as shown here on the left, is variant pathogenicity. So what that really means is, how do we determine whether a particular genetic variant is causing disease in a person? So in order to help determine the clinical impact of genetic variants, ClinGen uses guidelines that were developed by the American College of Medical Genetics and the Association for Molecular Pathology. And those guidelines were developed back in 2015. And as you can see here on the left, the guidelines basically allow us to use lots of different types of evidence in order to assess whether a particular genetic variant is causing disease or not. And for each of those types of evidence, they can either support that the variant is pathogenic or causing disease, or benign, which means it's not causing disease, it's just there in your genome and isn't really doing anything in particular. And so for the different types of evidence, you can actually apply different weights of strength. And then once you've collected all of your evidence, that may come from the medical literature, from diagnostic testing labs, from different databases, you put all of that information together, and then the combination of these different criteria that were met and their strengths allows you to classify the variant. And there are 5 different classifications that we typically think about. So pathogenic means that we are fairly sure that this particular variant is causing disease. And on the other end of the spectrum, we have benign, which means the variant is not causing disease. In the middle, we have variants of uncertain significance, which means that we really just don't know. And sometimes that can arise because we just don't have enough information. And sometimes it can be because there is conflicting evidence. So some evidence says yes, and some evidence says no. So while these criteria have been really helpful in standardizing variant classification, they were written for a broad range of different Mendelian disorders without guidance for specific genes or specific diseases. So for example, what sort of functional assay can I use in order to determine if an SLC6A8 variant is causing disease or not? Or how common is too common for a variant to be causing GAMT deficiency? So in order to address these challenges, ClinGen assembles groups called variant curation expert panels, which are groups of individuals with expertise in a specific gene or disease area. And what they do is to provide disease-specific specifications to those ACMG criteria for different genes and diseases. And the group then uses those specifications to evaluate evidence to classify genetic variants with respect to a particular disorder. So as I'm showing on the bottom here, this is a stepwise process that the group goes through. And then ultimately, the group can submit their variant classifications and supporting data to public databases. And this includes the ClinGen evidence repository and also NCBI's ClinVar, which is a really large genetic database. It's public, freely available, and provides a lot of information from many different submitters, including clinical testing labs, researchers, and so on. And for ClinGen expert panels that submit our data to ClinVar, it is part of an FDA-recognized database. So one of the 62 different variant curation expert panels that ClinGen has formed is for creatine deficiency syndromes. And so you'll see our group members here on the left, and you may recognize some of them. Some have already talked today, some will be talking later at this conference, but I am very delighted to be a part of this group. And so what we have done is to develop guidelines to assess the clinical impact of variants in all 3 CCDS genes. And as of last summer, our group was approved to submit variant classifications and supporting data to those public databases, so ClinVar and the ClinGen evidence repository. And that then, of course, allows anyone who wishes to access that information the availability to do so, whether that be clinicians, researchers, families, anyone can have access to that data. So I'm showing here just an example of how variant classification works. Obviously, I don't have time to go through all of our different criteria in detail, But this is a screenshot of our ClinGen variant curation interface. And what we can do here is to enter the data. As I said, that data can come from many different sources, like the published literature, clinical diagnostic labs, databases, and so on. What we can do is enter the data. We can apply the different evidence codes at the particular strength that we wish. And then our interface will then classify the variant according to the codes met and the strengths were used. So this is an example of a pathogenic variant in SLC 6A8. And then just another example here. This is a variant in the GAMT gene. This is a different classification. This one was classified as benign because it's very common in the general population. So what I'm showing here then is what our entries would look like in the National Center for Biotechnology Information's ClinVar database. As I mentioned, this is a large database of genetic information that accepts submissions from many different sources like clinical labs, researchers, and so on, and also expert panels. So this is for a variant that is commonly found in individuals with GAMT deficiency. So you can see here the overall interpretation for this variant is pathogenic. You can see that the information has been reviewed by our expert panel, so it gets the FDA recognized database tag. And then if you were to scroll down the page, you would see information from each of the different submitters, including here our expert panel. And if you were to click more, you would see a summary of all of the data that we collected and how we assessed that data. And then there's also a link to our ClinGen evidence repository as well. So just to summarize, we've now submitted 160 different variant classifications to ClinVar across the 3 different CCDS genes. This is just a kind of overall breakdown. So we have assessed a fair number of loss of function variants. So these are typically nonsense frame shift and canonical splice site changes. And as expected, the majority of these were pathogenic or likely pathogenic just by the nature of the type of variant. We've also classified a bunch of other types of variants like synonymous, intronic, in-frame insertions and deletions, and then also almost 80 missense changes. And perhaps not unexpectedly, you'll see a large percentage of these missense changes are variants of uncertain significance. And this is not at all unusual when we look at variant interpretation for different genes across many different clinical domains. Missense changes can just be notoriously hard to classify, especially if there's not a lot of information available on them. So this is something that we're still sort of working on. The one thing I wanted to address then, obviously, if you have a variant of uncertain significance, it's really not helpful because we just don't know if it's causing disease or not. So what can help us then to resolve these variants of uncertain significance? Well, one of the key things that we really need is more data. So I would highly encourage patients and families to participate in the ACD registry and would like to thank everyone who's already done so. I'm going to talk a little bit more on the next slide about how the registry data how we're using that data in our work. Clinicians, if you're publishing any cases, of course, with the consent of the patient and family, just including any detailed phenotype, biochemical data, including things like enzyme activity, analyte levels, and so on, and genotyping data. Even if that has to go in a supplemental file, it's just incredibly helpful. And then also to consider choosing clinical labs that will share data with ClinGen. Excuse me, researchers, we've already heard just some fabulous talks about prediction of variant effect. And then also development of well-controlled functional studies is very helpful for us as well. And I know we're going to be hearing some talks on that also. So this is really fantastic to see how this work is moving ahead. And then updated recommendations as well in terms of variant classification. So just to talk a little bit about a couple of those points, the first thing I wanted to talk about is the work of the ACD registry and how we are collaborating with the registry. So the registry has already shared a list of variants with us, 25 variants in SLC 6A8. So of course, that's the gene for the creatine transporter, and 12 in GAMT. So we have this list of variants, but we're also hoping to get some additional information from individuals with these variants that can really help us with the variant classification. So that includes things like biochemical data, things like creatine and guanidino acetate levels, any magnetic resonance spectroscopy data, any parental testing results, and so on, to look at inheritance in the family. But what we've done so far with this list is to collect all available, publicly available data. So this is, again, is coming from published literature, databases, and so on. And we've used our classification guidelines to classify these variants based on the information we have. So on the right here, I'm showing the classifications of those variants. And then our next step is to prioritize these variants regarding which of them could really benefit from additional information. Obviously, if we've already classified them as pathogenic or benign, no amount of additional information is going to change that. So those they're done, they're finished, and we don't need to, they're in the database, we have their classification. The ones that are most likely to benefit then are the likely pathogenic variants, and of course the variants of uncertain significance. So our next step will be then to request additional data from the ACD registry. So if you are a part of the registry, and if in the near future you see a message from the registry, please do consider providing this information if you're comfortable doing so. It could just be incredibly helpful to these classifications. And then we will incorporate that data, complete our classification, and send it on to ClinVar. So the other point I wanted just to mention very quickly is the use of updated recommendations. And so new guidance that came out recently from our ClinGen sequence variant interpretation working group, which oversees all of our expert panels, is regarding the use of in silico data or computational algorithms, which can really help us to predict, in this case, whether a missense variant is affecting the function of a protein or not. And one of the algorithms that we use and that was covered in this paper is called RAHHAL. And what it does is to provide a score from 0 to 1, with 0 meaning that the tool is predicting that that variant has no impact on the function of the protein, whereas 1 is suggesting with very high certainty that there would be an impact on the protein. And so before these recommendations came out, we would use this in silico evidence, but only at a very low level of certainty. But now, as you can see, based on the score for a particular variant, we can now actually apply higher strengths for certain variants dependent on their RAHHAL score. So that really can impact the classification of the variants. So we took a look at this by taking variants of uncertain significance in the 3 different genes, and then we applied the new recommendations with these new thresholds for the RAHHAL scores, and we reclassified the variants based on that. And as you can see here, in about half of the cases, we were actually able to reclassify them. So certainly a number of them actually went down to likely benign, and then some went up to likely pathogenic. So we have actually looked at the genes individually just to see how comfortable we would be applying these new recommendations for each gene. And I think for GATM and GAMT, we're pretty comfortable doing that. We had basically compared the classifications with no in silico data and then how that compared to the RAHHAL scores, and we were pretty comfortable doing it for the 2 enzymes. We're still investigating SLC6A8, and I've just been so excited to hear all the talks about variant effect prediction for this gene, because I think we definitely want to follow up on that and just determine whether that's something that we could use for SLC6A8 as well. So just to finish up, that was a very quick fly through our expert panel's work, but for future directions, we definitely want to continue working on trying to resolve some of those variants of uncertain significance and also conflicting classifications in ClinVar as well. As I mentioned, more data always helps, and certainly hearing a lot of the talks here today, I will definitely be reaching out to some folks to see how we might be able to collaborate. Of course, the ACD registry is a wonderful way that you all as patients and families can share data as well, so really appreciate that. We also want to look into classifying all published variants using our classification guidance. And we are working on publication of a manuscript of our classification specifications. So finally, just so many people to acknowledge, and thank you so much to all of the folks in the ClinGen CCDS VSEP. I'm showing them here, and various folks in ClinVar and ClinGen, and also, of course, to the Association for Creatine Deficiencies for this opportunity. Thank you. And I will pass this over.
Thank you so much, Jenny, for that walkthrough, all of the wonderful work you're doing. I'm going to keep us going on the agenda. And next up, I have a recording from Dr. Ingoglia. Couldn't be here in live today, but he was kind enough to pre-record a presentation for us. So I'm going to be playing it on my screen. So while I bring this up, can you guys tell me if you see this fine? A blank screen. I know you're seeing a blank screen for now, but hopefully you are seeing the blank screen. And I'm stopping before I start the recording because I wanted to tell you a little bit about Dr. Ingoglia before we get started on his talk. So Dr. Ingoglia is a biochemical geneticist and a assistant professor in the Department of Pathology at the University of Utah. He's also the medical director at the ARUP laboratories. With his background in biochemical genetics, he's focused on creatine metabolism in patients with urea cycle disorders and ultimately on the development of methods to determine the effectiveness of potential therapeutic agents for the treatment of GAMT deficiency. And with that brief intro, I'm going to turn this video on and wait for some feedback.
Hello, everybody. I'm Filippo Ingoglia. I'm an assistant professor at the University of Utah and a medical director at the RAP laboratories in the biochemical genetics lab. And today I will talk about functional studies in creatine transporter deficiency. Just before starting, please let me thank the organizers to having me here presenting this project today. So thank you. I have nothing to disclose. And during this presentation, I will talk about how creatine transport is measured and how creatine functional studies can support a creatine transporter deficiency diagnosis and the classification of the SLC6A variant. So let me take the pointer. I think it will be easier now. Okay. So the creatine transporter deficiency that I will call during this presentation CTD is one of the 3 cerebral creatine deficiency syndromes. So the gene involved in the CTD is the SLC6A8 gene, and is located on the X chromosome. So we are talking about an X-linked disorder. The gene will encode for the creatine transporter, the CT1, and the lack of the transporter will impair the supply of creatine into the brain. So the CTD accounts for about 1 or 2% of all the X-linked intellectual disability, and patients are presenting with developmental delay, intellectual disability, hypothonia, seizures, and autistic-like behavior. So for the diagnosis, so the disorder can be diagnosed through using a magnetic resonance spectroscopy or using also a biochemical test, evaluating and observing an elevation of creatine-creatinine ratio in urine, or doing DNA sequencing. So for the therapy, there is not an effective therapy for the CTD, unfortunately, but some patients are able to respond to the creatine supplementation and also to the supplementation of the creatine precursors as arginine and glycine, so the arginine and glycine. The outcome is a static encephalopathy with seizures and responsive to standard therapy. So let's focus a little bit on the CTD diagnosis. So the diagnosis relies on the measurement of the urine creatine-creatinine ratio. And if this ratio will be elevated, so the CTD the diagnosis needs to be confirmed by using a magnetic resonance spectroscopy, possibly showing the lack of the creatine peak into the brain, and demonstrating also a pathogenic variant through an SLC6A8 gene analysis. But these two techniques, they can present some difficulties. So the magnetic resonance spectroscopy is not present in all the institutions. So just specialized institutes have the magnetic resonance spectroscopy. And it needs also the sedation of the patient. So about the gene analysis, it's easier, but sometimes can be inconclusive. So the gene analysis can identify variants of unknown significance, so what we call VUS, or can miss the identification of a variant, possibly located in a non-coding region. So in those cases, a functional assay can confirm the diagnosis, can definitely confirm the diagnosis. So when I'm talking about a functional assay, is a test that will allow us to measure the activity of the transporter, so the activity of the CT1 transporter. And so at the end of the test of the functional assay, we will know how much creatine the transporter is able to import intracellularly in a specific amount of time. So for a functional assay, two steps are needed. So the first step, this functional assay is designed using fibroblasts. And the fibroblast needs to be isolated from skin biopsy. So it's needed a skin biopsy from the CTD patient. Then once the fibroblasts are isolated, together with the normal control fibroblast, so the normal control fibroblast, they need to be plated. Once ready for the second step, they will be incubated in the presence of creatine. So I talk about this second step. So I refer this. So identify this second step, sorry, as the transport, the actual transport study. So let me change slide. So just to focus on the second step. So this is a functional assay that won't use any radioactivity. So we will use, so it uses stable isotope. And for the quantification of creatine, it uses an LC tandem mass spectrometry system. So again, once the fibroblasts are ready to go through the second step, they will be incubated in presence of creatine. So here is just a representation of what can happen during the creatine incubation, and when we have fibroblasts from a patient with a zero activity of the transporter compared with the normal fibroblasts. So at the end of the incubation period, so we do not have any accumulation of creatine in the intracellular space for the CTD patient, but we will have, the creatine accumulation in the normal fibroblast. So then at the end of this accumulation step, the intracellular content will be extracted and prepared for the LC tandem mass spectrometry analysis. It will be also important to keep those cells into the plate because the protein needs to be quantified. So it's important to have those the quantification of the protein because they are going to be used for the normalization of the values. So here is what transport a functional assay can say, and the information that we can take from it. So here we have the transport values. Here is just a representation of 2 CTD patients compared with a normal fibroblast, but different patients. So one patient with no activity and a patient with a residual activity. So this assay should be sensitive enough for separate and identify patients with residual activity in order to identify those patients that can benefit from the creatine supplementation. The functional assay can be also very useful for the investigation of SLC6A8 missense variant and evaluating the impact on the CT1 activity to supporting and evaluate supporting the classification of those variants. To do that, it will be necessary to immortalize CTD fibroblasts with zero activity of the CT1 transporter. They can be immortalized using an SV40 system, and once are immortalized, they can be transfected with plasmid vectors. We will have plasmid vectors just carrying like a normal SLC6A8, but we can also create those plasmid vector carrying, for example, a VUS that we want to investigate. And that is possible just through a molecular technique, the PCR-mediated side-directed mutagenesis. So once we have the mutated, so the variants on the SLC6A8 gene, and so the plasmid, so the vector is going to be ready, we can transfect those immortalized fibroblasts with those vectors and let the cells express the mutated protein. At that point, we can compare those immortalized fibroblasts carrying the mutated protein with the immortalized fibroblasts carrying a normal CT1 protein. So going through the functional assay and evaluate the creatine transport. And here is just an example of what we can observe if we express a VUS that actually will be can be classified as a pathogenic variant. So in this way can support the classification of that VUS. So in summary, creatine transporter deficiency impairs the supply of creatine into the brain. Treatment for CTD include the supplementation with the creatine, or alone or in combination with the creatine precursors. And patients with residual transport activity can respond to the treatment. And that's why it's very important to identify those patients. And definitely you can identify those patients just running a functional assay. So a genetic test can miss variants outside the coding region or can detect the VUS. In such cases, a functional assay can be used for the confirmation of the diagnosis. So the expression also of missense variants in cells without any creatine transport activity can assess the functional effect and characterize the VUS of the SLC6A8 gene. So thank you. Thanks to all of you present, virtually present here. Thanks again to the ACD, University of Utah and REP laboratories, and my collaborators that will work with me on the development of this project. So Dr. Longo, Dr. Pasquali, Dr. Lai, and Dr. Balakrishnan. And of course, thanks to all patients and their family. So thank you.
Okay, so that was a recording from Dr. Ingoglia, and hopefully that shed some light on the use of fibroblasts, the need to characterize patient mutations, and I hope it's something that resonates with everybody here in the audience. I'm excited to welcome Dr. Baxter, who's our next speaker. Hi, Dr. Baxter. Very happy to have you here. For everyone in the audience, Dr. Baxter is the Associate Director of Genetic and Genomic Data Sharing and a genetic counselor in the program in Medical and Population Genetics Translational Genomics Group at the Broad Institute of MIT and Harvard. Her work has a strong focus on data modeling, scaling processes for data sharing, and most importantly, partnering with patient advocacy groups to improve genomic research. We first learned about Dr. Baxter's work through an opportunity that came to us as part of the Chan Zuckerberg Rare as One network. And our initial interactions with Dr. Baxter led to her taking a much closer look at CCDS and understanding the prevalence associated with these diseases. I will hand it over to you, Dr. Baxter, now for your talk.
Thank you for that intro. So let me go ahead and just share my screen. So as mentioned, I will be talking about the genetic prevalence work that I perform, and specifically I'll be focusing on GAMT. I recognize there's other genes as well, but I'll talk about some of those limitations and why we chose to focus on GAMT for this work. So this study is actually part of a larger study that involves all of the Rare as One network and within the Chan Zuckerberg Initiative, or I should say all recessive groups within the Rare as One network. And it's using data that already exists out in the clinical ecosystem or clinical genetics ecosystem and bringing that information together to estimate the number of individuals we think have a recessive rare disease. So quickly before I start, there's a few terms I just want to make sure we're all on the same page. And the first is the one that I'll be using the most, which is genetic prevalence. So we often hear about the word prevalence and incidence in the setting of counting the number of individuals with a certain diagnosis. But genetic prevalence is something slightly different. Genetic prevalence is the estimated proportion of a population that has a causal genotype for a genetic disorder. The reason I want to point this out is because it's different than most of the definitions we hear when we think about prevalence and incidence. This is really taking a different lens and trying to figure out how many people are there with a certain diagnosis. And we use population data, which I'll talk about in a second, to kind of take a sideways approach to say, based off of the number of individuals out in the population that are carrying this disease-causing variants, then how many people would we expect for recessive disease to have an inherited one copy from their mom and one copy from their dad? The reason we want to make this very clear is that there's a couple things that come along with these numbers. The first is that this is an estimate only and can change over time with new data. So the entire kind of approach of this is based off of our understanding of what are the disease-causing variants for a particular variant. And I think over the last couple hours, you've definitely seen talks that show you that this information can change over time. And also, how frequently do we see these variants circulating in the population? Population databases have grown over time and they've improved overall with diversity, but we're still continuing to work on that. And so both of these databases changing and our understanding changing can change the way the estimates come out. And I'll show you exactly how that worked out with GAMT over the last couple years. Also, I just want to remind everybody, genetic prevalence is not a direct count of a number of individuals with a clinical diagnosis. Again, we're taking a sideways approach of saying, how often do we see disease-causing variants circulate in the general population, and then estimating the number of people we would expect to be born with a certain diagnosis. And this all depends on the list of disease-causing variants that we've seen. And again, as you've seen through some fabulous talks, this list and understanding of what are the disease-causing variants is something that's dynamic. Also, quickly, just to call out what population databases are, because I know not everybody is familiar with that. A population database is a collection of genetic and sometimes phenotypic data. So both the sequence data that goes along with it, and then phenotype, do they have a certain diagnosis? Do they not? Are they considered an overall healthy control, or were they recruited for a specific disease for some reason? All of this information goes into a central database, and they are often trying to get a snapshot of a certain population. So this could be something regional, specific to a hospital, something national, specific to a country, or something global, where we're trying to really get a snapshot of the entire world. The reason we want to call this out is because there's certain assumptions and limitations based off of the different databases that we use. For the rest of this talk, when I talk about population data, I'll be talking about the database that we use, which is GNOMAD, which is currently one of the largest and most widely used publicly available collection of population data. Another thing I want to call out is that it is not representative of the entire world's population. So there are absolutely continents and countries that are not covered within GNOMAD. And so those are limitations we always have to remind ourselves when we're doing these estimates. So the whole purpose of this study and why we've partnered with the Chan Zuckerberg initiative and the Rare is One network is really to help answer the question, how many of us are there? I've talked with many rare disease groups, and at no point have I talked to them and they've said the traditional definition of incidence and prevalence, where we go into an EHR or we reach out to clinics, are an accurate representation and we think we've captured everybody. So we've come up with a way of using an equation called the Hardy-Weinberg equation, which is sort of the foundation for genetic counseling itself, which is the kind of mathematical equation that represents that the frequency of variance should remain constant from one generation to the next. And we can kind of use that approach to then estimate genetic prevalence. This came about and started to be used more heavily with population data in this particular method in about 2015. But what happened early on was that we were very trusting of the data that was out there. And we said, if somebody else called it disease causing or said that it would, disrupted the way the protein functioned, we would include it in the math. And what actually ended up happening is that we were really overcounting the number of individuals. And so my team over the last 6 years has developed an approach where we use the curation techniques that Jenny so wonderfully presented, and some other techniques that we've developed in-house for diving into population data to get closer to this true genetic prevalence, where the number represents genuinely the number of individuals that would have a genetic diagnosis, and in this case, 2 disease-causing variants in GAMT. So there's a few things that we always think about when we look at these numbers. The first is representation. So are we concerned that there are missing or underrepresented populations that we believe this disease could be more common in? Curation, I think you've already probably gotten sick of me talking about this, but the importance of making sure that every variant we include in the list has been seen to be disease causing. Clinical sensitivity. So in the last talk, you already heard about there are certain areas of a gene that can be harder to sequence and there may be variation in there. We really want to capture and understand when we're looking at a particular gene, how well do the average clinical tests capture everybody that would have a diagnosis. Your group is particularly lucky in the sense that there is a functional assay that can be performed as well, and that's really helpful for understanding clinical sensitivity. And then also disease spectrum. So things such as symptomatic carriers, so people who only have one copy, but actually may have some symptoms that keep them from being enrolled as a control in a study or may keep them from wanting to participate in research. Impacts to life expectancy, particularly whether we know whether every variant combination is even compatible with life. Because again, genetic prevalence includes the estimate of anybody who could have any 2 variants that are out there. If the combination of 2 particular variants actually would result in recurrent miscarriage or unrecognized pregnancies, those would actually be captured in the numbers. And so we always want to compare these numbers with other incidence and prevalence numbers to make sure that we're not missing either side of the spectrum, or if we are including them, that we can talk about that. And then reduce penetrance and variable expressivity, just the general sense of what is the disease spectrum and are we capturing everybody? So how do we build this puzzle, and how did we build this puzzle particular for GAMT? Well, we first start with our sources. And so for our sources, the main databases that we use are first the ones where we say, what are the variants known to cause disease? And by far and away, the number one for that is ClinVar, which is an open source database that is a place that researchers, clinicians, even patient organizations through a portal called Genome Connect can deposit their variants, and also more importantly, the evidence for why those variants they believe are disease causing or not. And we can go through and gather that information and look to see which of the variants we want to to bring into our pipeline. We also have a paid for one that is not open source, which is HGMD. They do a different approach. They scour the literature for any time a variant has been listed in a particular gene, and then either listed as DM, which is disease mutation, or DM question mark, which means they're not completely sure about it. And then any disease specific databases or partnerships with patient organizations that may have a list of variants will include those in the pipeline as well. For the question, how often do we see these variants in the general population? Just like I said, that's what we use NOMAD for. And so NOMAD is an aggregate of exome and genome information and returns out allele frequencies, which basically is saying how often do we see a variant floating around in the population. And we can use that information together to then come up with our list that we then go on to curate, as Jenny had pointed out. And in this particular research, we were actually lucky enough to be able to use the piloted guidelines from their group back in 2021. But then we're able to go back and as they developed further their pipeline and perform curation, we could actually use that information to update these numbers, as you'll see shortly. So quickly, I just wanted to point out what Hardy-Weinberg equation looks like and how we actually perform this math. For anybody who did not like high school algebra, I apologize ahead of time for this binomial equation. But for rare disease, the good news is we actually don't have to pay attention to this long equation that many people don't like to look at. And we can actually look at some much simpler math. So for rare disease, the occurrence of variants in the population is so rare that actually we can simplify this quite a lot without risking losing accuracy on the numbers. And so when I talk about sort of this general frequency of the variants going around there, the symbol for that is Q. But what that means is that it's the aggregate allele frequency of all the disease-causing variants that we included in our list. So it's literally just adding up how often we saw the variants to give us sort of a total, how often do we see disease-causing variants. We times that by 2 to get the carrier frequency, because you have one opportunity to inherit a variant from mom and one from dad. So you times it by 2, and that's your carrier frequency, or we square it, and that's the disease frequency or the genetic prevalence. I will return the numbers in conservative and relaxed. Conservative meant that we had a very strict threshold for what we called likely path and path, but we recognize that there are things that need additional evidence. We've heard about missense variants needing a little bit more information and harder to get over that line. And so sometimes we'll include these borderline and the relaxed numbers because it can give a good sense of potential variants that could be disease causing and how they would impact the numbers. So now we get to the good stuff. What did the numbers return for GAMT? Well, when we first performed this estimate in November of 2021, we had 23 variants that we included in our list, and we came back with a carrier frequency of just shy of less than 1 in 1,000, and the genetic prevalence of about 1 in 4 million. The relaxed went up a little bit, but not much. And when we returned these numbers, we immediately knew, because I try to elicit this feedback from all the groups that we work with, that these numbers were a little disappointing. They really weren't meeting the numbers that Lady and others had seen in other studies that had been presented. And I said, give us some time. You have a VSEP, you have some great networks, you really have the foundation to improve these numbers over time, and let's see what happens. At ECMG this last year, which is our American College of Medical Genetics, you guys had a booth there, which was great, and Jenny was there, and we actually had this opportunity to come together and say, the guidelines have come out, the VSEP has been doing their work in curating these variants. Let's go back and revisit the numbers, which is what we did in July of 2021. And you can see that the numbers, although the number of variants did not increase at all for relaxed and only went up 1 in conservative, some of the variants that were included in the list swapped out. And actually it resulted in the numbers going up significantly. So significantly is always a relative term. I'm not meaning that necessarily statistically, but the numbers got a lot closer to what has been seen kind of in newborn screening around the country. And the carrier frequency came closer to 1 in 850 and closer to 1 in 3 million. The relaxed went up a lot more, and these actually included some of those missense variants that Jenny talked about with the good Ravel score. And we were able to return to the VSEP and to your organization a few key variants that would be really important to follow up on that could really tell us where in between these two numbers the true genetic prevalence falls. This is a quick shout out. So like I said, my group's been working on this genetic prevalence for 6 years and partnering with patient organizations. I don't ever want to be a bottleneck to other groups being able to perform this. And so we've developed an open source tool called JUDI, which is the Genetic Prevalence Estimator, that will let any group perform this at any point in time. We're currently in beta. You can go to any of these links to try it out and give feedback. And we will be launching early next year when we have the larger version of Nomad, which will improve the overall allele frequencies and improve these scores. I also wanted to quickly show numbers of how you can take those genetic prevalence ratios that I gave and apply it to estimate the overall number of individuals. Again, this is something that we need to remind everybody. Nomad is not representative of the entire world's population, and so these are just estimates. But the numbers come out that conservatively we estimate about 113 individuals to be diagnosed with GAMT-related creatine deficiency in the US. And the reason I'm giving you the US number is because in about 10 seconds, I'm hoping to go through the numbers that have been shown previously in newborn screening, which for sake of time, just trust me, come out to in Utah, we would have estimated about 91 individuals across the US with GAMT related CD. And based on newborn screening in New York, we estimate about 125 individuals. And you can see in that conservative number, we fall right in that range. So these are all estimates, but it's showing us that this method and this approach is getting closer and closer to other sort of checks and balances. And that's very reassuring. So my final takeaways, I think I've covered everything of the global estimates and that these are just estimates, but I did want to quickly mention the future directions. We are working on a method for dominant, and X-linked, which I know is relevant for you guys for SLC 6A8, is actually going to be the third of the inheritance patterns that we are going to attempt, because X-linked diseases do have some concerns with female carriers presenting with some phenotypes that may keep them from enrolling in studies. And also across the board in X-linked, about 1/3 of variation is de novo, which means that they would not be present in population databases, and we need to figure out the modeling for that. But we are working on it, and it's coming soon. So with that, thank you very much, and thank you for letting me go over slightly.
Thank you so much, Dr. Baxter. Every time I'm putting numbers for GAMT and CTD, I pause and I think to myself about prevalence at this after listening to your talk. But it's very interesting to learn how all of this is calculated. I want to invite Dr. Goldstein also to turn on her video and to invite our moderators, Dr. Sarah Young, and from our community, Heidi Wallace, to please take over the Q&A.
Thank you. Thank you, Dr. Baxter and Goldstein. Those are wonderful. And I'm particularly excited about both of these endeavors and their application to our community, and how fortunate we are to have both of you doing the work you're doing that is raising awareness and understanding. And we're just very, very appreciative of everything that you're doing. I'll grab the first question from the Q&A box. This one's from Kim. Once variant reclassification is performed and made available publicly at ClinGen, how quickly do companies that provide SNP exome sequencing data incorporate this in the results they provide to patients?
Yeah, that's a great question. The data is publicly available, so anyone who wants to use it can. And I believe that diagnostic companies would incorporate this into their diagnostic pipelines. If they're doing like exome sequencing or whatever, they would be able to incorporate it. I don't know for each specific lab how quickly that happens, but certainly it is immediately publicly available as soon as we submit the variants to ClinVar. It does take maybe a couple of weeks for them just to go through various checks, but then it is publicly available. So,
thank you.
Yes. Thank you. And I'll follow up with a similar question for Jenny. If a patient's family is not in ClinGen or they notice it's classified incorrectly, how can they get that changed?
Right. So I guess my question would be about, sort of classified incorrectly. Classifications can change over time, as we've talked about a little bit, and that is mainly due to the amount of evidence that's available. So for example, when a variant is first seen, it may well be classified as a variant of uncertain significance because we just don't have a lot of data. But then if additional patients are identified, there may be new functional studies that come out that allow us to apply different evidence codes or apply those codes at a higher weight, then it may go up to a likely path or pathogenic. What I would suggest that is if anyone has questions about the classification of a variant on their genetic testing reports, they could talk with their genetic counselor or healthcare provider. If there continue to be questions, they could contact the lab. And as an expert panel, we're always happy to look at available evidence as well.
Thank you.
Okay, this one is for Sam. How can we learn more about the prevalence of CTD and what can parents do?
So I think it's, again, I always try to reinforce that genetic prevalence is a piece of the pie. And so efforts like newborn screening and kind of continuing creating the patient registries and patient organizations is a key part of this. I think on the genetic prevalence side of things, we have a Nomad has a new version coming out. So all of the research I've been doing over the last 6 years, I also am the operations manager for Nomad. So I have the fun thing of doing the work that's going to blow up my other work in a matter of months, but it's more the best thing. So the new version of Nomad is going to have over 800,000 individuals in it. The current version has about 140,000 individuals. That's really going to improve not only representation, but in rare disease, these rare variants are incredibly rare. And so we need a lot of people to even pick up those more rare ones, the ones that you sometimes are told are private. They're private to your family, that they're really not that often seen. The larger these populations grow, the more that we can accurately include them in the math and know how often they're floating out there. The other thing when it comes to families, please enroll in Genome Connect or get your information in there. I know Heidi's done a lot of work to partner both with ClinGen, but you have a VSEP, in Jenny, and that is a huge part of learning more about what are the variants that cause your disease, accurately curating those, and those feed into the pipeline. So the more you can just encourage labs that you work with to share data, have the sequencing when you can choose at labs that share data. This is really a method that is improved by everybody putting their cards on the table and be willing to share in the most HIPAA protected private way, but we have infrastructure for that.
So I'll just add one comment on what you said about Genome Connect and what Jenny said about the ACD's CreatineInfo registry. We do have a collaboration with ClinGen or ClinVar, where our variants are going from our registry to the Genome Connect, and they're marked as having come through there. So it's really nice for the variant curation expert panel because they recognize, oh, that is one that's in the ACD's registry, and we may be able to get some supporting information on this and reclassify it more easily. So that's how we've encouraged our families to participate in Genome Connect, is a little bit of a unique way, but very, very helpful.
Yes, incredibly.
There's a question here, which I think is actually more for Dr. Ingoglia, but I think it's just useful to mention here, and perhaps we can email it to him. The concern is how do patients get their residual enzyme activity or transporter activity measured? How do families determine rather the residual activity for their children?
Well, the way to do it is having fibroblast. And one of the issues that, there is no lab offering commercially the test. If we could get the testing going, actually there would be clinical testing available from a CLIA certified lab, which is a big advantage. I mean, the problem is they discontinue the testing for how much the testing costs. It costed much more just to maintain the test with all of the quality control that you have to do and the radioactivity. If one can remove the radioactivity, that removes a barrier of maintaining the test. And that is why it would be important.
Following up, is there any, Heidi, can you please remind me, is biobanking a goal?
Yes, we are actively asking our community to donate to Coriell. So that is what I'm hearing from this conversation is participate in the registry, participate in biobanking, and make sure that your child's mutation is investigated and understood. And yes. Okay, I think I am next. Another question for Sam. Do you expect these numbers to keep changing? It still seems like a low prevalence for GAMT.
Yes. So one of the things I will always say, and I tell everybody, we start conservative. I want to give you a strong foundation to work from. It's not always the favorite approach of everybody, but it is the one that will make sure that when you get to pharma, when you get to the people who are going to dive into these numbers, you want to start conservatively and you want to give them as tight of a range or a tight as an approach as possible, because otherwise, if you start including questionable variants, they're going to question the entire method. So I recognize that these probably are still lower than people want, but it is, I've talked to a lot of pharma, I've talked to a lot of people. We think that this is the right approach, especially when people are going to be using these numbers. I absolutely expect them to change. My hope is that when we have a bigger data set, rare disease will be better presented and we'll be able to see those numbers go up. But with statistics, you never know. Numbers can go down slightly, but I also think with the VSEP and continuing to, see all the functional work that you guys have where you can refine this big bin of variants of unknown significance that are, some of these are disease causing and they are present in the population. The more we can refine those and bring those in systematically, knowing they truly cause disease, the numbers will go up from there. I feel very confident in that. I can't make any promises because you just never know until you know, but I've talked with Jenny and Heidi about it. And I do feel like we are giving you guys very conservative numbers that will be able to go up over time. How much they change is, I'll know a lot more in January of 2024 when we redo these numbers again for the third time with new data.
Excuse me. I do see a couple of related, the variant, how is it possible to determine if it's partially broken or completely broken? Well, that would be to have a skin biopsy and then donate that to Coriell. And then the research labs would then take over and measure the activity. Excuse me. So to Sam, how does the prevalence of private mutations affect the prevalence estimates?
So I think that was kind of going into the what I was talking about before with having larger and larger population databases, we can find these more rare variants. And so typically that's what people mean by private mutations. It's something that kind of is said in the clinic as a way of saying we've really only seen it in your family, but often with rare disease, that doesn't mean your family are the only people who have these variants. It's just that the people out there are carriers. They're so few and far between. They really have only met up with so many other people and presenting in clinic and somebody with the diagnosis. So we think that as these population databases grow, and the great news is for population databases, a few years ago, it was harder and harder to get people to enroll. It's now something that people want to put as part of their grant applications. They want to say we are sharing the data. So I think we're going to really start seeing exponential growth in population databases. And my hope is that with this increased power, really rare disease families are going to be the ones that benefit from it the most.
Okay. I have another question. This is our last. It sounds like at this time, this data is most robust for US-based prevalence. Can you speak to enrichment of certain mutations in other countries and or certain populations, ethnicities? Is there a chance that there are more GAMT patients in other countries?
So yes, I did return the number talking about US, but we actually do look at 8 different genetic populations, so genetic ancestry groups that are present in NOMAD, and we returned the numbers to the organizations with those as well. Again, the lack of representation from every country and every genetic ancestry group is always something that we consider. We don't know what is not represented, but we are able to look at if there are certain trends in Europeans or African Southeast Asian, and we're able to kind of look at those. Again, we want representation to improve, so we can continue to include that. The focus on the US was really just because I usually give this presentation in an hour and a half, and I have to kind of pare down the numbers quickly. So we focused on the US for this population. Also because a lot of US-based patient organizations are going to US-based pharmaceutical companies, and they want to hear about US numbers. So that's really just because of the slant of who I'm presenting to, but we are able to give representation from other ancestry groups as well.
Great.
Thank you. So we have 1 minute left, and I have Dr. Longo on the hook there. So I figured I'd swoop in with a question. My question was related to the functional testing in fibroblasts, and I know, Dr. Longo, you mentioned that donating the fibroblasts and would be the first part of the puzzle, and the second part of it would be for functional testing to be conducted on these. But at this time, is the functional test is that a clinically validated test in order to be able to inform insights relating to supplementation? Or is this type of finding still something that we can look to in a paper that might still guide clinical insights for physicians, but it's a little different from saying that here's a codable clinical diagnostic test? Dr. Longo. Sarah, I don't know if I'm not able to hear you if you are.
Oh, yes. Oh, sorry. How Dr. Longo responded earlier was there are, I mean, first of all, you have to go through a validation process to bring on clinically, but there's also has to be a demand for the clinical test. And so I think currently all this testing that's being done in the various labs is all under research. Whether or not families are able to recover that information from the researchers, I'm not entirely sure. It depends on the processes that are set in place. But these are all very good questions, and it's very helpful, I think, for clinical labs to understand that there is an interest from families to provide this type of testing. And I do have one more question for Sam. With all the newborn screening pilot studies using exome and genome sequencing, is that providing any useful data for these disorders?
So right now, our population databases don't necessarily include that, but I think over time, again, any of this aggregated information, one of the hopes long-term for Nomad is that we'll actually set up federated data sharing, which means that there'll be a hub that people can send the aggregate allele frequencies. So this will allow protected populations or sensitive databases that don't want to share the entirety of their raw data, and rightfully so, to be able to use our methods that we've developed and put out there, calculate their own allele frequencies, and then because they all use the same pipeline, you're able to add it at the top. So I think that it's coming. They're not all in there together right now, but I think that again, the tides are really turning where everybody is seeing the benefit of this, and the power of it coming together at the end, I think, is going to be really cool to see.
Yes, I remember one talk at ACMG where I'm pretty sure they identified some CCD patients in the newborn screening programs.
Yeah, absolutely. I think there's gonna, like I said, I don't want to make any promises, but I feel confident and hopeful that the numbers are going to continue to improve the more representation we have.
Thank you. Thanks, everyone. With that, we come to the close of this session. We're going to take. So in this next session, while the poll is going on, I'll just talk to you about the next session. This next session is going to feature updates from the CreatineInfo registry. And just looking at this poll here, it's very timely to understand
how many people are already on the registry, know about the registry, and how many, maybe there are people in the audience who are just learning about the registry now and still plan to join it but have not yet. And I hope that this next session will shed some light on how this data is important for our community, and I hope it will motivate a lot more people to take that next step and join the registry. Okay, we're at time now. So Faith, can we end this poll and share results? There we go. Okay, that's really good to know that 83% folks are already on there. A great show of participation from the community. And for those of you on the fence, please consider taking that next step as soon as possible. With that, we'll get started with the next session, and I would like to call on Sung to please invite our speakers.
Sure, thank you, Sangeeta. Our next speaker is Dr. Sylvia Stockler. She is a professor in the Department of Pediatrics at University of British Columbia and a distinguished clinical biochemical geneticist in the Division of Biochemical Genetics at BC Children's Hospital in Vancouver, Canada. With her background in biochemical genetics and pediatric neurology, her main interest resides in finding treatments for children with neurometabolic conditions and rare diseases, including CCDS. With a passion for improving the lives of young patients and their families, Dr. Stockler has devoted her career to advancing our understanding of rare metabolic disorders. She uses an integrated approach based on clinical, biomedical, biochemical, molecular, and molecular genetic and imaging analysis. Her insights have shed light on the important biochemical pathways that underlie these conditions. She is the PI of several investigator-initiated and industry-sponsored clinical trials for rare diseases treatments, and is especially interested in practice and observation-informed evidence in rare diseases. She also serves on the ACD Scientific Medical Advisory Board. The title of her talk is Creating a Core Outcome Set, a collaboration between CTD and GAMT deficiency parents and physicians. This work centers on developing a core set of critical outcomes in collaboration with patient groups. So Dr. Stockler, I'll hand it over to you.
Well, thank you for the extensive introduction. I'm going to share my screen.
Dr. Stockler, I think we may not be picking up your mic properly.
And so you can hear me now?
Very good, thank you.
Yeah. So, yeah, as already indicated, I'm going to talk about a collaboration between parents of children with CTD and GAMT deficiency and experts like physicians and scientists who are who do have some expertise in creatine deficiencies in a project that's about creating a core outcome set for creatine transport for creatine deficiencies. I will talk about challenges with clinical trials for rare diseases, and I will also explain what are core outcome sets and what are the benefits and how do they relate to the challenges with clinical trials. And how does that apply to creatine transporter and GAMT deficiency? I will explain the methods and first results of our core outcome set development project, and of course then, end with future steps and with acknowledgements for all those people who have participated so far. So first of all, what are challenges with clinical trials? As you know, clinical trials assess if an intervention is safe and effective by comparing its impact on various endpoints. In clinical trials, we speak about endpoints or outcomes. So that's kind of equally used. And I will use it equally also in the course of this presentation. Historically, there has been significant variability in how outcomes are defined and measured in clinical trials. And this has posed lots of challenges in interpretation and real-world application for their results. Additionally, outcomes included in research trials have not always been those that patients themselves regard as important or relevant for their quality of life. And the solution, or one of the solutions for these challenges, is to create an agreed upon standardized set of outcomes, which is called core outcome set for various diseases like creatine deficiency syndromes. So again, what are core outcome sets? Core outcome set means what is an outcome? Outcome is an observable or measurable symptom or sign of a disease. It could be clinical symptom or it could be a lab test. In clinical trials, outcomes are often named as endpoints, as said before. Symptom, signs, outcome, and endpoints are closely related. So we're talking about the same thing throughout the whole presentation. And what is a core outcome set, and why is a core outcome set used at all? A core outcome set is a small set of disease-specific outcomes deemed important by stakeholders, parents, physicians, and they are used as a minimum set of endpoints in every clinical trial. The benefit of such a minimum set of endpoints is that it allows comparison across different studies. So if there are two trials evaluating the same drug, it will allow a comparison across both clinical trials. And therefore, it will also allow a more efficient use of resources and minimize the risk of bias. Core outcome set can also be more patient-centered if they include patient-important outcomes, and overall will accelerate drug development and prevents measurement of inappropriate outcomes. So regarding to calls for creatine transport and GAMT deficiency, one has to say that there's currently no or just suboptimal treatment for creatine transporter and GAMT deficiency. And there are also many research studies ongoing, as we already have seen in the presentations today, to develop new drugs and of course to test the effectiveness of these new drugs in the patients. The aim of our study is to develop a core outcome set, which will include 8 to 10 outcomes for creatine transporter and GAMT deficiency to ensure consistent evaluation of therapies using clinically significant outcomes for patients and caregivers. And the methodology according to which we are developing core outcome set is informed by the COMET initiative, which is an international consortium which is working hard on guidelines, how methodologically correctly one can develop such core outcome sets for various diseases. And it is funded by BCORI, Patient-Centered Outcome Research Institute, through a project that's called PARENTS. So what are our timelines and stages? As you can see, we started the project last year with stage 1, and we hope to have completed stage 3 in November 2023. And in the following few slides, I will explain to you what we have done in stage 1, stage 2, and what we're going to do in stage 3. So stage 1 is about evidence review and collecting information about outcomes, clinical symptoms, signs from many other resources. So one of the resources is a formal literature review, which analyzes all the literature for symptoms and signs ever reported for these two diseases. Another very important resource is the patient and caregiver input, because this will provide the core outcome set with outcomes that are important for the patients themselves. We have done this in person and virtual focus groups with parents from children with these two disorders. And the third resource of information we are using is, of course, the ACT CreatineInfo registry. Once you have taken all the information, of course, there's a lot. You have to prune it down and to refine a list of possible outcomes. For this, would need definitely the input of research teams and healthcare experts. We actually, taking all these resources and doing all the pruning, we came up with a finalized list of 85 outcomes for the Delphi survey. But please keep in mind that this is much too much for a core outcome, because no clinical trial can assess 85 outcomes. This would be just too laborious. And as you might remember from the previous slide, we aim at about 10 outcomes. So I'm also going to explain to you how we're going to, reduce those 85 to 10. So, but just before that, stage 1's evidence review, you can see here, we did the literature review. It was a formal literature review, which is called scoping review, where we screened all the titles and abstracts about these conditions in the various databases. We did full text screening, extraction of articles, and after all of that, we kind of just noted everything, every outcome, every clinical presentation that was reported in those articles, and we came up with, as you can see, with less than 800 outcomes, which is of course a lot. As already mentioned, we did patient caregiver focus groups in person, which was last year at the ACD symposium in Utah, and also virtually for those on Zoom, for those who were not able to attend. And finally, we at the creatine registry, where we extracted 24 additional outcomes. Please note the number of patient caregiver focus group outcomes, which is quite a lot. So overall, we came up with a total number of 1,148 outcomes. So outcome pruning also takes a couple of steps: removing duplicates, evaluating uniqueness of outcomes. Sometimes one outcome has two different names. For example, epilepsy could also be described in the literature as seizures, but it's the same outcome. So you have to make sure that all the outcomes that you find one unique term for it, which of course helps to reduce the numbers. And you can see after removing duplicates, we could reduce the numbers from 122 to 79. And all this was, of course, supported by feedback from healthcare professionals and also parents and caregivers. So the final number of outcomes was 85, which is still too much. And therefore, in stage 2, we used a method that's called Delphi Survey, where we are asking families, healthcare providers, and researchers to rate the outcomes according to their opinion, how important they are, and of course to develop also a consensus. This is a stage that is still ongoing. So what is a Delphi survey? A Delphi survey is designated to gather opinions from multiple people and to reach agreement, a so-called consensus. For our project, we developed 3 rounds of Delphi surveys. they were administered on a REDCap database electronically, and participants consisted both patients of children with creatine transporter and GAMT deficiency / their caregivers and experts. Outcomes were rated in each Delphi round, and those who were not deemed not important enough in one round were eliminated at the next round. So this is an example, and many of you probably know these examples because so many of you thankfully have participated in the Delphi survey. So here, for example, it asks for a specific outcome, which is developmental delay. How important do you think is research is to measure developmental delay in research study? And then the participant could rate from 1 to 9, from limited important to most important, what is their opinion about it. If they had a specific thought about it, if they had specific questions, there was also a possibility to write that down. So just briefly, after the first round, we defined that those outcomes would be moved to round 2 if 70% of the respondents of a group, let's say GAMT and or creatine transporter deficiency, rated the outcome 3 or higher. So we had quite a low threshold, right? Because 3 is just not really very important, but it's better than 1. So this kind of helped us, to come down from 85 to 81 outcomes. So you can see there wasn't a big difference. But after round 2, with those 81 outcomes, we defined that outcomes would be moved to round 3 if 70% of respondents would rate the outcome 7 or higher. So the threshold became much more higher, just to be more focused on the importance of the outcomes. So for round 3, we will have 50 outcomes. So we have already pruned them down significantly. This round 3 will launch in September 2023, and both participants in rounds 1 and or round 2 will be eligible to participate, and they all will receive a personalized survey link. In addition to the rating of each outcome, in this round 3, the participants at the end of the survey will be asked to rank their 10 top important outcomes, to rank them so that they only have 10 outcomes left. And this will help us to further prioritize and to come closer to our goal of 10 outcomes. Here you can see the number of participants. The orange bars are the round 1, and the blue bars are round 2. And here you can see, what are the groups. So we had patients directly participating, not many, but we had a lot of caregivers from patients with CTD and GAMT. Of course, CTD was much more prevalent because it's a more frequent disorder. But we had also a decent number of GAMT caregivers participating, and you can see that most of them participated both in round 1 and in round 2. And then we also had a good number of healthcare professionals and scientists and therapists who participated in both rounds. The COMET guidelines also advise to add health policy advisors and decision makers to these rounds, but we didn't have any, and it's not really obligatory. So after that, round 3, we will end stage 2 of our project, and stage 3 will actually be a consensus workshop. So all the outcomes that will be left after round 3 will be lumped together, and in an in-person consensus workshop together with parents and experts, we will try to get it pruned down to our 10 outcomes. And this will be in November, so the stage 3 will hopefully be concluded in November 2023. And so we have already planned the consensus workshop in November in Salt Lake City, and parents, healthcare providers have already been invited or are being invited, and the COAS research team, of course, will participate as well. There we will review and discuss the results of all 3 Delphi survey rounds and finalize the COAS to 8 to 10 outcomes for both disorders. So at the end, those COAS will support the following domains. So we will not only have we will try to not only have a core outcome set of just laboratory values or of just magnetic resonance imaging values, but we will distribute them among life impact. So what are the outcomes that help to improve quality of life for patients and families, for daily living, pain, sleep, adherence to treatment are some of those outcomes that are part of life impact. Growth and development contains cognitive function, developmental delay, intellectual disability, but also somatic growth and somatic functions, communication, and overall functioning. Pathophysiological manifestation are those which we as doctors like to measure the most because they are easy to interpret. It's cardiac. We have heard about, cardiac manifestations in creatine transporter deficiency. It's something like neurologic outcomes like seizures or epilepsy, but also all the blood tests, guanidinoacetate for GAMT deficiency, for example, MRS results, brain creatine studies, or MRI studies of brain imaging. And fourth, but not least, resource use, what would be outcomes that would help to have the access to therapies and treatments.
Dr. Stuckler, you have 1 more minute.
Yes, I'm already done. Development and validation of tools to measure the outcomes in the final course, and we will also try to publish those findings and share them with the community, and by that also motivate groups of people with other conditions to do the same thing. Finally, very important, acknowledgements and funding. So the research team is Heidi Wallis, who is our lead. I mean, she tries really to keep us on track with everything. Audrey, who is going to give a talk about further implications of those outcomes in the next few minutes. Sarah and Emily, who have been fantastic in doing the Red Cup surveys, in doing the analysis, and I'm most grateful to them that they prepared that presentation for me. So I had a good time. Celeste and Beth, who are patient representatives who have had a lot of very valuable input. And we have advisors, Beth Potter from the University of Ottawa, who is a rare diseases methodologist who has been developing core outcome sets for other conditions, and I had the privilege to be part of that as well. And we have Maureen, who is a patient advisor, and Alison, who is research coordinator for Beth Potter. So thank you very much, everybody, for contributing, patients, families, and doctors. And again, thank you for funding from PCORI.
Thank you. Thank you for your explanation on what is being done on core outcome set. If you have filled out Delphi survey in the past with ACD, hope her talk has answered some of your questions. If you have unanswered questions, please add them to the Q&A box. She'll be able to answer at the end of the session. Up next is our very own Emily Reinhardt. She has also, she has almost 15 years of research experience with particular emphasis in preclinical and translational neuroscience research. Through her love of science, she's passionate about using data to improve the lives of people within her community. Emily is a graduate of Kansas State University, earning both her BS and MS in psychological sciences with a focus in behavioral neuroscience. Emily currently serves as ACD's patient registry coordinator, which allows her to put science into action by collaborating directly with patients, caregivers, and researchers. The title of her talk is CreatineInfo Patient Registry Data in Action. Emily, please take it away.
Thank you so much, Sang. Let me start my slide. Okay. Hello, everybody. Good afternoon, good morning, depending where you are. As Sang said, I'm Emily Reinhardt. I'm with the ACD. I have the privilege of serving as the patient registry coordinator. And today I'm going to be talking to you about our patient registry, providing a little bit of an update and how we are utilizing your data. So just briefly, today I will give a very high-level overview of the registry and where we stand. The bulk of my time, I'll be discussing how our data in action and our strategy there. And you'll see the banner down at the bottom will pop up, and it will I'll be highlighting specific areas, tangible areas that we are able to use your data, use data from the registry in real ways. And then finally, talk a little bit about the future of the registry and where we're headed. So currently we have 187 participants who are enrolled in our CreatineInfo registry. Of those, 69% are CCD, 30% have GAMT deficiency, and 1% have a GAT deficiency. On the right there, you will see the donut chart that has the age breakdown. And you'll see the majority of our participants, at 57%, are age 6 to 17. So that's the breakdown of our community right now. Our community is primarily males, making up 81%, for females 19%. And then finally, we are an international organization. Our registry is global. We welcome people from all around the globe. Right now, 65% of our registry participants come from North America, followed by Europe. But this is something that we are working towards and trying to have a broader global footprint. And I'll talk a little bit more at the end of my talk about how we're working towards that. So just briefly, I want to highlight, especially those who are new to our community, new family members, new clinicians and researchers, just broadly, kind of three key aims of our CreatineInfo patient registry natural history study. One, we want to demonstrate the natural history of the three creatine deficiencies. We want to facilitate the development of treatments and therapies. And finally, we hope to improve diagnostic and screening processes. So I'm going to share just some highlighting some interesting findings that have come from the registry in each of these three aims. I'm going to start very briefly talking about how we're meeting this first aim, looking at the natural history study. And so actually we're doing that through our diagnosis survey. So we currently have 133 responses to this survey. We've seen an increase of 16% from this time last year. And really the goal of this survey is to document patients' experiences prior to and while they are receiving a diagnosis. And this really reflects the first step in chronicling the natural history of creatine deficiencies. And so I just wanted to pull out an interesting finding here. We asked survey participants, which symptoms did the participant or the CCDS patient exhibit prior to receiving their diagnosis? And I think you'll see here at 81%, global developmental delay was overwhelmingly reported by 81% of participants, followed by speech delay at 74%. But I really think that the take home of this graph here is it just demonstrates the diversity of symptoms that our patient community experiences broadly, but also within a disorder. So even within CCD or within GAMT deficiency, we see this broad range of symptoms. And so this data came from the diagnosis survey. We were lucky enough to be able to provide this data in the voice of the patient report will be submitted to the FDA. And it will serve as an official record of the patient experience to aid in CCDS treatment approval. And Sangeeta mentioned this at the beginning of the session this week or today, that the voice of the patient is supplements the CFDD meeting we held in January, where we heard from patients and caregivers just kind of documenting the experience. And so we were able to supplement that report with data from our registry, including data that came from our diagnosis survey. So the second aim that I'll be focusing on is facilitating development of treatments and therapies. And we're doing this in a couple of different ways. First is through the Patient Meaningful Outcomes Survey, or the PMO survey, as we like to refer to it as. We currently have 75 participants who have participated in this survey. That's an increase of 17% from last year. And overall, the goal of this survey is really to learn from CCDS patients and caregivers about what aspects of the disorder are most important to them and really what they would like to see change with the treatment. And so in this survey, we asked participants to rate in importance 24 outcomes that were split up into 5 separate categories. So we had outcomes that focused on cognitive, neurological and sensory, motor, feeding and growth, and behavioral and social. Again, I just want to highlight an interesting finding here. We asked participants to rate pick their top 5, the 5 that are most important to them. And overwhelmingly, these are the outcomes that were selected most often by participants. Number 1 being speech and communication, being selected by 72% of participants, followed by learning difficulties, seizures, reduced independence, social interaction difficulties, and focus and attention. And as Dr. Sackler just spoke about, we were able to take data that was provided in this survey, and it was included in the evidence review for the development of the core outcome set for CCD and GAMT deficiency. Again, illustrating that the data that's coming from the registry is being used in purposeful and meaningful ways. So another survey that we have in our registry is the oral medication survey that was discussed previously. This was done in partnership with Ultragenyx. We launched this survey this year in March of 2023, and we already have 44 responses. And really the goal of this survey was to assess the oral medication preferences of CCDS patients, aid in the development of oral treatments. So again, just highlighting a couple of interesting findings. We asked participants what attributes of the medication are most important to them. And overwhelmingly, patients or survey participants said that a manageable volume and quantity of dose, as well as a taste that is neutral or desirable, is so important to them. And so this is really, really an interesting finding and kind of really going to be beneficial for Ultragenyx as well as future treatments and developments to understand what are patient preferences here. And another interesting finding that the team we keep talking about is we just asked participants what flavors do CCDS patients prefer. And overwhelmingly, they prefer unflavored. And so again, just highlighting why it's so important to ask our community, because I will say I was not expecting this outcome, so this was definitely a surprise. So as I said, we partnered with Ultragenyx on this, and we provided them a de-identified aggregate data report that will inform their plans for the development of their oral formulation for UX 068, which is a possible therapy for CCD. So again, taking data that's coming from the registry and using it in an actionable sort of way. And then finally, I just want to say the third aim here is we want to work to improve diagnostic and screening practices. And we're doing that through the collection of patient genetic and medical reports that we've discussed quite a bit, that Jenny Goldstein has highlighted. We currently have 44 unique variants within that participants have provided within the registry. And really the goal of collecting this data is really to better understand the biological metrics of protein deficiencies and how they're related to other symptoms. And so this is less of data and more of a plug, which again, we've highlighted already, but when we really want you to think about, okay, what test results first made your doctor to suspect that that the patient or that your child had a creatine deficiency. And that's going to be things like a genetic report, the urine and plasma results. So anything that's measuring creatine and guanidino acetate. And if you had an MRS completed, we would be really, really interested in collecting that data. And so you can upload those items to the registry, upload those things to the medical notes. And as we said, ACD and the registry, we've partnered with ClinGen and the CCDS variant curation expert panel to review de-identified genetic and health data, supporting the reclassification of new genetic variants and really improving that diagnostic accuracy. So just to wrap up, I've shared a lot of things. We've got a lot of stuff going on in the patient registry. So what's next? Where are we headed? This is hot off the presses, literally launched today. We have a new survey. The Cardiovascular Health Survey is live. It's on the registry. It's ready for you to complete. We've done this in partnership with Dr. Mark Levin, who's the pediatric cardiologist at the NIH. And really the goal of this is to advance our understanding of cardiovascular issues among CCDS patients. As we heard earlier today from Dr. Berger, we know that there is a relationship between creatine deficiencies and cardiovascular health, and we need to learn more. And so we want to hear from you. So I really encourage you to please participate and take this cardiovascular health survey. And then what else is coming up? The next thing is seizure surveys coming later this year. We're really, really excited about this. We've partnered with Dr. Andrews. This is going to be a big survey, but it's really important for our community. We also next year will be migrating. We are currently on our registry is hosted on NORD's I Am Rare registry platform. They have created a new platform. So we'll be migrating there, and really excited because we will be able to offer our surveys in both Spanish and French, with the options to translate into other non-English languages. And so again, just really working to ensure that our registry reaches as many people, anybody in our community as possible, and not let language be a barrier. And then finally, we want to publish our first registry paper. But to do that, we really need to meet a goal of 200 participants. So again, another call is we need 13 new participants to join the registry. So if you haven't joined the registry yet, please do so. You can visit creatineinfo.imrare.org to join. Email me with any questions that you might have. We would love to have you involved. And then just finally, how can you participate? Yep, son, I see my time. If you're a patient or caregiver, again, join the registry. If you've already joined, log back in, make sure you're up to date on your surveys, including the cardiovascular health survey. And then if you've got lab reports and standard reports, upload those. And then finally, if you're a clinician or a researcher, we encourage you to invite your patients and join the registry and collaborate with us. We work with clinicians and researchers all of the time. That's a key part of our development process. So if you'd like to connect with us, email me at registry@CreatineInfo.org.
Thank you, Emily, for the
Thanks,
valuable
everybody.
information. If you're a caregiver and have not registered your loved ones, please register today. And our next speaker is Dr. Audrey Thurm. She received her training at DePaul University and Boston Children's Hospital, Harvard Medical School, and conducted a postdoc fellowship at Johns Hopkins School of Medicine. She has been an NIMH since 2002, serving in the extramural program until 2006 as chief of both the Autism and Social Behavior program and the Compulsive Repetitive Behaviors program. In 2006, she moved to the intramural program to help launch the autism research program, and she has expertise in longitudinal studies and an interest in markers of the early diagnosis of autism, as well as genetic conditions associated with neurodevelopmental disorders. The title of her talk is Clinical Trial Neurodevelopmental Outcome Measurement Lessons Learned. And I'll hand it over to you, Dr. Thurm.
Great, thank you so much. Can you hear me?
Yes.
Great. So I'm aware that we're all behind and I'm the last talk to get us to the panel and to dinner, so for those on the East Coast. So I am going to try to make up time here and be quick, but I'm wearing 3 hats, I think, in this talk today. And let me just see if my slides will progress. Yes, I have no disclosures. My 3 hats here are number 1, as a researcher on the Vigilan natural history study that you heard about, and I'm going to kind of go a little bit underneath that, what you heard about the results today. And then, as you just heard, as a member of the team on this PCORI grant, which we're looking for core outcome sets. Those things come together with what we heard first thing this morning from MILA'S mom about our need to get clinical trial ready, and especially because we may be sooner than we think with CTD, and what we need to do that with getting core outcome sets, and then bringing them to measures that we can actually use in trials, and doing this not just for each individual disorder, but as a whole, because we need, even each individual disorder needs to do this because the FDA cannot handle what's coming to it, and it will be easier for each condition if it makes sense to them in a harmonized way across the conditions, and they're not all unique. We know that will help us. So again, we heard about the Vigilan trial, where for CTD, even though that's a small natural history style of 50 study of 50 patients, but it's large given the population of CTD. And I'm just going to focus here on the goal of contributing to the development of therapeutic intervention programs by identifying those study endpoints relevant to CTD. So we were doing that in this study, and we're doing this in the PCORI with the outcome sets, and we keep hearing over and over again certain variables that Dr. Stockler just pointed out. And I'm going to focus on the growth and development one, which is a pretty important one that cuts across, that we need to really think about. And this is something that the FDA puts out about how you get from understanding the disease, which we're still pretty new at with CTD, all the way to getting outcome measures that actually work for approval for FDA studies, including gene therapy studies and even precision medicine. And so focusing in here on identifying those concepts of interest, which, remember, normally are cancer survival rate. That's easy. How you feel, even if you're depressed or if you have pain, reducing those symptoms, which patients can often do themselves, and then functioning. And so I'm going to focus on the functioning because we actually are best able to do that with CTD. And that's a lot of what the parents are saying are the interests feels is pretty hard, actually, in terms of pain, in terms of patients not being able to report themselves because of communication. And I just want to focus on that with, again, you saw earlier some findings from Vigilan, and this is actually what we had to work with when we started, before we started Vigilan 7 years ago, what we had out there of data about CTD, and we only had retrospective data from reports, which is similar to what goes into the registry. So another plug from the registry. And that showed us, we knew going in that there was quite a bit of language speech impairment, that a lot of kids and even adults are not speaking fluently. And we also knew that there was a very significant degree of intellectual disability. But what we knew from these retrospective reports, we didn't know what we were going to see in front of us, because we know that there are going to be cohort effects as we go through, and that as more kids get diagnosed, which we hope continues to happen. So we really didn't know what we were going in on. And so what I want to emphasize here is that us as researchers, we have to make guesses sometimes. And so we did that. We had to make guesses with what the best measures would be. And you heard earlier that we do not have the best measures specifically for the cognitive language sort of IQ realm for this population. What we heard was that there is delay and a majority of the population doesn't speak fluently, that we saw that there was universal intellectual disability from those retrospective reports. We didn't know if that was really going to continue to be the case. It looked like kids were getting worse with intellectual disability as they get older. We heard a little bit about why that happens is because our tests get worse and worse when they're standardized across ages when you get older. And then we actually did see from that original report that that a lot of the intellectual disability was more in the moderate to profound range, where our tests work even worse. And so what I'm going to focus here is diagnosing intellectual disability, which is important not just because it seems to be very important for the functioning outcome, where the FDA does want measures directly from the child. They really do not like the proxy reports that we get for adaptive behavior. They really want what they call direct functioning, or outcomes that are directly from the patient. And so they do want to have this no matter what, even when we say that our measures aren't good. So I just want to emphasize a little bit here about how not good they are, and what a lot of parents may have experienced when they've gotten this testing either from schools or in our studies, is that we have to find the right construct. So we're talking about development, and we know that for adults, we're talking about reading, complex problem solving, and things like that. For younger children, we're talking about fine motor skills, expressive language, using words, and these are different constructs. So we're actually measuring things differently. And just in case it isn't clear, we don't have tests that span the entire age group. That doesn't exist really very well in cognitive IQ testing. We do use different tests according to different ages because there are different constructs of interest. So what we had to do here is decide what test to use, which we really weren't sure of based on what we knew, where we can see that there are floor effects. None of these tests actually go down into the profound and severe range, which is under 40. These tests literally do not have scores for children under 40 with IQs under 40, which is what we're seeing here. And so we had to use a hierarchical approach, try one test, and then if not, use another. This is not ideal for the children, for the families, and for us giving it, because we don't want to have to prolong and make it more difficult. I just wanted to give the schema here that shows some of what's required for the early tests, the developmental tests, that they really do require trying to get a child to match, trying to have them see if they understand just even one word. But then it bumps up with the IQ test to being able to understand complex directions and really do very complex kinds of tasks. So this just gives you more explanation about why we had to use developmental tests, which are called out of age range, because they're not actually normed for the ages that we were using it for in older children. And this is what it looks like when you do that. You see, even though mental age or actual skills improve, which is what you saw that Dr. Miller showed in adaptive functioning, we see that in our cognitive tests too, that kids are learning, and we do see skills improving over time, but we see those scores, no matter how we calculate them, going down over time. And that is because our tests are not normed extensively and for these older ages. So we have a problem in what we can present to the FDA, and the data that we got from the natural history study was so helpful in allowing us to figure out that we need to use tests now that do not just have these standard scores, that also have these ability scores that allow us to express performance in absolute terms, not just standardized with children of the same age or adults. And that we can use these scores and show what the standard scores are and show what they are in ability scores to compare, especially over time. And I just want to emphasize here that what testing goes on in schools and what testing goes on diagnostically for autism and intellectual disability evaluations are usually snapshots, and they're not designed to test children over time. What we have to do for FDA approved studies, even if they're no matter what they, even individualized, personalized, or group studies where we have control groups, is use tests over time. And that's something that we just haven't been prepared for as this field has been developing. And so we need to develop tests that are appropriate across the lifespan in order to be able to follow kids and have scores, but we also need to make sure that they are being developed with this age, with this kinds of groups with more severe and profound intellectual disability in mind as we're developing them, so that we can capture the range and variability so we can see the improvements. So that's where I'm going to conclude, is to say that the data from the natural history study is so critical in telling us, unfortunately, sometimes what tests don't work, so that we can really have the data to show grant agencies and researchers what we need to do, what tests we need to do to develop, because the data from these natural history studies really are the external control data, because we're going to have to start with open label trials no matter what. For safety, and we're going to have data from natural history studies and the registry to some extent to allow us to compare. We need to recognize our limitations in these existing measures so that we can actually have justification for developing new ones. And then we really need to think, what are the best metrics that we use in these trials? Because we want to get clinical trial ready before the drug is ready. So we're in a race for that. And so these are just some of the things that we need to do to get there, at least in this growth and development realm. And I just want to thank everybody, especially all the families here, for sticking with us and hearing about these more difficult topics at the end, and to all of our collaborators. Thanks so much.
Thank you so much, Dr. Thurm. As a caregiver, especially with a child who was diagnosed with CTD fairly recently, I understand the survey and questionnaire fatigues you may feel. And there are so many questionnaires that I can fill out as well. But hopefully you are motivated again based on today's session to fill out any upcoming registries and surveys. Because these will really help to develop outcome measures that can be used in clinical trials and to develop treatments for our kiddos. So your participation is extremely valuable. With that, I'll invite the speakers back for Q&A, and we'll turn it over to the moderators, Dr. Longo and James Mon.
Hi there. So I had a few questions to start with that have come in from caregivers. Starting with Dr. Stockler, a question around, obviously, defining the core outcome set. What does the process then look like to turning that, I guess, into measures in the context of preparing for clinical trials? And I guess an extension to that, do you see that it would be, I guess, a combination of physical examinations coupled with kind of biological markers for things, with an MRS of the brain, for example, be expected to be one of those markers?
Yeah, thank you for that question. I think I'll start with the last question. I mean, most of the time, these core outcome sets, as we know from other diseases, will contain outcomes in various domains. So definitely one domain will be biomarkers, blood tests and MRIs. So that will be definitely one outcome, but there will be also outcomes that pertain more to functioning, as Dr. Thurm has pointed out, and to quality of life. So there will be, we will try to have the core outcome set covering various domains, not only one, not only biomarkers. So that's the one question, and the other question is more difficult to answer and bridges definitely to what Dr. Thurm has pointed out, that once we have identified those core outcomes, we have to think very hard about how are they going, how can we measure them? So, I mean, are they measurable at all? So if there are outcomes that have turned out to be important for caregivers or for physicians that are not well measurable, we have to, to bridge to other outcomes that would include that one outcome to have it measurable. And of course, the crucial question will be, how good are the tools that we have already available, for example, for functional cognitive outcomes? And there's still a lot to do, I think, as we saw from Audrey's presentation.
Great. Dr. Longo, I don't know whether you wanted to ask any questions?
No, sorry, I had to go to see a patient, and that's why I disappeared. So I do have a life, unfortunately.
Okay, I'll carry on with some questions from caregivers then. So a practical question for Emily. Someone's asked about the medical notes section of the registry. I know you mentioned what were kind of the key documents and what kind of things you're looking for in there. It might be worth giving a bit more background and maybe reinforcing, I guess the huge amount of reports spanning many years that parents have got. What should we be uploading? And also, I guess, as new exams happen, so my son, for example, has a heart scan every year. Can we go back and kind of continually update the profile?
Yeah, so this is a really great question. We recognize that patients have a ton of data. And that medical note survey is really just to serve as a placeholder. But I recognize that it can be very, very overwhelming. So I would start with just what was the data that was first used when you received a diagnosis. So if you have a genetic report, get that uploaded. Did you have blood tests done? Get that uploaded. Did you have an MRI or an MRS? Get that uploaded. If you're finding it overwhelming, start with the initial diagnostic process. If you've already done that, don't feel like you have to go back, years and years and years, and that I recognize is overwhelming. Just start a habit now. So maybe, like you said, there's a new cardio test result that just happened and happens yearly. You come home from the doctor's appointment, upload that. You can just start creating some new habits now. I don't know if that's helpful. You can definitely go back and add things as you get them. So it's definitely updatable. And shoot me an email if you need some more direction on that. I'm happy to help.
Great. Okay. And then moving on to Dr. Thurm, we had a question talking about, obviously with a broad range of, I guess, severity of symptoms. Is there a consideration in measuring a patient that might be more severely affected versus someone who's probably more moderately affected? And would there be a different set of measures personalized to individual patients?
Yes, and that is, at least for cognition, that is what has had to happen so far in natural history studies. That gets difficult. The FDA does not like that, which is why we actually advocate for adaptive behavior, what you saw Dr. Miller present on, which is really how the child's functioning, which actually the measure does span all age ranges and function levels for that. That is a much better construct, we think, and it's functioning. So it's a good outcome that avoids those problems. The problem with that is that right now, our measure for getting that is a proxy measure, which is from parents. And the FDA does not like to hear from anybody but the patient themselves. They don't quite get the issues that we have in this population where communication doesn't allow for that. So we've got one set of problems on one hand and the other set of problems on the other hand. But we're grappling with that. And I think having both be outcomes is what's going to be most helpful.
Brilliant, thanks. And a follow-up to that, what role, in your opinion, would a measure of creatine in the brain using an MRS as a primary endpoint in a clinical trial play?
Right. The FDA is likely not going to take our functioning variables as the primary outcome. They're likely to take a biomarker. I'm not going to say which one it is, but I think what we've seen in other conditions is that some of these brain biomarkers, if we can actually show correlation between the two, that is very powerful. And so I think the more data we can get on MRS and then trying to look at those correlations, that I think will be actually, very powerful for everybody.
Great, thanks. And a question for the group, really. How do you deal with the different weightings that different parents might put on certain symptoms or certain benefits and outcomes? So, for example, seizures, which I think around 2/3 of kids with CTD would have, whereas a third wouldn't. For those 2/3, it would be super important. For the other third, less so. So, how would something like that be kind of measured?
Yeah, I mean, regarding importance, this is what we try to do with the Delphi surveys. Of course, and of course, I mean, Delphi survey doesn't represent every single participant's priority, because if there are 70 participants and 60 participants rate, an outcome very high and 10 rated very low, I mean, it will be more a matter of statistics, what's the final impact of that outcome. So definitely that's a drawback. But I mean, in order to correct for this, we do have this consensus group meeting where we will try, to hear again all the opinions, from patient, parent sides and from expert sides. But definitely, yes, this is one of the weaknesses that the prioritization depends, particularly from parent caregiver side, will depend on what they see with their own child, and that might not be the same all the time.
James, I think we have time for one more question.
Okay. I'll maybe do a practical one then for Emily. A question coming in about, obviously, especially with medical notes and very personal data, confidential data for patients. It's great to hear that we're sharing data with partners and organizations, which I think was a big thing that a lot of parents wanted to see. But what kind of measures are in place to make sure that we only share it with the right partners and we're kind of sensitive to anonymizing data, for example?
This is a great question. I'm glad that you brought it up. So yeah, data that is shared within our patient registry is, first of all, ACD owns that data, and there's only a handful of people within our organization and in the NORD platform that we're hosted upon that have access to your complete set of data. So when I talk about, we've collaborated with with Ultragenyx, or we've shared your data with the variant creation panel, all of that data has been de-identified. And so I have gone through and pulled out any personal information that could potentially be traced back to that participant. And so the data that's being shared is oftentimes just aggregate numbers. And so it's not even any sort of raw data. And so that's one way that it's protected. We also are regulated by the Institutional Review Board to do all of the research that oversees us to ensure that the research that we're doing is done ethically and that we have data protections put into place. So there's a lot of things happening behind the scenes to ensure that your data is secure. And then we're very particular about who we partner with and making sure that they're individuals and researchers that are going to agree that the protection of that data is a priority as well.
Thank you so much. I think this concludes today's sessions. And I'll hand it over to Sangeeta for an overview of tomorrow.
Thank you, Sung. Thanks, everyone. That was a great session. So happy that everyone survived day one. I won't keep you very long. I just have a really quick plug about tomorrow. So join us back, for day 2 at 8 a.m. tomorrow. Format will be much the same. Please, I hope everyone will make every effort to be there. And really quickly, we're interested in hearing from you. So if you could just take the community survey either today or tomorrow, we'd love to hear how we can hear from you and understand how we can better serve you and your family's needs. Here's a quick snapshot of day 2 tomorrow. I'm sure you would have looked at the agenda already, but we have lots of interesting talks on models and gene therapy and the future phase of therapeutic development for CCDS. With that, I will leave you guys today with your thoughts for today, and see you again tomorrow bright and early. Thanks, everyone.